come a bit late, yeah. I think you've met her, Prof. Chris, Dr. Hafiza, yeah, Prof. Tere. The other is Prof. Lucia. I think she's coming. She's actually our veterinary viral, I think, yeah, subspecialist in viral. Not sure which one, the virus. Oh, I see, yeah. The parasitology and virology, yeah. Prof. Lucia is one of our senior here. And this event will be half days. So the first section, we will have all of the speakers here presenting, and we are going to have the QA session. And later, after the Zuhur prayer, we will have the oral presentation competition, yeah, among the participants, yeah. And after that, please be kind, all of the speakers, to have the early dinner with us later, yeah, after the session is over. No further ado, I think. Bismillahirrahmanirrahim. Again, thank you very much for the committee, yeah. Do I forget something? Oh, Prof. Lucia. Prof. Lucia, silakan. Tidak apa-apa, Prof. Mereka. Alhamdulillah. That's it from me. Assalamualaikum warahmatullahi wabarakatuh.
Once again, thank you to Professor Viskasari for that opening speech. Next, it is our pleasure to invite the Director of the Postgraduate School of Universitas Airlangga, Professor Dr. Ahmad Husnu Romdani, to please deliver his opening remarks. To Professor Ahmad Husnu Romdani, the stage is now yours. Please give a round of applause for Professor Ahmad Husnu Romdani.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning and warm greetings to all of you. Our distinguished guest, Professor Insinyur Muhammad Amin Alamsyah, MSc, as a Vice Rector for Academic, Student and Alumni Affairs of Universitas Airlangga, Professor Viskasari Pintoko Kalanjati, MD, MKes, Coordinator of the Master Degree Program in Immunology, Postgraduate School of Universitas Airlangga, and also the Vice Director, Associate Professor Karnaji, thank you for coming. And our distinguished speakers, Professor Dr. Erwin Astatriono, SPPD KPTI Vinasi Mars, yeah, our senior, and right now as head of the East Java Provincial Health Office. Thank you for coming, Prof. Erwin. And Professor Duncan Richard Smith from Mahidol University, Thailand. Professor Dr. Nur Hafiza binti Zainal from University Malaya, Malaysia. Thank you. And also Professor Dr. Adi Idris from Queensland University Technology, Australia. And also our lecturer from Universitas Airlangga, and also the professor, distinguished academic, researchers, clinicians, students, and all participants. Ladies and gentlemen, it is my great pleasure on behalf of Postgraduate School of Universitas Airlangga to warmly welcome all of you to the 5th World Immunology Day International Conference. This year we gather under an important and timely theme about orchestrating the immune system in defense against infection and autoimmune diseases. The immune system is fundamental to human health. It protects us against infection, maintains the balance of the body, and at the same time, when dysregulated, can contribute to inflammation and autoimmune disease. Understanding this complex balance requires not only advance in basic immunology, but also collaboration across clinical, translational, technological, and interdisciplinary fields. This is why a conference such as the 5th WIDIC is so important Today we are bringing together researchers, clinicians, academics, students, and experts from different institutions and countries to exchange knowledge and then share research finding and explore new possibility for collaboration. And I am particularly pleased to welcome our distinguished international speaker from Mahidol University, Thailand, from University Malaya, Malaysia, and Queensland University of Technology, Australia, as well as our national speaker from the East Java Provincial Health Office, and all participants joining us online and offline, on site. Your presence makes this conference truly international and reflects our shared commitment to advancing immunology through collaboration and knowledge exchange. I would also like to express my sincere appreciation to the organizing committee, the immunology program of the postgraduate school, our speakers, partner, and everyone who has contributed to making this conference possible. I hope that WIDIC 5, the fifth WIDIC, will not only provide a valuable scientific forum, but also create meaningful connection that continue beyond this conference, leading to new research collaboration, innovative ideas, and stronger networks among immunologists and health professionals. To all participants, I encourage you to take full advantage of this opportunity, asking questions, exchange ideas, challenge perspective, and most importantly, build connection between us here. Once again, welcome to the 5th World Immunology Day International Conference, and welcome to Universitas Airlangga Surabaya. I hope you have an inspiring, productive, and memorable conference. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Oromdoni, for your inspiring words. Next, it is our honor to invite the Vice Rector for Academic, Student, and Alumni Affairs of Universitas Airlangga, Professor Insinyur Amin Alamsyah, to please deliver his opening remarks and to officially open the 5th World Immunology Day International Conference 2026. To Professor Amin, the stage is now yours. Please give a round of applause for Professor Amin.
Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahirabbil alamin wassalatu wassalamu ala sayyidina Muhammadinil ummi wa ala alihi wa sahbihi ajma'in. Firstly, I would like to express my deep gratitude to Professor Dr. Ahmad Husnul Romdoni, Director of Postgraduate School of Universitas Airlangga, Professor Viskasari Pintoko Kalanjati, Coordinator of the Master Degree Program in Immunology Postgraduate School of Universitas Airlangga, speakers and invited guests from Indonesia and around the world, and then also researchers, clinicians, academics, students, and participants. Good morning and warm greeting to everyone. On behalf of Rector Universitas Airlangga, it's great pleasure to welcome all of you to Universitas Airlangga for the 5th World Immunology Day International Conference 2026, hosted by Postgraduate School of Universitas Airlangga. Ladies and gentlemen, I would like to congratulate the organizing committee for bringing together this important scientific forum under the term orchestrating in the immune system in defense against infection and autoimmune disease. This term highlights one of the most fascinating and important challenges in a modern biomedical science to understanding and how the immune system maintain a delicate balance between protection and dysregulation. Infectious disease continue to pose significant challenge to global health, while autoimmune and immune-mediated disease are increasingly recognized as major contributors to human morbidity and reduced quality of life. Addressing this challenge requires collaboration. No single discipline, institution, or country can provide all the answers. So we need strong connection between basic research and clinical practice, between academia and industry, and between researchers across national and international borders. Therefore, I believe that the 5th World Immunology Day International Conference provides an excellent platform not only for presenting scientific findings, but also for strengthening academic network and creating opportunities for future collaboration. Universitas Airlangga is committed to strengthening research excellence, innovation, and international collaboration, as part of our contribution to addressing global challenge. I would also like to extend my appreciation to all distinguished speakers who have generously shared their expertise, to our participants from Indonesia and abroad, and to the entire organizing committee for their dedication and hard work. To our students and young researchers, I especially encourage you to use this opportunity to learn from the expert, present your ideas with confidence, and establish connection with fellow researchers. The collaboration and conversation that begin here may become the foundation of future scientific discoveries. Finally, I hope that the 5th World Immunology Day International Conference 2026 will be more than a scientific meeting, and may it become a place where ideas are exchanged, partnerships are established, and new direction in immunology are inspired. With great pleasure and on behalf of Universitas Airlangga, I officially declare the 5th World Immunology Day International Conference 2026 open. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Amin, for opening officially the 5th World Immunology Day Conference 2026. Ladies and gentlemen, our next agenda is a group photo session. So the photo session will be conducted in 2 batches, this side and on the further side. So we invite everyone to please stand up in front of their seats, and also to our honored guests to first of all take a picture on the left side of the room. So that will be my right side. All right, ladies and gentlemen, please look at the camera and give your biggest smile. 1, 2, 3. Again, 1, 2, 3. Can we have Uner Hebat? Please put out your fist like this. There we go. 1, 2, 3, and smile. Okay, thank you so much. And we also invite our honored guests to come to the right side of the room to take a photo with the participants on the right side of the room, which will be my left side. All right, ladies and gentlemen, please look at the camera and smile. 1, 2, 3, and smile. Okay, can we have Uner Hebat, please? Please put out your fist. 1, 2, 3, and smile. Okay, thank you everyone, and also to our honored guests. You may now return to your seats. Once again, we thank for the attendance of the Vice Rector for Academic, Student and Alumni Affairs of Universitas Airlangga, and also the Director and Vice Director of the Postgraduate School of Universitas Airlangga this morning to officially open the World Immunology Day International Conference 2026. Okay, ladies and gentlemen, our next agenda this morning is a scientific presentation from the Immunology Master's Program Coordinator of the Postgraduate School of Universitas Airlangga, Professor Viskasari Pintoko Kalanjati, who will give a presentation on the topic of orchestrating the immune system in defense against infection and autoimmune disease. To Professor Viskasari, the floor is now yours. Ladies and gentlemen, please give a round of applause for Professor Viskasari.
thought we are going to have to wait like for another year, yeah. But Alhamdulillah, it is showing, yeah. Alhamdulillah, yeah. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. First of all, I have to make an excuse because there are so many immunology specialists in this room, yeah. My research interest is actually in the neuroanatomy and neuroscience because I'm originally from Faculty of Medicine. My department is anatomy, histology, and pharmacology, subdivision anatomy. So among our participants, most of them are our student and alumni. So this is a bit of introduction, just at a glance, yeah. Orchestrating the immune system in defense against infection and autoimmune disease. Next, please. So, pardon for this, because I am we call it KPS, Coordinator Program Study Program in Immunology. So it is my habit to have the learning objectives and everything. But today's agenda overview will be like exploring the immune system, yeah, which is acting like an orchestra, like multiple cell types, signals, and pathways, yeah, work in harmony to mainly detect danger, eliminate threat, and restore tissue homeostasis while preventing excessive damage. Next, please. So I believe the speakers here will cover in depth, yeah, more in depth about the immune orchestra, as we know that there are innate and adaptive immunity, yeah, in the human's body, and there is a communication between cells and molecules that will ensure the balanced response, yeah. The innate immunity is the first responder, yeah, to any threat that comes inside the human's body. The cellular type usually involve neutrophil, macrophage, and dendritic cells, and also natural killer cells, yeah. Whilst the adaptive immunity, as we already know, yeah, the T cell and B lymphocytes, yeah. The innate immunity as the first line defense, if we are talking about infection per se, yeah, it will generally stimulate the PMPS, yeah. The pattern recognition receptor will link with this PMPS, yeah, through so many type of receptors such as toll-like receptors and everything else, which initiate the transduction of signal and activation of so many pathways, yeah. In this diagram, for example, it will, for example, activating the nuclear factor kappa B, yeah. I was reading it as nuclear factor kappa beta, and one of my colleagues like protesting, yeah. It's not beta, Professor, it's B, actually B cell type, which is, yeah, probably in some of the publication, yeah, NF kappa B, and then it will produce so many pathways involving cytokines, the pro and anti-inflammatory ones, the chemokines, and for the viral, for example, predominantly the interferons, yeah. Of course, if we are talking about the signs and symptoms in the patient, they will generally showing with inflammation signs and symptoms such as fever and some other inflammation signs and symptoms, yeah, like redness, swelling, and everything. Next, please. While the innate immunity, as we already know, yeah, in the first semester, our students learn about the interaction between, for example, dendritic cells, yeah, via the MHC type 2, yeah, and CD4 T cell later as the adaptive immunity effectors. Also the CD8 T cells, yeah, to activate also the humoral immunity via the B lymphocytes, yeah, to produce, for example, immunoglobulins. They are working usually in harmony, yeah, except if there is specific event, such as in immunotolerance and or immunocompromised state. Yeah. The adaptive immunity actually provides specific memorized type and long-term protection, yeah, the response. Next, please. I'm not sure I want to go in details about the cytokine and the next one, yeah, assets such as the TNF-alpha, the interleukin-6. I believe some of our students and the alumni already doing and done the research about these type of cytokines, yeah, in rather different exposures, such as Plasmodium malaria, the last batch, yeah, of our student. They are taking analysis, in-depth analysis about the effect of certain phytopharmaca, such as the bunga telang. I'm not sure the Latin word for bunga telang, I forgot actually. And other herbal phytopharmaca, yeah, against, or rather than its potential as the anti-plasmodium, yeah, anti-plasmodium, and also somehow working via the to enhance the homeostasis between the anti-inflammatory and the pro-inflammatory cytokines. Yeah, this is Iqbal here, yeah, and other students, yeah, taking in-depth analysis on that kind of field experiment, yeah, using animal models. I think Plasmodium berkei angka, yeah. The next one, please. This one, I think our speakers will go in depth about the immune response to infection. Prof. Erwin will be the internist subspecialty in the infection, yeah. Will provide this kind of discussion later, the presentation, yeah, Prof. Erwin. Also the Ketua Dinkes Provinsi Jawa Timur, yeah. I think inshallah will explain in depth, yeah, more than me, yeah, about this pathway. So I'm going to skip a bit, yeah. Next. We know that in the infection, the excessive or prolonged immune activation can damage a host tissue via so many mechanisms, yeah. Examples just years ago, yeah, like 3 years ago, I think, we just survived from the pandemic of COVID-19, and in Indonesia, we're still fighting several infectious diseases, including, sadly, tuberculosis, yeah. And it's not just the lung TB, but also outside lung TB, such as the meningitis and also the spinal TB, yeah, which is totally have different pathophysiology with the lung TB. In the current student batch, actually, Prof. Erwin, we have several students taking the research about biomarkers in tuberculosis patient. I think in several areas, Probolinggo and Pasuruan, where is Januar, yeah, which is very good. Inshallah will help the patient in the future, yeah, at least from the immunological perspective. Yeah. Next, please. This is actually the specialty of my senior, yeah, autoimmunity. We usually, once upon a time, yeah, this study program was built by my teacher, yeah, Prof. Suhartono Taat Putra. His teaching so many students, and now the students already become our senior, yeah. The last coordinator study program, Prof. Tere, is actually one of his students, yeah. She's from the dentistry faculty. Later, inshallah, will join us, Prof. Tere. And she's doing a lot about autoimmunity and immunotolerance, yeah. We know that in the normal tolerance, the self-antigen will be ignored, yeah. But then in the certain condition, there will be the breakdown of tolerance, probably due to genetics or non-genetic factors, which will produce the chronic inflammation and lead to tissue damage and organ dysfunction. Yeah. Next, please. I'm not going to tell you about the pathogenesis of autoimmune disease, as we will. This will be covered, inshallah, by other speakers, yeah, today. Just to know that it will, the genetic susceptibility, of course, we are going to talk about the SLA and the triggers, so many of them, including infection and non-infection. Yeah, today, focus in the infection. Next, yeah. Yeah, 5 more minutes, the MC, yeah. Thank you, yeah. Just skip this, probably to the last slide. No. Before we have the take-home message slide. This is for yesterday, lab methods in human immunology. So on Thursday and Friday, actually, in conjunction with World Immunology Day Conference, we held another workshop. We call it Halimun, yeah. Probably you read the banner in front of our building, yeah. Hands-on laboratory on immunology. We were having the ELISA technique, yeah. Before that, please. Not the last one, before this one. So I want the students in particular, so sorry for the other participants, yeah, to really pay attention to today's topic and learn a lot, yeah. Ask so many questions, yeah. Remember that the immune system is a coordinated network integrating between innate and adaptive immune systems, yeah. And successful defense requires balance, yeah. Effective activation without excessive damage. Please do remember that not all pro-inflammatory cytokines are the bad guys. They are needed in certain level, yeah, in response to so many pathogens, for example, yeah, in the infection event, yeah. In certain cases, your patients, or there are cases, because you are not from medical faculty, yeah, some of you in the vet, some of you in biology and everything, some of you in the dentistry. Remember, there is special cases of autoimmunity, immunotolerance, and immunocompromise, which will which may significantly affect, yeah, how the body responds to these infection pathogens. Do remember the basic pathophysiology. There is not only infection, actually, that can provoke inflammation. So many other underlying mechanisms can provoke inflammation, such as neoplasm, we call it cancer, yeah. Vascular disruption, congenital abnormality, yeah, and probably the mechanical trauma, and anything else you can read it by yourself in, for example, the pathophysiology textbook, yeah. So today we held this conference to learn, yeah, to understand the immune orchestration that guides the treatment of infection in particular, yeah. for today's topic, yeah, and also a bit about autoimmune disease, and the most updated, not therapeutic, yeah, but the management. Remember, the therapy is only a part, but the management of the cases is the one that I think holding a more comprehensive method, yeah, to decrease the adverse effect of infection and autoimmunity effect, yeah, especially in humans, yeah. I think that's all from me, because my section is a short one, just to open the pathway, yeah, so the students can understand what you will, not only the students, yeah, for also the all participants, what you are going to learn together, yeah. I am also going to learn, yeah. We are going to learn together with other speakers, yeah. Please ask many questions, yeah. Jangan malu-malu adik-adik, yeah. If you cannot speak in English, that is totally fine. You can ask in bahasa. Just don't ask in other language, because we don't have any other translator here. For example, Arabic or French, I cannot. Yeah, Prof. Amin can speak French. I cannot. Huh? Okay? Don't speak in Thailand. Yeah, I cannot speak in Thai. Yeah. Okay. I forgot to tell you this morning. Although the committee, I know, has already worked so hard, yeah, to prepare everything for this. Thank you very much. As the head coordinator, we realized there will be probably so many lacks, yeah. Please do apologize, yeah. Any inconvenience, late something and something, please do apologize. But let's work in the positive energy, so our immune system can perform well, yeah, not only today, but in our daily life, inshallah, yeah, amin. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Viskasari, for that insightful scientific presentation. Ladies and gentlemen, our next agenda is a series of keynote lectures that will be given by our expert speakers. And before we go into that, I would like to remind all participants who are joining us offline or online to be active during the Q&A and ask any questions, such as what Professor Viskasari has already noted. Because as she has said before, the immune system is a complex network of active and innate systems, and we are here to learn together. Ask your questions and don't be shy. Ladies and gentlemen, we will now begin our first keynote lecture, and the first and second keynote lecture today will be moderated by Dr. Niluh Ayu Megasari. Please join me in welcoming Dr. Niluh Ayu Megasari as she moderates today's keynote sessions.
Thank you very much, Mas Nabil, as our MC. Okay, good morning, ladies and gentlemen. So our first speaker for the World Immunology Day International Conference 5 2026 will be Professor Dr. Erwin Astatriono. Good morning, Prof. Selamat pagi. Yeah, Prof. Erwin is an internist and consultant for tropical infectious diseases, and also master in hospital management. Yeah, Prof. And Prof. Erwin is currently served as the head of East Java's Provincial Health Service and has a long professional experience as both clinician and also lecturers. Okay. So Prof. Erwin will be delivering his keynote lecture about precision medicine in infectious disease. Ngapunten Prof untuk waktunya 40 menit nggih Prof. Kami mohon izin mengingatkan nanti. Bisa lebih lama diskusinya gitu, Prof. Yeah. Without any further ado, the time is yours, Prof. Erwin. Monggo, Prof.
Ya, baik. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Good morning. Best wishes to all of us. Yang pertama-tama terima kasih Prof. Viska yang telah mengundang kami. Meskipun ini perjuangan karena ya sebagai kepala dinas didorong untuk paham tentang ilmu yang sedikit kompleks ya terkait dengan imunitas. Terima kasih kawan-kawan panelis dan moderator, and welcome to distinguished speaker from Thailand, Malaysia, Australia, Prof. Smith, Bu Nur Hafiza, and Mr. Hadi Idris. Ya. Bapak Ibu yang hadir dan para mahasiswa, ya, baik dari dalam negeri maupun luar negeri yang ikut secara online maupun offline. Please accept my apologize because I will be presenting in Bahasa, but don't worry, I have prepared the presentation materials in English. Ya. Bapak Ibu yang kami hormati, slide berikutnya, next slide. Ini tawar menawar antara panitia dengan kami, ya. Karena kalau kita nanti fokus imunitas, ya itu pasti penguasaannya ada di Profiska. Sehingga saya sering, ya kurang dekat ya. Ya, terima kasih. Sehingga kami berusaha untuk memposisikan, menghubungkan antara klinisi sebagai internis, kemudian ya sedikit ditambahi dengan isu terkait dengan imunologi, dan nanti bisa dikaitkan dengan kebijakan, karena gimana pun nanti imunologi jangan berhenti pada ilmunya, tapi lebih cenderung gimana cara mengimplementasikan di lapangan. Slide berikutnya. Bapak Ibu yang kami hormati, ya ini adalah tantangan kita semua Bapak Ibu. Saya hanya menunjukkan 5 poin ya, 5 poin setting lapangan yang kadang-kadang kita harus memahami tentang isu infeksi terkait dengan imunologi yang ada di masyarakat. Nomor 1, sepsis. Sepsis ini menjadi tantangan karena salah satu penyebab kematian terbanyak di Indonesia. Jadi kalau kita mau jujur, 3 besar penyakit yang ada di masyarakat saat ini adalah keganasan yang terus meningkat, kemudian metabolic syndrome yang terus meningkat, dan sisanya infeksi. Infeksi ini yang paling penting yang perlu kita pahami adalah isu tentang sepsis. Yang kedua adalah emerging and re-emerging infectious disease. Ini menjadi tantangan. Karena dengan era perubahan iklim, perubahan cuaca dan sebagainya membuat penyakit-penyakit yang kadang-kadang mungkin dulu nggak ada menjadi ada, atau yang dulu sudah dilupakan menjadi muncul lagi. Ya. Yang terbaru misalkan hantavirus, Ebola, ini menjadi isu penting. Yang ketiga tadi sempat disampaikan secara imunologi oleh Prof. Fiska terkait dengan TBC. TBC ini menjadi tantangan utama karena 72 tahun Indonesia melakukan manajemen TBC, tapi ternyata enggak pernah tuntas. Saat ini kita menempati ranking 2 di dunia setelah India. Ya. Jawa Timur, mohon maaf, menempati ranking 2 setelah Jawa Barat. Apalagi sekarang sudah mulai bergeser ke isu multi-drug resistant of tuberculosis. Ini menjadi tantangan baru. Ya. Karena pengobatannya lebih susah dan adherence atau kepatuhannya juga menjadi tantangan tersendiri. Yang gak kalah pentingnya Bapak Ibu semuanya tentang antimikrobial resistant, AMR. Ini menjadi hal yang khusus. Kenapa khusus? Karena di negara-negara berkembang ini menjadi isu utama, ya, tapi seringkali diabaikan. Dalam tanda kutip, penggunaan antibiotik seringkali tidak menyesuaikan dengan kaidah-kaidah pilihan antibiotik yang terbaik. Selalu dikatakan bahwa yang terbaik dianggap yang terbaru, terluas spektrumnya, maupun yang termahal. Padahal antibiotik terbaik adalah yang sesuai dengan pola kuman rumah sakit. Tugas kita lebih cenderung gimana caranya menggunakan antibiotik yang baik sehingga bisa mencegah atau menghambat timbulnya resistensi antimikroba. Yang kelima Bapak Ibu semuanya terkait dengan model diagnostik. Ya. Kadang-kadang kita disuruh buat pilihan, apa diagnostik yang terbaik? Kalau terkait dengan mikroba, maka kultur menjadi isu terbaik. Yang jadi masalah seringkali kultur butuh waktu lama. Dan kadang-kadang terkait dengan era BPJS jadi nggak menarik. Kenapa? Karena paketnya nggak cukup kalau mau dilakukan kultur. Ini menjadi tantangan. Sehingga harapan kami nanti isu imunitas, isu infeksi, ternyata nggak mudah dicocokkan dengan isu kebijakan kendali biaya. Sehingga tantangannya ada di sebelah kanan. Dulu one size fits all. Jadi dalam tanda kutip sekarang juga ya, ini dulu dan sekarang. Sekarang kita ngobati itu seakan-akan dibuat sama antar individu. Ya. Seakan-akan bahwa respon terhadap terapi yang diberikan dianggap sama. Ya. Ini menjadi tantangan. Nanti kita tunjukkan di setting slide berikutnya di mana respon masing-masing individu terhadap obat, penyakit dan dosis yang sama ternyata responnya bisa berbeda. Ini menjadi kata kunci terkait dengan pentingnya farmakogenomik sebagai upaya kita untuk kita sosialisasikan lebih gencar lagi. Ya. Slide berikutnya. Yang jadi pertanyaan berikutnya adalah mengapa respon terapi itu bisa berbeda-beda antar pasien. Di situ tantangan besar dalam praktek klinis modern, ya, di mana variabilitas respon pasien terhadap obat yang variabilitasnya berbeda terhadap obat yang sama. Jadi dua pasien dengan pengobatan yang sama diberikan obat dosisnya sama, diagnosisnya identik, ternyata bisa memberikan hasil yang berbeda. Mohon maaf, satu sembuh, satu enggak efektif, satu malah timbul efek samping. Ini situasi di lapangan. Ya. Jadi fenomena ini bukan suatu kebetulan, tapi melainkan cerminan dari kompleksitas biologis yang mendasari setiap individu. Kita lihat di situ variabilitas yang sering terjadi. Hanya 30-50% aja yang responnya bagus. Sehingga seringkali kita berhadapan sama pasien 50% nggak ada masalah, no problem. Tapi sisanya gagal terapinya, atau efek samping yang muncul, atau interaksi obat. Dan yang terakhir adalah alergi atau intoleransi yang akhirnya membuat kepatuhannya jadi rendah. Dan ini terjadi terkait dengan TBC, ya, di mana dengan intoleransi kepatuhan yang rendah memberikan kontribusi resistensi yang muncul. Apa yang menyebabkan? Memang banyak faktor di situ, ya. Ada usia, ada fungsi organ, ada komorbid, ada interaksi obat. Tapi yang saat ini kita lagi gaungkan adalah sering diabaikannya variasi genetik antar individu. Ya. Jadi gen-gen yang mengkode enzim metabolisme obat, transporter, reseptor target ternyata memiliki polimorfisme yang membuat setiap orang memetabolisme, merespon obat itu secara unik, bisa beda-beda. Ya. Dan ini mengapa pendekatan one size fits all relatif sudah tidak memadai lagi. Ini yang nanti kita berikan penekanan terhadap isu hari ini. Slide berikutnya. Isu terkait dengan farmakogenomik banyak kita kaitkan dengan precision medicine, ya. Jadi kita ingin mempelajari bagaimana profil genetik setiap individu dalam menentukan respon terhadap terapi. Sehingga harapan kami kita semua terapi lebih aman, efektif, personal, dan bisa memberikan manfaat jauh lebih baik ke masing-masing pasien kita. Slide berikutnya. Farmakogenomik memang diindikasikan untuk pengobatan yang lebih tepat. Ya. Definisinya adalah ilmu yang mempelajari pengaruh variasi genetik seseorang terhadap respon obat, sehingga kita semua nanti dapat informasi terkait dengan efektivitas dan keamanan terapinya. Ya. Tujuannya clear, supaya kita dapat obat yang paling sesuai, paling optimal, resikonya lebih sedikit dan mendukung precision medicine. Ya. Slide berikutnya. Bapak Ibu yang kami hormati, ini adalah pertanyaan yang relatif lebih fokus lagi. Kenapa farmakogenomik diperlukan? Artinya pengobatan berbasis genetik kenapa diperlukan? Kita lihat di situ bahwa respon pasien terhadap obat berbeda karena variasi genetik. Ada pasien yang diberikan obat A dengan dosis tertentu, obat bekerja optimal. Obat bekerja optimal, ya. Sehingga efek terapi maksimal, dosis sesuai. Ada juga pasien yang justru obatnya kurang efektif. Apa hasil akhirnya kalau kurang efektif? Maka perlu penyesuaian dosis. Yang ketiga bisa terjadi malah efek samping. Apa yang terjadi? Kepatuhan minum obat juga bermasalah. Jadi kesimpulan slide ini adalah obat yang sama belum tentu memberikan hasil yang sama pada setiap pasien. Pemeriksaan farmakogenemik membantu menentukan terapi yang lebih efektif, aman dan sesuai dengan profil genetik masing-masing pasien. Dan ini adalah gen yang sering berperan dalam farmakogenomik. Ya. Bisa terkait dengan metabolisme obat, bisa terkait dengan transport, bisa terkait dengan metabolisme obat tertentu. Slide berikutnya. Kalau kita fokuskan terkait dengan farmakogenomik di bidang infeksi, prinsipnya relatif sama. Jadi permasalahan yang paling utama adalah tidak semua pasien memberikan respon yang sama. Kadang-kadang dosisnya bisa berbeda. Dosis yang sama memberikan respon yang berbeda. Efek sampingnya muncul. Apa penyebabnya? Karena perbedaan genetik. Setiap individu bisa mempengaruhi tubuh untuk menyerap obat. Memetabolisme, mengantarkan maupun mengeluarkan obat. Sehingga farmakogenomik pada prinsipnya membantu kita semua dalam memilih obat dan dosis yang sesuai dengan profil genetik dari pasien. Dan ini bisa kita kaitkan dengan obat-obat di aspek infeksi. Di situ bisa antibakteri, antiviral maupun obat-obat untuk anti jamur. Ya. Bagaimana obat ini bisa mempengaruhi respon? Bagaimana gen bisa mempengaruhi respon terapi? Ini ada kata kunci yang kita perlu kenali, di mana setiap orang memiliki variasi kecil pada DNA yang disebut dengan single nucleotide polymorphism, SNP, ya. Di mana perubahan pada genetik ini bisa memberikan dampak terhadap kecepatan metabolisme, kadar obat dalam darah, efektivitas maupun efek samping. Sehingga outcome yang dimungkinkan adalah ada individu yang mendapatkan poor metabolizer, intermediate, normal maupun fast metabolizer. Ya. Slide berikutnya. Ini adalah contoh kasus. Ya. Contoh kasus di mana kalau pasien itu didapatkan metabolisme yang kurang baik atau rendah, kalau tanpa pemeriksaan genetik testing, farmakogenomik, maka dia akan diberikan dosis standar. Begitu diberikan dosis standar, maka karena metabolismenya rendah, maka terjadi akumulasi obat yang hasil akhirnya toksik. Tapi kalau kita bisa lakukan pemeriksaan dengan farmakogenomik, maka harapannya dokter paham bahwa pasien ini adalah metabolisme lambat, sehingga dosisnya bisa dikurangi atau obatnya bisa diganti, harapannya lebih baik. Sehingga kesimpulan pada slide ini menunjukkan bahwa variasi genetik pada setiap tahap dari metabolisme, konjugasi maupun transport akan memberikan kemungkinan obat kadarnya terlalu besar atau terlalu sedikit, terlalu cepat, atau justru malah efek samping. Sehingga farmakogenomik terhadap infeksi ini digunakan untuk mempersonalisasi terapi infeksi sehingga pasien memperoleh obat yang tepat, dosis yang tepat, waktu yang tepat, dan profil genetik yang sesuai dengan profil genetiknya. Slide berikutnya. Ini adalah 4 pilar dari farmakogenomik. Ya. Pada prinsipnya farmakogenomik ini akan membantu para klinisi ya, untuk membuat keputusan terapi yang presisi berdasarkan 4 elemen kunci yang saling melengkapi. Dan pendekatan ini akan menggeser paradigma dari pengobatan reaktif menjadi prediktif, preventif, dan lebih personal. Obatnya lebih tepat, dosisnya lebih tepat, pasiennya lebih tepat, dan waktunya bisa lebih tepat. Jadi variasi genetik terutama pada single nucleotide polimorfisme dapat mengubah cara tubuh memproses dan merespon obat secara fundamental. Slide berikutnya. Ini adalah manfaat yang kita harapkan. Ini hanya contoh di mana kalau ada pasien-pasien dengan infeksi dengan farmakogenetik study, farmakogenomik, dan ada faktor-faktor non genetik, maka harapannya jenis obat maupun dosis bisa menyesuaikan sehingga harapannya pengobatannya lebih aman, perjalanan klinisnya lebih menguntungkan, lama rawat inapnya bisa lebih pendek, dan layanan kita jauh lebih maksimal. Slide berikutnya. Ini adalah contoh Bapak Ibu semuanya. Misalkan kita bicara tentang efavirenz dan peran dari gen CYP2B6 ya. Kita tahu bahwa efavirenz ini tugasnya sebagai obat untuk HIV, di mana dia punya tugas atau termasuk dalam golongan non-nucleoside reverse transkriptase inhibitor. Ya. Dia punya tugas untuk nyetop reverse transkriptase sehingga perjalanan virus ini menjadi berhenti replikasinya. Kenapa bisa berbeda-beda responnya? Jadi di setting lapangan ada pasien yang dengan dosis tertentu efek sampingnya muncul. Ada yang nggak ada masalah. Ada yang setelah itu timbul resistensi. Ini yang jadi masalah. Karena kadar di dalam darah itu kadang-kadang bisa berbeda. Dan penyebab utama yang kita ingin sampaikan yaitu variasi genetik pada gen CYP2B6 menghasilkan enzim yang memetabolisme efavirenz di dalam hati. Ya. Jadi efektivitas dan keamanan sangat tergantung pada gen ini. Artinya apa Bapak Ibu? Nomor 1, kalau misalkan ternyata variasi gen itu memberikan dampak poor metabolisme lambat, maka resikonya kadar obat dalam darah akan meningkat. Maka toksisitas akan muncul. Kalau normal nggak ada masalah, manfaat dan toksisitasnya relatif aman, tapi yang rapid justru membuat ini yang kadar dalam obat dalam darah turun sehingga menyebabkan isu resistensi. Slide berikutnya. Gimana dengan pengobatan anti jamur? Voriconazole. Voriconazole ini prinsipnya obat yang bagus untuk jamur, khususnya untuk aspergillosis, kandidiasis, dan infeksi jamur sistemik lainnya. Ya. Pada prinsipnya dia akan memberikan dampak meskipun dosis sama, respon sama pada setiap pasien, itu bisa memberikan dampak yang berbeda. Penyebab utamanya adalah variasi genetik pada gen CYP2C19 yang mempengaruhi metabolisme voriconazole di dalam hati. Ya. Jadi keberhasilan terapi voriconazole ini tidak hanya ditentukan oleh dosis obat, tapi juga sebetulnya pengaruh dari variasi genetik pasien tersebut. Sehingga sama, dampak yang bisa kita dapatkan kalau kita bisa memahami pemeriksaan variasi genomik tadi, maka kita bisa tahu bahwa pasien itu termasuk golongan metabolisme lambat, intermediate, normal, atau cepat. Dan nanti banyak dikaitkan dengan dampak dari manfaat maupun toksisitasnya. Artinya yang metabolisme lambat, maka resiko obat kadar dalam darah tinggi sehingga menyebabkan toksisitas. Slide berikutnya terkait dengan aminoglycoside juga sama, tapi ini dikaitkan dengan efek samping. Ya. Aminoglycoside bisa terdiri dari gentamisin, amikasin, streptomisin, neomisin maupun tobramisin. Ya. Pada prinsipnya ribosom mitokondria pada sel rambut di koklea itu mirip dengan ribosom bakteri. Ya. Karena mirip, sehingga ada variasi pada gen MT RNR1 pengkode 12S rRNA akan meningkatkan ikatan imunoglikosida pada ribosom mitokondria. Ini yang membuat adanya variasi ini memberikan dampak efek samping oktotoksisitas. Ya. Jadi variasi itu yang membuat kita akhirnya lebih hati-hati dalam memilih penggunaan aminoglikosida pada kelompok pasien yang punya variasi gen tersebut. Artinya pada pasien yang punya variasi gen pada MTRNR1 akan mudah terjadinya ototoksisitas. Slide berikutnya, yang gak kalah pentingnya adalah obat-obat TBC. Ini yang tadi sempat dibahas secara imunologi oleh Prof. Vizka. Ini obat TBC yang gak tergantikan oleh obat yang lain. Ya. Dan saat ini ada sedikit tantangan karena isoniazid juga dipakai untuk terapi profilaksis tuberkulosis. Ini yang jadi masalah. Karena biasanya obat-obat yang dipakai untuk profilaksis cenderung mudah sekali timbul resisten, sehingga hasil akhirnya malah menjadi hal yang minus, karena pengobatan dengan niena ditakutkan nanti memberikan kontribusi resistensi yang meningkat. Tantangannya adalah pada gen NAT2, ya, yang mana dia perannya pada metabolisme isoniazid. Artinya pada kelompok slow asetilator, maka siap-siap kadar obat dalam darah akan meningkat dan resiko toksik akan meningkat. Pada yang fast, maka kadar obat dalam darah relatif rendah dan justru malah memberikan kontribusi kemungkinan resisten karena kadar obat dalam darah relatif berkurang. Ini tantangan kita di lapangan kalau kita tidak terbantu dengan data salah satunya dari farmakogenomik. Slide berikutnya. Ini adalah manfaat yang nanti bisa kita dapatkan pada saat kita melakukan implementasi klinikal, farmakogenomik. Harapannya pada pasien terapinya lebih efektif, reaksi efek sampingnya lebih sedikit, pengobatannya relatif personalized, dan menghindari trial and error. Karena pengobatan khususnya dengan antibiotik yang bisa kita lakukan di lapangan seringkali kita berikan terapi empirik, evaluasi 3 hari, kalau nggak ada respon kita segera pikirkan apakah ini harus diganti antibiotiknya atau dosisnya yang diperbaiki. Dengan farmakogenomik diharapkan kita bisa mengurangi trial and error. Untuk tim klinisi profesional maka membantu membuat keputusan klinis, ya, meningkatkan patient safety dan mengoptimalkan penggunaan obat-obat, dan yang paling penting adalah evidence base, ya. Jadi kita mendorong para klinisi untuk mengimplementasikan pilihan-pilihan pengobatan yang berbasis bukti untuk mengurangi resiko-resiko ini, sehingga dampak klinis yang bisa kita dapatkan adalah keputusannya jauh lebih presisi, outcome-nya jauh lebih baik, dan keselamatan pasien bisa lebih diperbaiki. Slide berikutnya. Bapak Ibu yang kami hormati, tantangan berikutnya adalah gimana mengkaitkan antara kendali mutu yang kita bahas tadi, karena mutu layanan kan salah satu penopang yang kita bicarakan adalah farmakogenomik, dengan kendali biaya, di mana itu dipresentasikan oleh teman-teman dari BPJS. Kuncinya adalah membridgingkan antara dua isu. Area medis itu lebih banyak area abu-abu. Area abu-abu itu artinya kadang-kadang kita nggak bisa berlaku hitam putih untuk menentukan diagnostik tertentu. Masalahnya BPJS dalam tanda kutip pembayarnya itu senengannya yang hitam putih. Ini yang menjadi upaya kita untuk membridgingkan antara kendali mutu, kendali biaya dalam bentuk clinical guideline.
Ngapunten Prof, 10 menit lagi nggih Prof.
Slide berikutnya. Ini adalah upaya kami. Diharapkan dengan integrasi farmakogenomik infeksi pada guideline, diharapkan betul-betul bisa membalansing antara kendali mutu, kendali biaya. Karena kita hanya fokus kendali mutu tanpa memperhatikan biaya, maka tidak adil juga. Tapi kalau hanya fokus kendali biaya tanpa mutu, jadi masalah kembali. Maka dua-duanya menjadi isu yang nanti ke depan perlu kita bridgingkan. Slide berikutnya. Ini adalah visi dari precision medicine. Ini bukan hanya slogan, tapi sebetulnya mengubah cara berpikir kita, ya, supaya menggantikan pendekatan trial and error menjadi keputusan terapi yang dipandu dengan bukti klinis, clinical evidence, kemudian data genetik, sehingga terapinya lebih presisi, efektif, dan personal. Dulu atau saat ini kedokteran konvensional menunggu pasien dan melakukan monitoring, tapi dengan precision medicine maka kita bisa memprediksi respon sebelum terapi dimulai, sehingga bisa menghemat waktu, biaya, dan beban pasien. Dari populasi ke individu, dulu kita membuat kebijakan untuk general, tapi sekarang kebijakannya by patient, ya. Setiap pasien adalah individu yang unik, ya, dengan profil genetik yang tidak dapat digeneralisasi. Dan ini menjadi tantangan cara membacanya. Kelihatannya cukup mahal untuk farmakogenetik ini, tapi ternyata bisa mengurangkan biaya akibat kegagalan terapi, rawat inap berulang, penanganan efek samping, dan dalam jangka panjang bisa mengurangi dampak yang lebih serius. Slide berikutnya. Ini adalah salah satu studi di mana dengan menerapkan farmakogenomik kita bisa mengurangkan efek sampingnya. Manfaatnya bisa 2 kali lipat, ya. Tadi udah kita sempat bahas, terapi konvensional 50% aja yang tepat bisa memberikan dampak terbaik. Dengan farmakogenomik diharapkan bisa lebih baik lagi. kita bisa berhemat anggaran, dan yang paling penting ini kalau dari data FDA ada 70% obat yang sudah mendapat tambahan informasi tentang farmakogenomik. Tantangannya Bapak Ibu semuanya, ketersediaan tes. Gak banyak lab yang bisa menyediakan ini. Yang kedua, edukasi klinis. Kita perlu memberikan edukasi kepada para dokter, kemudian apoteker dan sebagainya dalam menerapkan farmakogenomik. Kemudian integrasi dengan electronic medical record. Dan yang paling penting adalah regulasi. Karena ini menyangkut data-data yang sensitif. Ya. Tantangan ke depan terutama untuk teman-teman yang suka penelitian, ya. Ini memang mulai teknologi next generation sequencing mulai lebih banyak, ya. Moga-moga harganya bisa lebih turun. Kemudian cara membacanya bisa dibantu dengan artificial intelligence. Dan yang paling penting yang nomor 3 ini, populasi Indonesia itu masih terbatas. Yang penelitian farmakogenomik terhadap populasi Asia Tenggara itu masih terbatas. Ini kesempatan kita sebetulnya untuk bisa leading terkait dengan upaya precision medicine. Dan yang terakhir adalah policy. Slide berikutnya. Ini kesimpulan kami Bapak Ibu. Ini merupakan transformasi dari diagnosis yang sifatnya umum, kemudian terapinya uniform, menjadi terapi yang personalized, lebih presisi dan seterusnya. Sehingga diagnosisnya lebih cepat, lebih tepat, pengendalian infeksinya bisa lebih efektif. Slide berikutnya, sebagai penutup, pharmacogenomic is one of the key pillars of precision medicine. Ya. Ini by integrating gene information into clinical decision making, therapy can become safer, more effective, and tailored to the biological characteristic of each individual. This approach is expected to become an essential component of the future healthcare system. Safer, personalized, more effective, and the future ini menjadi standar baru, new standard dalam pelayanan kesehatan of healthcare dengan mengintegrasikan genomik ke dalam setiap keputusan klinis. Jadi genomic information is integrated into routine clinical decision making. Langkah selanjutnya, next step, kita mempertimbangkan untuk mengintegrasikan tes farmakogenomik dalam praktek klinik, khususnya untuk obat-obat dengan jendela terapeutik sempit atau faktor resiko yang serius. Collaboration among clinician, pharmacist, yang ahli genetik, laboratori spesialis, and healthcare policy maker, saya kira perlu dimaksimalkan. Itu yang bisa kami sampaikan. Terima kasih, semoga bermanfaat. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Professor Erwin, for the lecture, the talk about precision medicine. Ya, Bapak Ibu, kami persilahkan apabila ada pertanyaan yang ingin disampaikan kepada Prof. Erwin. Karena waktunya terbatas, bisa langsung angkat tangan, ya, Bapak Ibu. Oke, ada satu dari sisi sana. Oke, Pak. Kita 2 penanya dulu, kalau masih ada waktu akan kita tambah sesi tanya jawabnya. Seperti itu nggih, Prof. Erwin. Silakan Bapak diperkenalkan dirinya dan pertanyaannya nggih.
Okay, thank you, Prof. Erwin, for your talk. I'm Gergoris Kinamaran, the freshman student from Master in Immunology program. So, about your presentation, I have one question. So given Indonesian population diversity across geography and also nutritional status, how do you feel the variation in immune competence between this population could affecting the national infectious disease control strategies let's say Indonesia, we know that we still work with the one-size-fits-all approach in health policy. For example, the vaccine implementation. Is it still relevant, or maybe is population-based immunological stratification is needed? Thank you.
Pertanyaan dari Bapak Gregorius, jadi karena karakteristik demografik di Indonesia ini kan sangat beragam, apakah approach one size fit all untuk penerapan kebijakan baik yang kesehatan, misalnya vaksin seperti itu, Prof, apakah masih relevan gitu, Prof, ataukah ada perlu penyesuaian seperti itu ya, Pak Gregorius ya? Oke, boleh satu pertanyaan lagi nanti dijawab berbarengan seperti itu nggih, Prof. Silahkan, monggo diperkenalkan dirinya dahulu.
Baik, terima kasih banyak Ibu Ayu. Thank you very much for your kesempatan untuk saya. Di sini mungkin saya izin menggunakan bahasa Indonesia untuk bertanya kepada Prof. Erwin terkait pharmacogenomics sebagai, di sini mungkin saya melihatnya sebagai personalized therapeutic gitu, Prof ya. Jadi memang terapi yang disesuaikan dengan keadaan seseorang. Jadi di sini pharmacogenomic ini merupakan advantage untuk supporting diagnosis. Tetapi di sini melihat realitanya khususnya pada pasien-pasien penerima BPJS, mereka masih memiliki yang namanya sistem referral atau rujukan. Jadi pertama ke vaskes pertama dan seterusnya, sehingga mereka bisa mendapatkan ke spesialis gitu ya. Sedangkan kalau di sini melihat yang pharmacogenomics ini sepertinya masih belum bisa diimplementasikan di vaskes pertama tadi karena limited facilities. Mungkin di sini saya izin bertanya sebagai untuk tata kelolanya, mungkin dari saran dari Prof bagaimana kedepannya pharmacogenomics ini yang amat sangat banyak benefitnya tadi ya bisa diimplementasikan di semua kalangan, bahkan di vaskes pertama tadi. Dan di sini juga ada tadi menyebutkan 4 fundamental dari pharmacogenomics, yaitu right time ya Prof. Nah untuk right time-nya ini bagaimana? Mungkin bisa dispesifikan mungkin Prof terkait seperti apa right time untuk diagnosa menggunakan pharmacogenomics ini, karena umumnya ya untuk mayoritas di Indonesia para pasien itu khususnya suka yang sesegera mungkin gitu, Prof. Jadi mungkin itu saja pertanyaan saya terkait bagaimana tata kelola dan kebijakan yang bisa digunakan terkait pharmacogenomics ini ke depannya. Terima kasih.
Baik, monggo Prof langsung menjawab 2 pertanyaan dari 2 penanya, nggih, monggo Prof.
Ya, terima kasih. Ilmu infeksi itu nggak lepas dari triangle epidemiologi ya. Ada agent, host, dan environment. Sehingga untuk menyelesaikan masalah 3 ini, tetap 3 hal ini harus dimaksimalkan. Sakit ya tergantung 3 triangle ini, proses kesembuhan juga menyangkut 3 triangle ini. Terkait dengan keberagaman masyarakat Indonesia, kadang-kadang pertanyaan awamnya itu seperti ini. Suka enggak kita hidup di daerah tropis? Jawabannya pasti suka. Tapi kita takut-takut sedikit. Daerah tropis merupakan daerah yang kaya sekali. Kuman apa yang tidak bisa tumbuh? Semuanya bisa tumbuh. Mau bakteri, mau virus, mau parasit, mau jamur, ini menjadi masalah. Dan masalah ini cenderung menjadi tantangan kita. Kalau kita pada kondisi imunokompeten nggak ada masalah, karena imunitas kita udah cukup. Tapi begitu masuk pada kondisi imunokompromis, ini jadi masalah. Sehingga pasien-pasien dengan HIV, dengan chronic disease itu menjadi tantangan pada kondisi hidup di Indonesia dengan banyaknya kuman. Yang menjadi tantangan berikutnya, gimana caranya supaya kita bisa menyelesaikan masalah ini? Secara individu 2 pilihannya. Kita mau sakit setelah itu timbul antibodi, khusus untuk yang imunokompeten, atau kita mau vaksin supaya kita muncul antibodi. Ini menjadi pilihan. Pada saat COVID, nggak sengaja dua kelompok ini muncul. Nggak sengaja. Kelompok pertama adalah kelompok, mohon maaf, teman-teman Madura, yang saat itu nggak suka dengan vaksin. Tapi begitu nggak divaksin, banyak yang sakit tapi underreported, tahu-tahu diperiksa zero surveillance-nya, antibodinya tinggi-tinggi di sana. Artinya apa? Pilihan mereka adalah sakit untuk mendapatkan antibodi terbaik. Kebetulan di sana imunokompetennya cukup banyak. Tapi pilihan kedua yang dipilih oleh negara adalah vaksin. Sehingga bagi kami vaksin itu adalah salah satu upaya untuk menyelesaikan masalah selevel preventif promotif. Ya. Sedangkan kalau sudah masuk pada tahap kita masuk kelompok imunokompromis, personal medicine ini sangat diperlukan supaya kita tidak masuk dalam kategori penyakit yang lebih berat yang notabene kegagalan terapi atau toksisitas atau terapi yang tidak adekuat. Ya. Jadi kombinasi ya antara upaya untuk preventif promotif melalui vaksin dan upaya farmakogenomik untuk membuat terapi kita jauh lebih baik lagi. Kemudian yang kedua terkait dengan isu gimana cara ngeklopkan antara farmakogenomik yang harganya relatif mahal dengan BPJS yang nilainya dalam tanda kutip sangat berhemat kan gitu. Memang perlu komponen ketiga yang notabene nggak bisa kita paksakan hari ini harus kita kerjakan. Maka tahap yang kita usulkan adalah tahap, kalau istilah bahasa budgeting itu, kalau ada kebijakan cost sharing. Cost sharing itu artinya ya BPJS nilainya segitu nggak ada masalah, tapi begitu ada person yang mungkin punya anggaran berlebih, silahkan mainkan sendiri ya cost sharing. Karena harganya ini kalau dihitung-hitung secara swasta itu masih cukup mahal. Tapi tadi udah kita sampaikan, mahalnya ini sebetulnya nyucuk sesuai dengan manfaatnya. Kalau pada saat kita menemukan pasien dikasih obat ini, kok efek sampingnya cukup tinggi. Kita mau ganti obat, sensitivitasnya nggak sesuai. Maka kita tetap harus pakai aminoglycoside misalkan. Maka data ini yang kita butuhkan. Ya. Jadi nggak mudah memang untuk membridgingkan antara kendali mutu, kendali biaya, tapi inilah salah satu pintu masuk kita untuk mulai berproses. Ya. Karena pusat sendiri juga mulai mengumpulkan data ini terkait dengan genome sequencing-nya yang nanti bisa kita polakan. Ya, kira-kira orang Indonesia suku mana itu seperti apa pola genetiknya. Dan kalau itu bisa terrealisasi dengan bagus, maka ke depan kita bisa melangkah lebih jauh meskipun masih perlu diperjuangkan. Terkait dengan right time, itu memang ada beberapa obat. Saya harus membaca lebih detail karena hari ini kita sekarang belajar tentang infeksi. Sedangkan obat-obat yang kita bicarakan sekitar 14 obat yang masuk dalam farmakogenomik infeksi memang nggak ada kaitan langsung dengan right time-nya. Tapi ada obat lain di luar. Karena farmakogenomik ini yang paling pesat itu perkembangannya pada obat-obat keganasan. Itu hampir 50% yang tersedia itu mengarah pada obat keganasan farmakogenomik. Kemudian ada metabolik, ada neuro, ada psikiatri, ada infeksi. Itu masing-masing sekitar 7-10%. Mungkin ada kaitannya dengan obat-obat kemoterapi yang terkait dengan right time. Cuma nanti saya harus baca karena itu menyangkut kompetensi yang berbeda. Terima kasih.
Thank you very much for the answers, Prof. Erwin. So there will be so many challenges in applying precision medicine, especially in Indonesia, and especially on the policy side, considering the vision of the precision medicine, so right drug, right dose, right patient, and right outcome. We hope that the precision medicine will be more accessible to the wider audience in Indonesia in the near future ya Prof. Terima kasih banyak Prof. Erwin. Boleh tepuk tangan. Can we give a big applause for Prof. Erwin? So before we close this session, we would like to hand an appreciation from our program. Prof. Vizca, kami berharap berkenan untuk menyerahkan. Thank you very much, Prof. Erwin, for the insights regarding the precision medicine in infectious diseases. Can we give one big applause for Prof. Erwin before moving on to the next keynote session? So we will be moving on to the next keynote lecture. So our second keynote lecturer will be Professor Duncan Richard Smith. Professor Smith currently serves as professor at the Institute of Molecular Bioscience with research focus on the molecular virology of emerging arboviruses. Professor Smith already published so many books and articles, and he has its index of 54. So if you would like to check out his work, you can access his Scopus or Scholar ID. And he will be talking about how virological and immunological factors shape Zika virus transmission. For 40 minutes, Professor Smith, and will be followed by a 5 to 10 minutes discussion session. The time is yours, Professor Smith.
Okay, thank you very much. Firstly, I'd like to thank the organizing committee for inviting me. I was very happy to accept because Erlanger is always such a wonderful host who takes very good care of us visitors. So it was my pleasure to come here and talk to you today. Next slide, please. There we go. So there was a survey of human pathogens. So this is how many things can make you as people get sick. And the answer, and this was quite an old survey for nearly more than 20 years ago, but they recognized about 1,400 human pathogens that could make us humans sick. About 200 of the species are actually viruses, which is where I work in, and about nearly 40% are regarded as emerging or re-emerging viruses. So human pathogenic viruses belong to about 20 virus families. Altogether, there's about 80 different families, but the pathogenic ones for humans are only 20. Next slide, please, thanks. So viruses are what we call obligate parasites. They have no life on their own. They're entirely inanimate, basically, and they require taking over a host cell to undertake their replication process. So all viruses have two things in common, which is there's some type of genetic material, which can be almost anything, single-stranded DNA, double-stranded DNA, single-stranded RNA, single-stranded, et cetera, There's a great variety. The other thing that all viruses have to have is a capsid protein to encapsulate the genomic material. Some virus, okay? Diane, sebentar. Okay, so all viruses have a capsid and a nucleic acid, and some viruses are like that. They're called naked viruses. Other viruses have a variable outer coat on them, and these are called enveloped viruses. So all viruses are either enveloped or naked. Next slide, please. Okay, virus transmission, we can get things very many different ways. We can have direct contact, HIV, HPV, indirect contact, Norwalk virus, droplet transmission, things like Ebola, influenza virus, and then airborne transmission, which is actually slightly distinct, which is tuberculosis and chickenpox. If you remember the height of the COVID, they were trying to work out whether it was droplet transmission or air transmission. And this made a big difference in how far that the disease would be infectable. We also have the very common fecal-oral transmission. That's all of our enteroviruses that make lots of people sick. But the class that I'm most interested in are the vector transmission class, the viruses that are vector transmitted. And the reason for that is all of this is essentially just human to human. The virus only has to be optimized for humans. But the vector one, the virus has to be optimized to replicate in both insect cells and subsequently in human cells. So it makes it a little bit more interesting in the biology. Next slide, please. There we go. So the main human virus disease transmission vectors are mosquitoes, sandfly, and ticks. And these vectors are all arthropods. And so the viruses that are spread are often called arboviruses, derived from arthropod-borne viruses, arboviruses. Next slide, please. Okay, so there's about 500 arboviruses, and the majority of arboviruses belong to these 4 virus families, which is that this is an old slide because it's now the family, family Flaviviridae, family Togaviridae, family Bunyaviridae, and family Rheoviridae. So the one that we're going to be interested today is basically the family Flaviviridae, because it has the viruses dengue virus, Japanese encephalitis, yellow fever virus, and Zika virus. Next slide, please. There we go. So actually, the genus that we're looking at is going to be the orthoflaviviruses. The name was changed from flavivirus a couple of years ago. So the ones around here are tick spread, so very much often in the colder countries with these tick spread. There's a lot that we just don't know how they're transmitted. We haven't identified anything. And then the majority, more than half of them, are mosquito-borne, either by Aedes mosquitoes, which would be dengue, Zika, etc., or Culex, which would be, a good example would be Japanese encephalitis virus, I think, what else? Murray Valley encephalitis virus. Next slide, please. There we go. So virus replication cycle is basically zoonotic in nature. So it goes between forest-dwelling Aedes species primarily, and it cycles either through birds, rodents, or monkeys. Okay? But you can find it in very many surprising places. So for example, alligators can get infected with West Nile virus. Chinese rat snakes can get infected with JEV virus, Japanese encephalitis. And bats are basically little flying bags of viruses. They carry thousands of different viruses. They're really quite horrible little animals. So that's what goes on in nature. But we get spillover, and then we set up an urban epidemic cycle. And this is usually through Aedes aegypti and Aedes albopictus. So it either goes in a cycle. Occasionally we spill over into a dead-end host. Japanese encephalitis virus, for example. If I got infected with Japanese encephalitis virus, however many mosquitoes bit me, I could not pass it back to the mosquitoes because the virus titer is low. So it affects me. I could very easily be dead with JEV, but I can't transmit it further, so I'm a dead-end host. Okay. Now the primary one now is the urban epidemic, where it goes between people, mosquito, and keep going on like that. That's dengue is the classic example of that. Next slide, please. So the genus Orthoflavivirus is 70 viruses separated into 52 distinct viral species. They're really some of the simplest viruses because they have a positive strength RNA genome, approximately 11 kilobases, with only one open reading frame. Now, positive sense single-stranded RNA looks almost exactly like mRNA. So you can almost think of these as very, very close to infectious mRNAs. They encode for 10 proteins, 3 structural proteins, they're the ones that are going to make up the new virus, and then 7 non-structural proteins. They're going to make the replication complex, replicate the genomic material, and package it all together. And they're called flavivirus because the first virus in this species was yellow fever virus. So that was flavus, means yellow in Latin. Next slide, please. Right, so as we say, yellow fever virus, it was the first identified mosquito-transmitted virus, and the mortality rate is very, very variable. It can be quite low at 3%, it can be at 50%, it can even be at 60 or 70% in some cases. And what was happening up here in America, where the story's going to be, is that yellow fever virus kept being reintroduced into North America. So we see one here in Savannah, which is in America. 700 people died of yellow fever on that year. Why was this? Because over here was Africa, where yellow fever is rampant, and there was a people trade. So the people were traded into, these are the Bahamas first, and then into America. So as they were being traded, they brought their infections with them, and that then got into the local population. Next slide, please. There we go. So the history of yellow fever is this gentleman should be one of the most famous people, Carlos Juan Finlay, because he's the one who said these diseases, yellow fever and malaria, are transmitted by mosquitoes. That was the first time that anybody had said that. And in fact, the name malaria, it actually translates to bad air, because they thought it was just bad smells that were giving the disease. But he postulated that it was transmitted by mosquitoes. This gentleman, Walter Reed, who has a famous American hospital named after him, found a filtrable agent in blood, that yellow fever was a filtrable agent in blood. This is Adrian Stokes, who first isolated the yellow fever virus and sadly died from it. And then this is 1927, this is 1937, Max Thieler. He had a vaccine, 10 years from the discovery of the virus to a vaccine. And it's a really good vaccines for yellow fever. Next slide, please. So the big thing that was going on next was that to ship from this side of America or from this side of South Africa to this side, you had to go all the way around. So 13,000-mile trip. But here, the Isthmus of Panama is very narrow, like about 20 kilometers. So what they tried to do is build a canal, which is now called the Panama Canal, here. And the French tried it for many years, and they couldn't do it. And the reason they couldn't do it was because everybody kept dying from yellow fever and from malaria. So the French tried unsuccessfully, couldn't do it, everybody died. So they gave it to the Americans to carry on with. So this guy is a military sanitation engineer. So he's one that takes care of the environment and makes it safe. And he believed Finlay saying it was mosquitoes. So what he did is he went in and cleared up all the mosquito populations. It was very, tough about it. They would go into homes, they would destroy mosquitoes, they would put oil on potential breeding surfaces, did everything that's effective anti-mosquito treatments. And it worked. People stopped dying just from his, going in and clearing it up. But the canal was finally finished, and the Rockefeller Foundation said, hang on a minute, haven't we just gone a very quick way to get yellow fever virus into our country? So there's, fine, we're going to cut it down so much that it is going to speed up the import of potential yellow fever virus. Next slide, please. Okay, so what the Rockefeller Foundation did is say, okay, let's map where yellow fever can be found. So we know which areas are problematic. We know it's broadly across here in Africa and broadly around here in South America. So what they did is all of these edge areas, They went in and put monkeys in cages, they put rodents there, they collected mosquitoes, and this one is actually in Zika forest in Uganda, where, next slide please. This little fellow, he was he's a rhesus macaque. Now, rhesus macaque monkeys are indigenous to Southeast Asia, not Africa. All right, so he was bought from Southeast Asia, put in a cage in Africa. He had a little tiny bit of a fever, not a lot, and from this, Zika virus was isolated. So this was Zika virus. And they also found it in mosquito populations, Aedes africanus in 1948, so this was 47, and subsequently the first report of a human infection was presented by Simpson in 1964, and he described very well the disease pathology. He was the person who got infected, so he wrote up his own story about being sick. Next slide, please. So they did some serology where people, they reckon about 6% of the population surveyed in Uganda have been exposed to Zika virus, and it was also 1 out of 15 monkeys tested. Next one, please. Okay, so the early serological evidence. So we are going to do some serology or immunology. So it was discovered in 1947 in Uganda, and it was believed to be transmitting in this area. Now, in some samples from 1954 from Thailand were screened for was there any evidence of Zika virus infection, and some samples from Philippines in 1953 were also screened. Next slide, please. But here's the thing. In those days, they didn't have the many immunological tools we have nowadays. Working out if somebody's has antibodies against the virus now is very simple. But in those days, it wasn't. And how they would do it is they would mix virus with the serum sample that they're interested in, and then they would inject it into the brain of a neonatal mouse. Now, there's two possible outcomes. If there's neutralizing antibodies against the virus are present in the serum, we have a happy little mouse. If there's no neutralizing antibodies, then the virus infection takes over, and we have a very sad mouse. Okay? So it was this, whether the mouse was alive or dead, that was the basis of the serological studies in those days. Next slide, please. How am I doing? Okay, so this is one of the early studies. Here we go. They say Zika virus. This is from Thailand. 8%, which actually equals to 2 people of the 25 sera. But if you look down here, they say Thai sera also tested for antibodies against dengue 1, and 80% of the Bangkok sera had antibodies to dengue. Next slide. Here's the methodology. It's an undiluted serum, LD50, positive if 4 out of 5 mice survived. Next slide, please. But look at these other ones. Nataya virus, Ileus virus, West Nile virus, Semliki. They've never, ever, ever been detected in Southeast Asia. This is a South American, this is an African. I think there's only been 4 cases worldwide of human infection. Semliki is not around here. So now we have to say, do we believe the mouse brain result? Next slide, please. And the same from the ones in the Philippines. We have Murray Valley encephalitis, which is limited to Australia, West Nile virus, Nataya, St. Louis encephalitis, Ileus, that's only in South Africa. So again, the same result that seems to say, yeah, there's pretty good Zika going around. But it looks like the methodology is deeply error-prone or flawed. So I don't think we can trust those early studies. Next slide, please. So the real first isolation of Zika virus out of Africa was in Bentong, there it is, in Malaysia, near your hometown, right? Yeah, in Malaysia. And it was found in Aedes aegypti. So this was the first time Zika was found out of Africa. Next slide, please. There we go. And the one that was found in Malaysia was considered genetically distinct from the African Zika virus. Next slide, please. Okay, so in 2007, so there were almost no cases of Zika virus infection. There was only 14 from the time it was discovered in 1947 up until the beginning of 2007. So nearly 60 years, there was only 14 cases that were known. But in 2007, an outbreak occurred in a little tiny group of islands called Yap Micronesia. Now, why was that important? Because it was the first time the virus had been found outside of Southeast Asia or Africa. This is part of Micronesia. It's not part of Southeast Asia. And there was about 49 confirmed cases. So, and the virus was shown to be the Southeast Asian strain, okay? And then a few years later, in 2013, another strain came out from Southeast Asia and went to French Polynesia, where it caused a minimum of 8,000 to 19,000 cases. So this was huge and highly impact on the population. And the virus then started marching on all of these islands until eventually it got into Brazil in 2050. Now we see 1 million cases of infection. And it went up into the USA. It did cause cases. It went up through all these little countries as well. But it got into USA. Not so bad in USA. Anybody have an idea why? It wasn't very penetrant. What does here have that here doesn't have? Aircon. Air conditioning. All the Americans are wrapped up tight in their homes with the air conditioning on so the mosquitoes can't come in. Down here, they live in slums. Water everywhere, no air conditioning down here. So that's why we had a really big impact in Brazil and a much smaller one in USA. So next slide, please. Okay, so clinically, dengue, which is the main dominant mosquito-transmitted disease. Let's have a look. Comparing between dengue and Zika. Fever, yes, yes. Headache, yes, yes. Muscle pain, yes, yes. Joint pain, yes, yes. Nausea and vomiting, yes, yes. Fatigue, yes, yes. Skin rash. The only thing possibly different is there was some evidence that the Zika cases led to conjunctivitis. I personally was never convinced by it, but there you go. But what we can see in dengue cases very often is hemorrhage, and we don't see hemorrhaging with Zika. Next slide, please. Okay, Zika virus in humans is found in the blood, semen, female genital tract, breast milk, urine, saliva, amniotic fluid, placenta. You know, you're just full of Zika virus. It's found in lacrimal fluid in mouse. Now, lacrimal fluid is tears. So what do they have to do to make a mouse cry? I don't want to know, okay? In monkey tests, it's in saliva, urine, cerebrospinal fluid, semen, vaginal, and that's in non-human. So it really does have quite a big tissue distribution. Next slide, please. Okay, transmission routes. Well, predominantly mosquito to human. That was millions of cases, but also male to female, sexual. There was a worker who had been working in Uganda, an American, and he went home and had good relations with his wife, and she got Zika virus infection. So he was obviously carrying it and passed it to her. But we also have female to male, mother to fetus, blood or platelet transfusion. There was an example of a monkey bite, which I think might have been Indonesia or Malaysia, I can't remember. And there's even some human-to-human non-sexual. That was a very strange case. Next slide, please. There we go. This is a sexual transmission of the virus. This here is a spermatozoa, and this signal here, the green, is Zika virus. So it's absolutely an amazing photograph. And RNA can remain for 141 days in semen, which is the longest of any tissue. Next slide, please. Okay, so is sexual transmission of Zika virus significant? If you look at the ratio of women to men, for Zika virus, it's 1.88, i.e. nearly 2 women for every man. But for dengue, which is transmitted by the same mosquitoes, it's about 1 to 1. So yes, sexual transmission is a factor. And it was also confirmed in a study undertaken in Puerto Rico, where the women got 963 per 100,000, and the men were 576 per 100,000. So Zika can't be transmitted by sexual transmission alone. The R0 is too low. But it's certainly a factor in the transmission. Next slide, please. Thanks. Okay, so why was Zika virus such big news? Next slide, please. Oh, well, it was because of cases of microcephaly. Okay, go forward one. There we go. It was this cases of microcephaly, increased cases of Guillain-Barré. That's not specific for Zika. Dengue can get Guillain-Barré and chikungunya can get Guillain-Barré. But it was this microcephaly that caused all the fuss. Next slide, please. So the Zika virus and microcephaly. The Zika virus was found in the brain, but in other tissue of aborted fetuses with microcephaly. This actually is generally not believed nowadays. It is in other tissues. Is detected in the amniotic fluid of mothers carrying babies with microcephaly. And there was an association between mothers who had Zika virus infection and the birth of babies with microcephaly. Next slide, please. There we go. Experimental. Zika virus infects human cortical neural progenitors, and it induces cell death in them. Next slide, please. Okay. So this was the whole point, that the virus infects the neural progenitor cells, and then the developing fetus, they're the ones that divide to give the mass of the brain. So if they're killed, the brain can't develop and it remains very, very small. So it's also very variable how many pregnant women infected in the first 3 months will have microcephaly. 1%, 13%, some studies 30%. So it's very dependent on definition. Next slide, please. So the big question, we have this huge epidemic going around the world, millions of cases. What was going on in Southeast Asia? Next slide, please. Almost nothing. In a period of 2010 to 2017, which covered all of these big epidemics in Brazil, the other countries in South America, there was only 2,500 cases of detected Zika virus infection, and that included the cases in Thailand and Singapore. So, why? Why was it so impactful in other countries and very little in Southeast Asia? Next slide, please. Okay, skip. Next slide, please. And skip. Next slide. All it's showing is that there was very little cases of Zika virus. So the first report of Zika virus was detected in samples from 2006. So it means that Zika virus has been circulating in Thailand for at least 20 years. Next slide, please. Okay, so what is it? Diagnostic, virological, immunological? Next slide, please. Okay, diagnostic. There we go. The two diseases are almost identical. And Thailand has an average of 50,000 cases of dengue per year, sometimes much higher. So the question, has Zika virus been hiding under that? Because in Thailand, if you get dengue, they say, yeah, it's probably dengue, go home and rest. So a lot of the time it could be Zika virus. We don't know. Next slide, please. Okay, so the virological, some studies showed that the virus, the Zika virus from America, is more transmitted by Aedes mosquitoes. Okay, so they say the American one that's got into America is more pathogenic, and if you inject Zika into a mouse and then let mosquitoes feed off it, you get more with the American, more viremia with the American one than we do with the Southeast Asian. I actually don't believe any of this. Next slide, please. There we go. And then this paper came out that said a single mutation in the PRM. My pointer's dead. Okay. So this paper came out and said that a single mutation in the PRM protein of Zika virus contributes to fetal microcephaly, and that's a science paper, which is wonderful for them, but the only point is it's wrong, because we did an autopsy case of Zika virus where we had brain specimens from a fetus aborted for microcephaly, and we showed there was no mutation in that. So there was definitely no direct correlation. Next slide, please. So, is it virological? Let's look. 4 flaviviruses circulate in Thailand. That's the 4 dengue viruses, Zika virus, Japanese encephalitis, Tembusu virus. And more than 90% of Thai adults have antibodies to at least one dengue virus serotype. And Thailand also has a national JEV vaccination program that's carried out in kids. Next slide, please. And all of these viruses are highly homologous. If you look at the top on the dengue and the Zika virus, they look almost identical, right? Antigenically, they can be quite similar. So cross-reactive antibodies have 2 functions. They can either neutralize or they can enhance. 10 minutes. Terima kasih. Okay, so the main process that goes on is with dengue is called antibody-dependent enhancement of infection. So if you're infected by dengue serotype 1, you'll be immune from that the rest of your life. But you're not immune from dengue serotype 2, 3, or 4. Now, the antibodies against dengue 1 can loosely bind to the other serotypes. They can't neutralize it, but that gets taken up by monocytes through the FC receptor. So the amount of infection then becomes dramatically more, okay? Antibody-dependent enhancement. And it's also why it's so difficult to get a good dengue vaccine, because if you underprotect on one serotype, you can get this going on. Next slide, please. Okay, so the question was, do pre-existing anti-dengue antibodies protect against Zika virus infection? Yes, they do. And no, they don't. 2 separate papers, one saying, yeah, they're good, and one saying, no, they're very bad. Science is wonderful. Next slide, please. There we go. So this seemed to answer some of the question. So if you take mice and just give them ordinary plasma and then infect them with Zika virus, then you're going to get a baseline level. Now, in the next one, we're going to add a low concentration of anti-dengue plasma. All right? Then infect them with Zika, and those mice get much sicker. They have a higher viral load and a lower percentage of survival. But if you have a high concentration, then they're essentially protected. So a high concentration of dengue antibodies, they're effectively protected against Zika virus. Next slide, please. However, this was done with rhesus macaque monkeys. And, in the world, there's a lot of monkeys that have been tested out for dengue vaccine design. So they had a whole group of monkeys that had antibodies to dengue, and then they infected them with Zika on top, and some that were not dengue immune, and there was absolutely no difference in the infectivity. So on balance, dengue antibodies probably don't protect against Zika. Next slide, please. Okay, so the methodology we're going to look at now is what's called the plaque assay. So if we want to count plaques, we have to dilute the virus, and then we're going to add it onto a layer of live cells. If there's one virus particle that goes in, it's going to kill the live cells, okay? And then you're going to get, there we go, it's back, blank spots. So if we do dilution, we can then calculate how much virus we have. Next slide, please. Okay, so this is what we did. Next slide, please. Okay, so the problem is how do you define a neutralizing antibody? And the answer is there's no single standard. So we can have PRNT5010, that's plaque reduction neutralization test. So again, if we have antibodies and virus, if they inactivate it, then when we do the plaque assay, there's less plaques to count, right? They're neutralized. There's all sorts of different levels. But the one that the World Health Organization defaults at is PRNT 5010, which is a 1 to 10 dilution of serum neutralizes half the antibodies, okay? It's not very stringent, but it's okay. So we're going to go with that for now. Next one, please. There we go. Everybody does different versions. Initial dilution 10, 50%, it might be 20, 90%. So 2090 or 1090 is very, very stringent. Next slide, please. So we looked at 135 normal, healthy Thai citizens aged above 18. PRNT 5010, at that level, 70% of the population had neutralizing antibodies to Zika virus. Okay? Obviously, the more stringent that we get, the percentage goes down a little bit. But at a level accepted by the World Health Authority, 70% of Thai adults in Bangkok had neutralizing antibodies. Next slide, please. Okay, just to determine that these were not cross-reacting antibodies. Next slide, please. There we go. 5 minutes. Thank you, dear. Okay, I'm dead. Okay, so we did this. We looked at the full print. So we did Zika virus, JE virus, dengue 1, 2, 3, 4, lab, isolate, etc. Next slide, please. And this was the other cohort, which were the print negative samples. Next slide, please. So here we go. So do antibodies to 2 or more flavivirus provide cross-protection? No, it doesn't. And if you count dengue as 1, 2, 3, 4, you get the top slide. If you count as dengue as one virus type, it's still not significant. So there's no evidence of cross-protection whatsoever. Next slide, please. Okay. Okay. So we do get antibodies generated against many other places. And one of the big ones is this non-structural protein 1. Next slide, please. So we looked at non-structural 1 protein. So we culture media from infected cells. We'll have multiple proteins, but the problem is that NS1 and E protein both have similar molecular weights. Next slide, please. So the answer for NS1 can make a dimer, right? So if we run it on what's called a native gel, i.e. with no SDS, we can see the presence of a dimer. So here we have ZIGV anti-NS1 polyclonal, not very good. Here's ZIGV anti-E, and here's human ZIGV positive serum. So this is NS1 and the dimer of NS1. Next slide, please. Next slide. There we go. So NS1 antibodies are very less cross-reactive. So this is a dengue 2 NS1 polyclonal antibody, only picks up dengue 2. And here's a Zika virus NS1 polyclonal antibody, only picks up Zika virus. So with E, we can have some cross-reaction. With NS1, we don't see it. Next slide, please. Okay, so here we go. This is a Zika positive serum, and this was a Zika negative serum. And what we can see in the Zika positive, in the Zika lane, the Z, we can see a band of NS1, the dimer, whereas on the negative serum, we don't see any band. Now, down the bottom are the actual print values, and there's a discordant result. If you look at Japanese encephalitis virus, this serum had a very high level of protection against JEV, but we don't see the NS1 band. You know why? Because Thailand immunizes people with an inactive virus. So if there's no virus replication, there's no antibodies against NS1. So the result is perfect. it shows that that person was vaccinated. Okay, next slide, please. Okay, so these are print negative samples where if you look in the Zika lane, we never get any signal. If you go down to the bottom, which are print positive samples, you can see we get the Zika signal showing up each time. Next slide, please. Okay. Next slide, please. Okay. So the utility of Zika virus NS1, we looked at the sensitivity and specificity. NS1 is actually pretty good. 1 minute, my dear, thank you. And the low sensitivity of a JEV result is from a national vaccination campaign. Next slide, let me just have a look what it is. Yeah, next slide. Okay, so why is the impact of Zika virus seen in Southeast Asia so different from what was seen in other countries? So there's clear immunological evidence of past Zika virus infection in Thailand. Nearly 3/4 of the healthy Thai sera examined had neutralizing antibodies to Zika virus. And these antibodies are likely to have risen through natural infection. And there's probably enough natural immunity to prevent an explosive outbreak of Zika virus. So I think I will leave it there if you don't mind. 1 minute, right? Okay, thank you very much. We can leave it there.
Thank you very much, Professor Smith. Very comprehensive talk about the flavivirus, especially the Zika virus. I think that was one whole semester of of topics packed into 40 minutes. We hope that. Okay. Okay, so if anyone is interested in Zika virus research, can we contact you and have like a discussion with you, Professor Smith? Okay, so next we are going to move on to the discussion session. We have our panelist here, Professor Lucia. She is an expert in parasitology, okay, Prof. Prof. Lucia will maybe have some questions for Professor Smith. Okay. Please, Prof. Lucia.
Sure, no problem.
Thank you. Very interesting presentation, Prof. Duncan Richard. At the beginning presentation, you emphasize the role of vector in virus transmission, especially the Zika virus. And note that the Zika virus has spread widely across several countries. As we know, that the factor for Zika, dengue, and chikungunya virus is the same. It's mosquito. My question is, can single mosquito can transmit more than one virus? As we know, given that mosquito feed repeatedly on different people and animals. That is my question.
Yeah, okay. That'll push me. Yeah, there is evidence that mosquitoes can be co-infected with two different viruses. I think a few years ago we had quite an outbreak of chikungunya virus in Thailand, and it was shown that mosquitoes could be infected with both dengue and chikungunya at the time. So yes, co-infection can happen. You're welcome.
Oke, Bapak Ibu, apakah ada pertanyaan yang lain? Sebentar. Dari penanya yang lain dulu mungkin. Kami persilahkan.
You don't have to be scared. You can ask in Bahasa and it'll get translated for me.
Ya,
And I don't shout at people. I like questions, okay?
Ya, Bapak Ibu monggo, boleh dalam bahasa Indonesia, nanti akan kami sampaikan dalam bahasa Inggris. Silakan. Ada? Oke. Diperkenalkan dirinya dulu ya.
Okay, thank you for the time. Let me introduce myself. My name is Maria Evata. I'm the currently master student of immunology. Okay. Maybe
Nice to meet you again.
Yes, Prof. Okay. So. Okay, sorry. Zika virus can be transmitted through many factors like sexual transmission and pregnant mom to their child. So the question is, when Zika infection occurs despite prior flavivirus exposure or vaccination, does pre-existing immunity can reduce or increase the risk of sexual or vertical transmission? So if person becomes infected with Zika virus before, can the resulting immune response in the next infection is reduced like that? Maybe you understand or not? Oh, okay.
I think I do.
I'm so sorry if my English was
No,
bad.
it's okay. It's a lot better than my Bahasa.
Yeah.
Don't worry.
Okay.
No, it can't be, because when you get infected with Zika, well, let's go to dengue first.
Yeah.
You generate immunity against the infection. So if you're, that's adaptive immunity. So if you have, let's just say dengue 1, and you get infected with it, you make antibodies against it, you are immune for the rest of your life to dengue 1. And we know from experiments in places like Cuba that the immunity can last for up to 60 years. So basically you're lifetime protected, okay? So with Zika, once you get infected one time, you're going to be lifetime protected. Now, one thing we didn't say about both Zika and dengue is that the majority of infections are asymptomatic, all right? Only 20% are normally symptomatic. So it means lots of people get infected with dengue or with Zika and they don't even know it. They just raise a good antibody response against it, okay? Now, dengue is slightly different because there's 4 viruses. So you'll be protected against 1, but you can be infected by 2, and then you can raise antibodies against 2, then you're protected. And then you might be getting broadly neutralizing, but you can also get infected twice more with it, which is very bad luck, okay?
Okay, thank you so
You're
very
welcome.
much, Prof.
Okay, please, Dr. Adi Idris.
How you going, Duncan?
Are we allowed to question?
Yeah, we are, I think. I'm a virologist myself, but I don't really work with Zika. Out of curiosity, I mean, Zika, there's no specific antivirals being developed for Zika. It's a blood-borne. As a Zika virologist yourself, what do you think is an ideal modality for treatment? Would it be like a monoclonal antibody treatment? Would it be a typical remdesivir? Because it's blood-borne, it goes everywhere. I just want your expert opinion if one were to develop one.
The problem with, which I'm sure you know better than I do actually, the problem with antivirals is they tend to be very, very specific, that it's one drug for one virus. And I don't think there's anything that shows effectivity or effectiveness against really any of the orthoflaviviruses. I mean, there's nothing for dengue, and we've been researching dengue for 80 years or more. So the big problem is these are RNA viruses, okay? And the difference with RNA viruses is they have no proofreading function. So they have to bring their own RDRP because they're going to make a negative sound at some point. But that RDRP has no proofreading. So it means you create mutations literally every time. And so whatever drug you get, you're almost certainly going to develop an escape from it very, very soon. So, but the problem is doing vaccination now. If you've got a great vaccine, how are you going to test it? Because there's no ongoing epidemic of Zika.
Okay, thank you very much, Professor Smith. So that was so many expert opinion, and I don't think I can summarize it briefly. So one closing question, Professor Smith. So there was clear immunological evidences of Zika virus infection in Thailand. And I think there are several published reports in Indonesia regarding Zika infection. Should we be worried?
If I remember, the Indonesian government said, no, you shouldn't be. And they refused to implement testing for it. But as we tried to put a project up, the three of us, a little while ago, right, where we wanted to look at the seropositivity in Malaysia and Indonesia, but unfortunately we didn't get through to funding on that. So one would have to know the incidence and how many cases are we getting. And even if you discriminate and say, okay, this is Zika, so what? Go home, rest. There's dengue, go home, rest. It's only in the really severe ones do we need to worry about it. So I would think that Indonesia is probably the same as Thailand, it would be my bet, that maybe we've had long ongoing transmission that's just completely been missed. But without the serology, we can't say that. But if I remember, the monkey bite case was from Indonesia. Was it Indonesia or Malaysia? Not Malaysia. Yeah, it was from Indonesia. There was a case where a tourist got bitten by a monkey and then got infected with Zika virus. And that was a long time ago in Indonesia. So you've probably had it circulating quite a long time here. But again, you're not seeing this big burst of cases. So without that, I wouldn't worry too much.
So if the doctor say go home and rest, so it will be okay for us.
Unless you're pregnant. If you're pregnant, you're going to take it a lot more seriously. But yeah, I mean, for dengue, the treatment is, there's no specific drug to treat dengue. There's no specific drug to treat Zika. So it's basically symptomatic, isn't it? Stay hydrated, take aspirin rather than paracetamol, and fluid, yes. That's it. And only if you get the hemorrhagic form, then you're in trouble. Yeah.
Okay. Thank you very much, Professor Smith. So can we give a big applause for Professor Duncan Richard Smith? Okay, to close this session, there will be an appreciation from our program to Professor Smith. Prof. Fiska, sekiranya berkenan untuk menyerahkan. Okay, thank you very much for our keynote speakers, Professor Erwin and Professor Duncan Richard Smith. So that will conclude our keynote sessions, and we'll be moving on to the next agenda. So I would like to give back the time to our MC, Mas Nabil.
Okay, thank you so much, Dr. Ayu. Just popping in quickly to let you all know that outside the room there are snacks that you can fill up on, the snacks that were just given to you if you want to refill the snacks that was previously given to you. And also coffee and tea is also provided outside if you wish to also take some also. And without further ado, I guess I will move on to the next agenda, which is the next two keynote speakers, which will be moderated by Dr. Waude Vivin Ervina. So to Dr. Vivin, the time and place is now yours.
Okay, thank you very much for our Master of Ceremony, Mr. Nabil. Good morning, everyone. Hi, I'm Vivian Ervina. I'm a moderator for this session. First of all, this is very interesting. Thank you very much for your attending for this event. It's very interesting. We're talking about the immune system against the infection disease and autoimmune disease. And now we continue for our speaker, our remarkable speaker, Dr. Nur Hafiza Zainal, PhD. But before I invite Dr. Nur Hafiza, I will read the curriculum vitae for Dr. Nur Hafiza. The education from Dr. Nur Hafiza, the PhD from Medical Microbiology in University Malaya and National Cheng Kung University. This is, she got a dual PhD program, mashallah. And then the Magister MRes in Molecular Plant Biology and Biotechnology in Imperial College London, UK, and the Bachelor in Biotechnology, and also her area of specialization skills in the field of the virology and infection disease in medical microbiology, and immunology also, and laboratory and technical. And Dr. Nur Hafiza Zainal got award and recognition from International Education Innovation Fund 2025 from Australian Government Department Education. Give applause for Dr. Nur Hafiza. And also Excellent Service Award 2023 from University Malaya. And now Dr. Nur Hafiza will talking about the dynamic of antiviral immunity, lesson from emerging viral infection. Dr. Nur Hafiza, time's yours.
Okay. All right, Assalamualaikum and good morning. Thank you for the kind introductions. And also thank you for the organizing committee, Dr. Ayu, Dr. Salma, and the others for inviting me to be part of this wonderful seminars or conference. And thank you also Prof. Viska, for the invitations. Okay, so I'm going to talk about the dynamics of antiviral immunity, lessons from emerging viral infections. I think it's already being emphasized throughout the talk, regarding the balance between the protections and also pathological effect of immune response. Because we know the immune response not only protecting, it can also be insufficient, it can be cross-reacting, right? And it also can lead to, harmful outcomes that we don't want. Okay, so can we go to the next slides? Before we start with the science, I need to introduce, this is requirement for us, if we speak outside. So University Malaya, we already here in the world over a century, right? So over the years, it then become our now UM. Next. And then we have about 21 faculties, academics, institutes, and also academic centers. We have more than 200 academic programs. At the site there, you can see that we have 82 undergraduate programs, 136 master degree program, 28 doctoral degree, 2 double degree. Who wants to do dual PhD, you can come to UM. 8 dual master program and 24 collaborative PhD program. Next. And we really support and really encourage student mobility, research collaboration and knowledge exchange. All right, next, done with UM. So we go to the science. Oh, not yet, oh, sorry. Okay, that is my faculty. My faculty, we actually connected to our University Malaya Medical Center. So this is me, Department of Medical Microbiology, and then my interest will be viral immunology, arboviral diseases, and so on. I think just now the moderator already informed. Okay, next. All right, to the science. Before I start, let's look at virus history. In 1892, Dimitri did research on tobacco mosaic disease, and he found out that the infectious agent could pass through bacteria trapping filters. So at this point, they know that these pathogens or this microbe is different than bacteria, because it's smaller. And then over the years, they understand more on virus. They will know virus actually very small. It cannot be seen with the light microscope. And as mentioned as Prof. Smith, I think we're going to have overlapping information. It's a simple structure. It's made up of genetic materials enclosed in a capsid. and it sometimes have envelope and some not. Yeah, and then it's also obligate intercellular parasite. It require host to replicate, so they don't have the machineries to make their own protein, so they will require the host machineries. And then it's also specific. Right, next. So over the years, when we understand more on the virus, we will then understand more on the antiviral immunology or immunity. Right, so in 1957, interferon discovery. So this is the protein that alert the cells and also establish the antiviral environment in the cells. And then in 1959, we found about antibody structures and how it binds to the virus. And then thymus function. Thymus is basically where T cells develop. So we know that T cells, especially CD8 T cells, is the one that help to control virus infection through killing infected cells. Yeah. And then in 1987, we understand more antibody diversity instead of detecting antivirus. Yeah. So throughout the years, we learn about the antiviral immunology. So I believe it is a basic framework, yeah, to understand antiviral immunity. Next slide. So what happened when the virus get into the cells? Yeah, this one also mentioned by Prof. Visca this morning. Once it, of course, it will undergo, it will need to go through our barriers. We have barriers, physical barriers, chemical barriers, biological barriers first. So when they get through that, it will then be recognized by our cells. Yeah. So as you can see, the virus, yeah, the virus can be recognized by TLR3, TLR7, RIG-I, MDA5, and also C-gasting. Yes, once it recognized the virus, the cell will then be activated. Yeah, activated, and then it will activate the signaling of type 1 interferon productions. Type 1 interferon will then react on the infected cell as well as the nearing cells, bystander cells, that it will induce the antiviral state. Yeah, how it induced that? By productions of ISGs, yeah, interferon stimulated genes. So these protein are the one going to block, yeah, during the innate immune response, yeah, going to block the virus replication at different stage. Yeah. And then at the same time, there's activation of innate immune cells, right? NK cells, macrophage, neutrophils, yes? Oh, yes. So that also help to control the virus infections. And then, of course, it then activates the adaptive immunity. Next slide. So for adaptive immunity, we know, yeah, so our APC, example DC, yeah, DC will then engulf the antigens and present it on the MSC, and then it will then activate naive T cells. Yeah? So T cell will then be activated. It can be either CD8 T cell or CD4 T cells. Yeah? So CD4 T cell are the one that going to help other cells. Yeah? It helps CD8 T cells to be activated, which then help with the killing of the infected cells. So CD4 T cell will then help B cells also to be activated and produce antibodies. Yeah, antibodies will then help to neutralize the virus. And then at the end, we're going to produce immune memory. We have memory CD4 and CD8. So these memory cells are the one going to help us. If we got exposed again with the same virus, yeah, it will then induce better immune response. Okay. So next. So it comes to my interest. I know, I mean, with all of these efficient antiviral response in hosts, why some pathogenic virus still cause infections? Yeah, the 20 types of the pathogenic virus. So do we need to enhance our antiviral response? So we do, I mean, in my lab, we do test a lot of compounds to see antiviral response against the virus, but they never understand the mechanism of it. They just assume that the compound will then stop the virus directly. Then I want to learn more. I want to understand more in that sense. So I do knock down, yeah, for all of these genes. I don't want to talk about this paper. It's going to take so long. So I just want to summarize it that we found that resveratrol, as you can see from the left side, with higher concentration of resveratrol, it can reduce the virus, yeah. And then B, just to confirm it with Western blot. But then when we knock down certain host protein, yeah, which is HMGB1 and SIRT1, we found that the resveratrol cannot exert the antiviral response. Yeah, it cannot exert the antiviral response. It can only exert the antiviral response when there's HMGB1 in it. So we believe that in terms of antiviral, we not only targeting the virus, we can also target the host protein to become better in terms of initiating the antiviral response. As you can see here, HMGB1 in the nucleus, if there's a resveratrol, it will then become staying. It will stay in the nucleus and then don't come out. It will not be acetylated. It will be deacetylated and stay in the nucleus and help with the production of ISGs, which is very important for antiviral environment in the cells. Next. We also try to see the effect of resveratrol to other viruses, flavivirus, Zika. So for this one, we also see reductions. Unfortunately, I don't have the resources to test the mechanisms of HMGB1 and SID1, but we will do it once we got the funds. And next, we also tested. Can we go to next? Next slide. Okay, so we also tested this resveratrol to other virus, chikungunya virus and also JE. We also find there is a similar impact on the virus inhibitions of resveratrol. And again, we don't really look at the mechanism, but we will do one day, hopefully. Yeah, that's my student actually down there. Okay, never mind. So next, let's move on to another part of our interest, which is cross-reactive. I think this is also mentioned by Prof. Smith. This is another dynamics of antiviral, because we know early on, adaptive especially will be specific to the specific virus, right? But in this case, it is not. Okay, there's a cross-reactivity. Yes, so cross-reactivity, what happened when immunity meets a new virus? So if you look at the left side, first it was infected by the first virus, and then it will then produce memory B and T cells. And then when they expose to other virus, it can actually be protective. I know you're not gonna be happy with it, but some found that it can be protective. We call it as a cross protections, where they can actually provide protection to other type of virus. It can be related or unrelated. If it's related, it can be like dengue virus just now, different serotypes, or maybe similar genus, flavivirus, dengue and Zika, or even unrelated virus. We're going to look at this later. So as you can see, beneficial outcomes. So next slide. So there are a few literatures that shows the protections of cross-reactivity. For example, dengue virus, CD8 T cell mediate cross-protections against Zika, and then JE also cross-protection against Zika as well. Okay. Next. However, I also want to emphasize there's a certain conditions that the cross-reactivity can lead to detrimental outcome. So this is the one. Can I get the pointer? We cannot get the pointer. Not yet. Okay, never mind. So at the red color one, it shows the detrimental outcomes. So the antibody now, from the primary infection, from the first infection, it's not helping. Instead of helping, it's actually helped the virus to, instead of neutralizing the virus, it's actually helped the virus to get into the cells and cause more infections, right? So increased viral replication will then they will then induce inflammation, more cytokine that lead to cytokine storm, and then tissue damage and injuries. That's detrimental outcomes of cross-reactivity. Okay, next. So this one shows the bad cross-reactivity impact in terms of dengue. So dengue, I think one of the disease that ADE has been studied quite comprehensively. It shows, I think the mechanism also similar as I mentioned just now. It's sub-neutralizing antibody or non-neutralizing antibody. Instead of helping to neutralize, it's actually help the virus to get into the cell through Fc receptor. And then when they get more inside, they will enhance replication and leads to all of these exaggerated immune activations and injury of tissues. Okay, next. So my next question is, can cross-reactivity happen beyond flavivirus? Can it happen beyond flavivirus or even dengue, Zika and all? So next. So we are, next slide. So we actually interested on SARS-CoV-2 infection, which is the one that caused COVID. If everybody already know or still remember, it caused pandemic in 2019. So we want to see whether there's a cross-reactivity of SARS-CoV-2 infections. Next. So if you look at the cross-reactivity immunity studies of SARS-CoV-2 and other viruses, you can see this one showing influenza with influenza virus or respiratory virus. There's a relationship between influenza vaccinations coverage and COVID-19 outbreak. In a way, they are showing that vaccination of influenza can actually shape the COVID-19 progression or disease. And then this one showing a bit on protections, I believe. Symptomatic MERS-CoV infection reduces the risk of future COVID-19 disease. So they found that those people who got MERS last time do not really get severe COVID-19. Somewhat of protections. And then here it's also mentioned about non-neutralizing SARS-CoV-2 antibodies against the influenza virus. Okay, next. And then this is showing cross-reactive immunity of SARS-CoV-2 with arboviruses. So here in Singapore, they found that COVID-19 can actually lead to false positive dengue serology, even though they are totally different virus, but they do have some sort of a cross-reactivity. And then this one showing anti-arbovirus antibody cross-react with severe acute with SARS-CoV-2. They found the cross-reactivity, but whether it causing protections or pathological effect was not known here. And also here, similarly, they just shows that there are cross-reactivity. Next. So that's bring to our interest in our lab to understand whether there are actually protections or pathological effect. So we collect samples from vaccinated or recovered COVID-19 patient samples, and then we perform virus assay, neutralization assay. I think similar with Prof. Smith just now. Thank you for the very comprehensive slide, so you understand what is neutralization assay. Basically we are using Hu7 cells for this case, and then ADE assays. So ADE assays, we are using those FC receptor cells, such as U937 cells. And then we look at whether this antibody induce more virus replication or reduce viral replications. Next slide. So this is our first outcome. When we look at the cross-reactive of SARS-CoV-2 antibody against influenza virus. So if you can see from this figure, these are our cohort. So we have double negative, meaning our controls. Those positive IgG SARS coronavirus 2 and negative to SARS-CoV-2 IgG and negative influenza A IgG. And then we have single positive. Single positive is only SARS-CoV-2 IgG and negative influenza A. And then single positive of influenza A IgG and then double positive. Right, so we did 2 dilutions, one in 320 and one in 1280, and then we look at whether this antibody can neutralize H1N1 or not. So as you can see from this figure, there are no significant protections. We can see some sort of a trend of reductions, but it's not significant. It's not biologically significant. So we believe there's no protections, no protection between SARS-CoV-2 antibody with influenza virus. Okay, next, and then we did the ADE assay. So we were using the FC receptor cells. So again, similar cohort. Similar exactly cohort. Double negative, single positive of either SARS-CoV-2 and influenza A, and double positive. So this is where it get quite interesting. Also, forgot to mention, for this part, we do 3 dilutions, one in 100, one in 1,000, and one in 10,000. From the dilution itself, you can see how the antibody helps with the virus replication, because the less the antibody, the less the virus replications. So this is antibody dependent replications. So when you look at the double negative as a control with the single positive of SARS-CoV-2 samples, we can see significant enhancement of the virus, especially in 1 in 100 dilutions. There were also enhancement in other dilution, but because of the samples, it's quite hard to get the patient samples for this case. It's not really easy to get influenza A negative because most of us have it, right? So that's why it's a bit hard to get a significant result. Yeah? I believe with more sample we can get better results. So enhancement was there, so suggesting a potential of enhancement of the virus. But then when we look at the influenza positive only, there's no enhancement. There's no enhancement ini. And then interesting as well, double positive also do not have any enhancement, similar like SARS-CoV-2 IgG positive only. I'm not sure whether having influenza actually helped to prevent this, right? But then we can only see those with SARS-CoV-2 IgG only that have this enhancement. Okay, next slide. So this shows the potential of ADE, of H1N1 virus infection in the presence of SARS-CoV-2 antibodies, right? So since both of these diseases, co-circulate globally, so the possibility of the prior SARS-CoV-2 immunity can cause some sort of a worsened influenza outcome need to be focused on, I believe. Yeah, if you look at the existing literature, right? Okay, this is not working also, never mind. So if you look at the upper right paper, yeah, so it shows association of SARS-CoV-2 vaccination status with the risk of influenza-like illness and loss of work days in healthcare workers. I'm not sure if you guys feel like flu is a bit severe after the pandemic. Anybody have any experience? I'm not saying that it's a cause, it's an association only. This is very early work. I think we need a lot more research to understand this. But yeah, it's probably that's the reason why we have severe flu after pandemics, allergy, that kind of thing. Yeah. Next slide. So we also interested to look at the cross-reactivity between SARS-CoV-2 and arbovirus, which is chikungunya virus. So similarly, oh my God, I'm so sorry I missed this. SARS-CoV-2 IgG here. So we have double negative. Yeah, they don't have SARS-CoV-2 and CHIKV IgG. And then we have single positive of SARS-CoV-2 IgG only, and then single positive of CHIKV IgG, and then double positive. Yeah, so we also have similar dilution, 1 in 80 in 1 320. So for this, we're using neutralization assay of Hu7, and we found that it's significant just between the similar virus. Yeah, this is not surprising because we know that once you got exposed to chikungunya, yeah, so obviously when you challenge it with chikungunya, it will then inhibit, right? But then we don't see here. It's probably again our limitation of the samples because it's not easy to get those without SARS-CoV-2 now. This is our retrospective samples. And then among that cohort, we need to find chikungunya virus IgG. So that's very hard. We're still working on it. And then of course we have double positive. So we can see the protections only by the same virus. So no protection by SARS-CoV-2 IgG, with the chikungunya virus replications. Next. So when we do ADE assays, same cohort, we found that there's no ADE, which is good news for us. No ADE if you are single positive or SARS-CoV-2 IgG. But then there's a big enhancement. This one is, I think, already being reported. CHIKV can also cause ADE if you got exposed to the same virus. But it was not significant in this case. However, the interesting part is our double positive show enhancement of viral replications. So at this point, we might think that those that have both, that have SARS-CoV-2 antibodies, when they get exposed to chikungunya virus next time, they will have a higher replication of the virus. Possibility, it's just suggesting a potential ADE. So we also in Malaysia have outbreaks of chikungunya recently after the pandemic, and there's a lot more people got chikungunya. Before it was mild, right? But then now there's more cases and then a lot more severe compared to last time. So I'm not saying or claiming there are connections, but we probably can see some trend there. Okay. Also, this is my students who are doing it now. Next. So potential enhancement of CHIKV infection was observed in dual positive. So co-circulation of SARS-CoV-2 and CHIKV raises concern that dual positive antibody may enhance CHIKV outcome, suggesting potential ADE risk. As you can see here also, there's a paper showing the antibody-mediated enhancement in chikungunya virus. Yeah, and then pre or post SARS-CoV-2 infection significantly increase CV dengue virus disease criteria. So we are just connected with the other arbovirus. Okay, next. So with people now receive at least one dose of COVID-19 vaccine, we have IgG against SARS-CoV-2 now. I'm not saying that we have definitely the risk, but at least be aware that there are possibility of enhancement if you got exposed to other type of virus. Next. So these are the side effects of the COVID-19 vaccine. Not saying that it's a causal effect, yeah, definitely from our findings, but we just want to associate, yeah, the possibility of having antibody, yeah, of SARS-CoV-2, and then you got exposed to other virus, you're probably going to be a bit more severe. Okay, next. Yep, so come to key takeaways, yeah, antiviral immunity is dynamic, it's not really protective only, it can also cause pathological effects, so it's important to understand the context. And then there's the cross-reactive immunity can have different outcomes, it can neutralize, it can have limited protection, so even can cause enhancement. So there is a potential risk of antibody-dependent enhancement mediated by the SARS-CoV-2 antibody against endemic of circulating virus infection, at least in Malaysia. And then that's why understanding immune history may help us better. It's not history actually, I changed it, but never mind. Understanding immune dynamics, yeah, actually can help us better predict antiviral outcomes. And then obviously very important that I am pro-vaccine. Yeah, I'm not saying that vaccine is bad. It's just that we need to be aware that there's a risk with vaccine. Obviously, when we weigh in the risk and the pro, yeah, it definitely pro. It can save lives, yeah. But be aware that it can also cause some sort of a pathological effect. And once you be aware of it, then we can then enhance or improve our therapy. Yeah, we can even improve our not only therapy, but we can also improve our vaccine productions. Make sure we also look at the possibility of ADE next time, not only for dengue, but also for other virus infections. Okay, all right, can we go to the next slide? Okay, I want to say thank you to everyone for listening to my talk, and thank you to Airlangga, yeah, for inviting me. And then my co-researchers and collaborator, Dr. Ayu, right? And then my mentor, Professor Adi Abu Bakar, Prof. Lind, Dr. Chin, Dr. Tan, and then all my students for helping us. We are actually a group, yeah, trying to understand about antiviral immunity. And thank you.
Okay, thank you very much for Dr. Nur Hafiza. And now we move to the first speaker, Dr. Adi Idris, PhD from Queensland University. Before I invite him, I want to inform you about Dr. Adi Idris. He is a group leader from Antiviral Therapy, Center for Immunology and Infection Control from Queensland University of Technology, and also he is an adjunct professor from Northwest Minsk University, China, and adjunct professor of Vellore Institute of Technology from India, and also adjunct professor Universitas Airlangga, Indonesia. And Dr. Adi Idris will talking about the developing RNA-based gene therapy, targeting therapy against viral infection, a new class of antiviral. And Dr. Adi, time's yours.
Thank you very much. Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera. Two things. Number one, thank you very much for inviting me. I like small conferences. I find that I make better friends better and more friends in small conferences. I think we can have better dialogues and better relationships. And also, number two, celebrating World Day Immunology, even though I'm trained as a viral immunologist and I'm no longer an immunologist, it's a great thing. So thank you for being here. This is going to be tricky because I talk very fast and there's a lot of stuff that comes out of my mouth. So I don't know how we're going to do the slide business. So let's see how we go. Next slide, I guess. Some disclosures, so I spend most of my time in biotech. I'm hardly in academia anymore. This is just a disclosure. Whatever opinions are raised here is not the opinion of the company, it's the university's opinion. Next one. These are my funding acknowledgements. Next. This is my team. Without my team, I'm nowhere. Without my team, they do everything. I don't do anything, right? I just come up with the ideas. I tell them what to do. They do all the work. So this is my acknowledgement slide for my entire team. Okay, next. This is QUT. We're located on the east coast of Australia, Brisbane City. You can see the Gardens Point campus there, sits on the mouth of the Brisbane River. Brisbane River is connected by the CityCat. So I note that you were at the University of Queensland. I'm also an alumni. I did my PhD 20 years ago at UQ. And the CityCat actually connects to the University of Queensland, right, if it goes downstream. So it's a really nice place to be. Please come in 2032 for the Olympics and support your fellow country people during the Olympics. Next slide. The technology I want to talk about, I'm going to talk about a very small part of my work for this entire workshop. We develop RNA therapeutics to target viruses directly. So we use RNA interference, and that RNA interference is the use of RNA that will hybridize a target messenger RNA, or viral mRNA, to stop that virus replicating. Right? So in a way, it's a type of direct acting modality. On the right there are some of the viruses I work on. So I work on all sorts of viruses with people from all over the world. So you can see veterinary oncogenic viruses. I also have an interest in tick-borne viruses. I'm not showing any of that data now. It's proprietary material. So actually a lot of the stuff is proprietary. So I'm just showing some data that I can show. Next. So we actually developed this RNA therapeutic, and we weren't looking for notoriety. We woke up one morning, and then the news articles just went crazy. We're like, what in the world's going on? So we got pressured by the university to, like, you know what, we need to spin out a company. So we did. We tried to. Next slide. I'm gonna get used to this next slide business. Yeah. So we tried to spin it out, open a company in the US, but the venture capitalists lost interest in us because the day after, Pfizer developed a new antiviral, which is actually not very good, right? They developed a new antiviral, and the venture capitalists lost interest, so we couldn't set up our own company. That's the number one. When you want to do something, raise funding, do it fast. Don't do it with the university, because they have their own speed and KPIs. You want to start something, you do it yourself. So what does universities do in Australia? They sell the license to another company. No, go back, no, yeah. They sell the license to another company. So they sold it to Gene Company. Then they sold the license to another company called IntelliGene. They went public and they became very rich. So after that, they said, hey, Adi, you developed this drug, let's join our company. So I did. So that's how I end up in there. So I spend a very small amount of time in academia now. Right, next. One of the technologies with RNA therapeutics is that you have to protect the cargo. RNA is very unstable, right? You can deliver it naked, but you have to chemically modify every single backbone, which costs money. So a company called Alnylam does that very well, but we want to make these drugs, I guess, affordable, so that it's able to be equitably, distributed elsewhere. So one of the projects is that we want to develop several siRNA drugs that target multiple respiratory viruses. So this is a project that's been ongoing for a while. We package all of that into a single drug formulation. So imagine a nasal spray that you can spray to target the nasal cavity to stop the viral replication and transmission of the virus, right, with one formulation. So that's what we're trying to develop. All right, next slide. Yeah. So here's just some data just to show you that when we package 3 drugs, right, into that target SARS-CoV-2, respiratory syncytial virus, which is RSV, and influenza, it can ameliorate viral infection in the lungs and also nasal cavity. So we're like, great, the concept works. We can turn this into a nasal spray. Let's keep moving. Okay, next one. Thank you. So that's the proof of concept, and it works. So we also tinker around. So we work with a lot of companies, right? So the stuff that we do is mostly industry-based. This is a company, ProRunner Biotech. It was actually started by my PhD student. So he finished his PhD. He said, I don't want to be a postdoc. I'm sick of it, right? So he said, I'm going to open my company. So he did. He opened one, and then he spun out a company and went to Qatar, and he became very rich, right? So what he's done is he's made a microfluidic thing, which looks like a cocktail mixer. So you put component A, B, and C, mix up with the RNA, and you get this drug with a very nice distribution, profile distribution. And then we tested it to see, how well it performs when you give it by spray or intranasal. Now, intranasal, that only targets the nose, whereas nebulization is good for lung targeting because your particles are smaller. It goes down to the bronchiole. So those who have hospitalized patients, for example, or severe to moderate, respiratory disease, this is perfect. So you can see that the knockdown of a target gene, so in this case, beta-actin, we use as a control, as a test. You can see it's knocked down, around 12 hours, and disappears after 24 hours. But just to show you, compared to a conventional lipid nanoparticle, the Geno LNP seems to be performing as well, and it's cheaper. There's no electricity, and there's no power needed. So imagine using this in low-resource environments, right? And it keeps it at a GMP scale level. All right, next slide. Here we also developed another therapeutic. So we thought, if we're going to give it up to people's nose, and the nose is such a mechanically dynamic place where we pick our nose and then we sneeze, we breathe in and out, so there's a lot of in and out. How are we going to keep that drug stuck in there? So we thought, how about we develop a thermostatic hydrogel? So with University of Queensland, my buddies, I told them, can you develop a thermostatic hydrogel whereby when it gets into the nasal cavity at body temperature, it gelates. So it's a liquid when it's cold, and then it becomes hard when it gets into the nose. I'm like, that'll be perfect. Then what you have is a controlled, a sustained release of the drug over time. So it's perfect. So what we did was we tried different formulations, and you can see that signal. So look at the signal there, right? So at 48 hours, we get to see our RNA drug being nicely distributed and bioactive at that time. So we thought this is a great formulation. I can't show you again too much of it, and here's the knockdown dynamics. At 48 hours, you can see a good dynamic knockdown of the signal. So we seem to have a very good formulation, and this is all animal studies, by the way. Next slide. So I did mention you don't need lipid nanoparticles. You can deliver it naked, but it's very expensive. So here's an example of an siRNA looks like, right? It's double-stranded, right? You have to modify every single known base to man on that thing, right? And it costs you thousands of dollars. So what we've done is, we're going to deliver this to the lungs. I want to keep it in the lungs long enough, so this is no more nasal cavity. So what we did was, it's an old chemical trick. Let's put a cholesterol at the end. So we did that, and we compared modified and unmodified, right, in the nose and in the lungs. And you can see quite clearly, red being a very strong signal, it just stays in the lungs longer when you have cholesterol, correct or not, right? So tinkering the bases and the modification of the RNA allows better kinetics of the drug. Next slide. So here we actually nebulize it. So you can actually nebulize that same siRNA and study stability of this drug, right? You can see compared to unmodified and modified, again, nasal cavity and in the lungs, you can see the dynamics is slightly different. I just want to highlight here that irrespective of whether you nebulized or not, whether modified or unmodified, the siRNA does not stay very long there. Why? You know what mice do? They do this. They scratch the nose, they drink, it's a very disruptive environment, right? The respiratory tract keeps moving along. I mean, forget about the cilia in your trachea. The nose is just disruptive. So a hydrogel is something that we're tinkering now. So we're going to extend this project into hydrogels and see if we can actually keep it up to 48 hours, right? Next slide. So this is another project we developed. So this is a nucomotive drug, and next slide. This is a stage thing. Next. Here. Now, why influenza? Influenza is a weird virus, right? It's one of those weird viruses where it has a replication cycle in the nucleus. And for years, people have tried to develop RNA therapeutics against influenza and never got 100% killing or more than 50%. We're like, why in the world can't this happen? It's because they go to the nucleus first and then shuttles out the viral RNA and then makes its protein. So we're like, okay, let's beat the virus to it. Let's develop an RNA, sRNA, that can go to the nucleus. And that's what we did. When we apply Nucleum technology, you can see the efficacy, the antiviral efficacy increase. In the lungs, when you give it intranasally, you can see it's much improved. Now, those that do animal studies, right, a 1 log scale decrease is pretty good. All right, next. You can apply this, You can apply this to other human targets, right? So this is a link RNA target. Next one. What's the next one? Yep, keep going. Oh, anyways, I had actually retroviral data. I think it's because of the ship. Anyways, I just want to show you that our Nucleum sRNA beats either conventional or current RNA therapeutics in this, and it's also non-stimulatory. Right? We made sure we design our sRNAs to not trigger NF-kappa B or IRF, promoters and stuff like that. Next slide. All right. So right now we're trying to spin off a company. We're in the middle of IP generating, locking in IP with the university. So we're actually doing some primate toxicity studies in IPB in Bogor. So I have very deep links with Indonesia. I come here like twice or 3 times a year. I have deep links with Airlangga, usually in chemistry, and also deep links in UNPAD in Bandung. So I'm going there in a few weeks' time too. So this is our timeline, and we're right now raising money. All right, next slide. Here's another one I want to talk about before I close up. So instead of lipid nanoparticles, which is synthetic, so we want to try and see whether we can use natural lipid nanoparticles. Extracellular vesicles, or EVs, I'm going to say from now on, are actually naturally secreted out of cells, right? So EV biogenesis is a bit like viral biogenesis, right? Those who are virologists would appreciate that a lot of viruses actually hijack this pathway, right, in cells. And a lot of people, there's evidence to show that EVs are made, virus particles are being released quite easily in host cells because they hijack this pathway. Anyways, EVs are great because they're natural. So we thought, hey, how about if we molecular engineer cell lines that can make EVs with different things? Antiviral targeting nanobodies, put some antiviral RNAs, organ-specific nanobodies, all sorts of things. Organ targeting, like, this would be great, right? If we could do this. So we started doing that over the past few years. All right, next. And here's one example. Yep, next. Yep, so here's one example where we put a big protein called ACE2, right? So ACE2 is a big protein that is, that obviously binds spike protein. And we then asked the question, does this neutralize the virus? And it did, compared to viral-like particles engineered with ACE2. Next slide. Here's another example where we put nanobodies on top. So VHH are nanobodies that are different from full-length antibodies. So these nanobodies are on the surface of the EVs, and then if you expose that to a virus, it will directly neutralize it, stop it from entering. So either stops viral entry or it stops the viral egress process, right? Should I stop or keep going out of respect? Sambung ya? Bisa. Yeah. Bismillah. Okay, so back to this. So you can see viral neutralization of different SARS-CoV-2 variants from ancestral all the way to Kappa. This is quite old work, but it's a proof of concept. What we're trying to show is there's a proof of concept of using this as an antiviral modality. I was going to click this for next slide. But anyways, next slide. Yeah. So here's another one. So we use the same technology where we put the anti-COVID nanobodies on the surface of EVs, but we put it in neural stem cells. Now, there's a story behind this. We actually showed a while ago that neural stem cell EVs have anti-inflammatory potential. We actually didn't believe it because we think a lot of these studies are bogus, right? We're like, oh, they all claim this, they go to the brain, you know, blah, blah. So we thought, lesson number 2, never believe what papers say, or do it yourself. I tell all my students, oh, but Adi, it's a Nature paper. I was like, I don't care. I have a science paper too, but others can say the same thing to me too, right? Always try yourself. So we did it, and guess what, right? We put it into lungs, and we did a nanocyte immunotranscriptomic study. So this is SARS-CoV-2 infected lungs, right? That one. And when you put the EVs, purified EVs, you can see this immune transcriptomic reprogramming in infected lungs, which shows that it has an anti-inflammatory activity. We're like, this is great, but it's not a lot, but it's great, right? It's got a lot of different genes are being switched off. Now, if you put it into the lungs of mice, so this is sparing doses of 2 doses, right? In a SARS-CoV-2 infected mice, you can see an amelioration of antiviral load there. It's not the best, but it's a proof of concept. So it's an idea that we're trying to do. Next slide. Here's another example where we put a full antibody, instead of a nanobody, a full antibody on the surface of EVs. So this is work done with my collaborators in China. And yes, I was doing that work at that time. This is against a virus called ECMV. So this virus is a pig virus. It's a veterinary virus. Not very serious nowadays. But in China, you know, it was serious at some point. It can cross to humans, but there's been no human-to-human cases. But it's a big issue for the pig industry in a lot of countries like China. So when we deliver these EVs into mice infected with EMCV, which is, by the way, a 7-day lethality mice, right? So a very lethal dose, right? You can see multi-organ antiviral activity across different organs. We're like, this is great, right? All of these examples I've shown you are just proof of concepts, right? And they all seem to work at different, you know, varying, you know, like intensities, degrees. So we're like, okay, let's try other things. So then, next slide. So then we thought, how about we load RNA, antiviral RNAs inside EVs, which is actually quite hard to do. So to spare you the molecular biology of this, so we developed a stable cell line that can actually produce EVs containing the RNA, the antiviral drug, right? So the RNA inside is the API, the active pharmaceutical ingredient, right, in there. And then we gave it to mice, SARS-CoV-2 infected, and you can see that you get this amelioration of lung antiviral activity. Now, here's the thing, right? We thought, oh, neural stem cell EVs, it should go to the brain. You know what? It didn't, right? A lot of studies says, you know, if EVs are from a certain cell type, there is a propensity for it to go back to its origin. Well, that's not true. It does go to the brain, but not at therapeutic efficacy, right? So that was nice. Next slide. So we were also interested. We're like, all right, so we can do that. How about we then play around with small cells? So we got interested in T cells because my PhD student was interested in HIV targeting, right? So we thought, how are we going to, we want to express a particular mRNA into T cells. So we looked at this old study, right? It's not my work, and they showed that, you know, HIV has this 5' untranslated region, right? But when you truncate it, this one, right, where the TAR domain is gone, suddenly you get a very high IRES activity, which is translation activity. We're like, we can use that information. How about we take HIV's idea, put it in front of another mRNA that you want to express, in this case, the green fluorescent protein, and then see what happens. and we load it into EVs and see if it's T-cell specific. So this activity is actually T-cell specific. We're like, great, we can actually develop a T-cell specific drug for not just HIV, could be other retroviral therapies or whatever cancer, right? I was looking for the next slide. Next. Yeah. So then we developed EVs engineered with this HIV truncated 5' UTR and slapped a GFP mRNA at the bit. And we asked the question, which cells express it preferentially? To cut a very long story short, it's only T cells. If you look at it in HEK293s, which are embryonic, kidney embryonic, and liver cells, it doesn't do it. So specifically in T cells. Next slide. And then when we, next slide. Yes, thank you. And then we designed some EVs. We're like, all right, we've got EVs with mRNA of interest. Now, I said, Fred, I want you to design EVs that can target specifically only to T cells. In this case, T cells, to put EVs with a targeting motif against CCR4, which, as you all know now, is preferential for HIV infection, right? And I asked the question, now which cells specifically express this mRNA? And lo and behold, only T cells. THP cells, which are monocytes, don't do it. All the other cells don't do it. So we're like, this is great. We're having a T cell specific therapy. Next slide. So then we thought, is there another way we can engineer EVs without going through the molecular biology? Because it takes months to develop. So I thought of an idea. I said, hang on a minute. I remember some old chemistry literature that shows me that aptamers, which are pieces of RNA, can bind specific T cells, right? So there's a particular aptamer that's been uncovered years ago. We thought, how about if we put a lipid at the end and embed it into the surface of the exosome, just by 1+1. No click chemistry, nothing fancy. It's just 1+1, and then give it a shake, and then pray to God that you get something at the end. And what you'll get at the end is an EV decorated with these aptamers on the surface. And those aptamers should bind to those CCR4 on T cells specifically. And lo and behold, compared to aptamer alone, you get this very nice enhancement. And this is flow cytometry data. Now, 20 to 10% may not be much to you folks, but for us that do EV engineering, it's actually very good, right? Because it's very hard to do engineering. So we thought, this is great. This is a good sign for it. Okay, next slide. Here's another virus I'm interested in. So this is work by Muskan. HTLV1, those who don't know, is very prevalent in indigenous communities around the world, especially in Japan. I believe it's endemic in Japan. 2.5 million people have HTLV1. HTLV1 is a class 6 Baltimore virus, i.e. it's like HIV. So it reverse transcribes its RNA into DNA and integrates into the. Unlike HIV, it actually causes a T cell type of leukemia, right? Majority of people are asymptomatic to it, but 5% of them develop these terrible disease, right? And in Australia, it is quite prevalent in the First Nations, Aboriginal, and Torres Strait Islander people, the remote communities. So this project is of importance to the sovereignty of protecting the First Nations people of Australia. So here we've developed some RNA therapeutics against HTLV. We discovered a HTLV link RNA that can actually stop the oncogene from being expressed. So in this case, it's TAX. So I told Muskan, I said, Muskan, how about you load this into EVs and see what happens. So she did that, and you can see that the EVs containing the short hairpin RNA actually knocks down that oncogenic target, which is great. Next slide. So other than short hairpin, we can also load long antisense RNAs. So long antisense RNAs are around 200 base pairs, like that COVID one I showed. So let's see if we can do it. And lo and behold, we did too, all right? So we're like, this is great. So we can actually show this not just for COVID, but so many other viruses. Next one. Sorry, next slide. Remember I said that, don't believe what papers say? So a lot of papers say, oh, this EV goes to brain, this one goes to brain. It's great that it goes to brain. It didn't work in our hands. So we tend to be very skeptical about these papers. So we're like, but when we did the studies, it does go to the brain, but not therapeutically efficacious at the doses we wanted. So what we did was, how about we engineer this? So we wrote this review, right? We're like, how about if we engineer these EVs with blood-brain barrier targeting moieties that allows the active uptake of these EVs to cross the blood-brain barrier? Because you all know a lot of drugs don't go to blood-brain barriers. If we have this solution, we can actually carry cargo, protein, mRNA, whatever you want, into the brain, right? Next slide. So that's what we did, and we spent a lot of months developing these stable clones that express these two magic proteins that will allow it to cross to the brain. So those of you who want this stable cell line, let me know. There's no material transfer needed, and there's no IP with this, but it took us a few months to do this, and it's stable, its transgene is stable in the cells. Next slide. So what we did was I told Ira, so Ira is a master student that works on this, and that's my postdoc. I said, how about you deliver this, right, by IV into the brain? So by IV, and then you can see a distribution in the brain. So compared to PBS alone, and at different time points, you can see the ones that have the blood-brain barrier targeting moiety, you can see a signal in the brain. So we're like, that's cool. We've never seen that before. Usually it's quite, like not much signal, but we're seeing that in the brain. We're like, we're onto something now. So right now we're just further finessing this, right? Another problem is when you inject EVs into an animal, a lot of them go to the liver. Actually, a majority, 80% of them go to the liver. First pass organs, right? This is a problem with pharmacokinetics. A lot of them just go to liver and spleen, right? So I actually personally don't like IV drugs, but the clinicians love IV drugs, especially for hospitalized patients. So it's like it's something that we're battling with. So that's great data. Okay, next slide. I don't know how much time I have left. Here's another one. So this is with UNAIR, Universitas Airlangga, right? So this is work with the chemistry department. We thought, forget about RNA, let's see if there's other modalities. So my collaborator from here developed these nano carbon dots. So these carbon dots come from, I think, bamboo or citric acid. So this carbon dot has on it a specific moiety called aminophenolboronic acid. And this particular moiety can target any glycoproteins on virus. We're like, oh, let's test the hypothesis. So we treated this and see if it neutralizes all the broad respiratory viruses, human metapneumovirus, influenza, 2 strains too, not just one, 2 strains, H1N1, what's this one, H2N3, H3N2 or something, I forgot, we've got too many strains, right? RSV, human metapneumovirus, all clinical strains, by the way, right, all clinical strains. And guess what, it neutralizes it beautifully. And when we did molecular docking backed up with data, it shows that it actually binds to the a receptor binding site of the hemagglutinin protein, the glycoprotein of influenza. We're like, this is great, right? What if we have a drug like this that can neutralize it quite potently? And the IC50s are crazy, like they're really good, like absolutely awesome, right? Next one. So here's my future slide. We tinker around with all sorts of technologies. People in my lab, I tell them, if you come to me with a project that's safe, leave my lab. Because we want to work on things that are risky that people have never done or don't want to do because it's high risk. So I tell my people, I want you guys to embark on very high risk projects because it's got high rewards, right? And if it's failure, then we come up with new things. So we're trying a very risky thing. So here's another risky thing we're doing, right? There's an mRNA technology we're trying to develop called TrimAway. TrimAway is great because it's these two particular proteins you put a nanobody and onto it a TRIM21. TRIM21 naturally gets taken up by the proteasome and it will degrade it. What if that nanobody can target, let's say, an insoluble protein in the cytosol? We're like, this would be great for like Creutzfeldt-Jakob disease, Kuru, these prion diseases, 0 treatment, right? No one knows how to target it. So we thought, hey, let's come up with this modality and target something like Kuru, any prion diseases. crazy project, but high rewards, right? And we're also exploring these carbon nanodots. I'm very convinced now that these are very good things. Next slide. So we're also moving away from lipid nanoparticles. Yes, during the pandemic and all the lipid nanoparticle hoo-ha, you know, Pfizer versus Moderna, 1 billion people vaccinated. Great, vaccines are great, but the lipid nanoparticles have a problem. Now we have antibodies raised against a, a particular thing on the lipid nanoparticles called PEG, PEG. And I don't know what the ramifications are in the future, but I don't like having PEG circulating in our body. So I don't like having lipid nanoparticles, synthetic ones, for too long. So I'm not sure. We'll probably see a 10-year retrospective study to see the effects of these lipid nanoparticle therapeutics in humans, right? So I told Zaki, I said, look, let's develop this nanodot, and you know what? Let's put some RNA outside of it. And he's like, are you sure? I said, yeah, let's try that. And he argues that, you know what, because the RNA is negatively charged, carbon dots are a different charge, if it binds it, it should stabilize it. So we did that. So sRNAs, if you put it in serum at 37 degrees, for over time, it degrades. So sRNA, it degrades, right? You put it with carbon dots, it doesn't degrade. We're like, great, it stabilizes it. So perhaps we can finesse this and do some, you know, chemistry, fun chemistry, and see whether we can develop a better platform where we can put multiple drugs against multiple viruses onto a single macromolecular platform. So that's what we're tinkering with. And my last slide, next slide, is this one. Further, again, acknowledgements to people, my old mentors, right? I've got I'm always having students coming to my lab. I've got 3 students from Unpad, Bandung, spending 2 months in my lab, and they're dentistry students. I haven't even spoken about that work. What I've shown you is only 25% of my work. I've got other things happening, right? And these are the people I need to thank, not me. They do all the work. Everything is done by them. And thank you for your attention, and I'm look forward to having more discussions. Terima kasih.
Okay, thank you very much for Dr. Adi. Thank you. And now we move to the next session is a Q&A. But first, our panelists, Prof. Lucia, maybe have a question for our speaker, for Dr. Nurhaliza and Dr. Adi, please.
Based on your presentation, there is hope for overcoming viral infection, whatever, by targeting the immune system and or acting directly on virus gene. However, there are certainly still many challenges regarding the treatment of this viral infection and associated immune response. And, I have a few questions. First for Dr. Nur. In many emerging viral infections, soft tissue damage is often driven by an excessive immune response, like as a cytokine storm, rather than directly viral cytopathic effect. Based on your dynamic you have observed, where is the tipping point between protective immunity and immunopathology, and how early can we really predict this safe?
Can you repeat again the question? Sorry. What?
Based on the dynamic you have observed,
okay.
Where is the tipping point between protective immunity and immunopathology, and how early can we really predict this safe?
Okay, so I think as we know, it's a bit hard to know the balance. I mean, to actually do the balance is hard. For me, I think it's more on being aware there's possibility of the pathological effect of anything. It can be antiviral, it can be vaccine. Because to have a perfect therapy it's quite impossible, I think, I mean, now, I'm not sure in the future we might have a perfect therapy for certain disease. But let's work on not denying the possibility of what you are developing can cause harm. And it's more on being aware of it and then try to develop something that can help to manage that kind of symptoms if it's really happen in the future.
Okay.
Is that okay?
Yes.
All right.
For Dr. Adi, they use extracellular vehicle or EVs to ensure RNA molecule enter target cells such as lung cell without being degraded by body enzyme. Represent novel approach to addressing the virus infection. Yes. Okay. To produce the biological vehicle like EV to meet pandemic reality emerging needs remain challenges, requiring significant investment and complex infrastructure.
Correct.
Yeah. How do Dr. Adi propose overcoming this challenge?
Interesting. The company I work for actually makes therapeutic EVs. We develop therapeutic EVs against viral diseases. So it is challenging. It's a significant amount of infrastructure and funding, right? So we have a pilot plant right now in Taipei. And I won't tell you how much it cost us to do it. But we rely on government also to support. So not we as in the company, as the universities and researchers, we rely on governments to support building these capabilities. Now, how hard is it for us to make these EVs? The hardest bit is to engineer it. The easiest part is producing lots of it. Because all you need is cells growing in a bioreactor. You just let them grow, spin it around, and they make lots of it in the supernatant. And then you then have a process development purification team that will purify this product. So that can be done by any expert protein purification team. The hardest is engineering it. But once you finesse that, then you can actually engineer all sorts of things against different viruses. The great thing about RNA is modular. What it means is you can actually change, all you need is the RNA sequence that you're targeting. That's all you need. There's no natural product drug delivery pipeline. There's nothing like that, right? Actually, to develop an RNA drug that's synthetic takes me 2 weeks. And you can deploy that really fast. Look at how long it took for Pfizer to deploy their vaccine. It didn't take very long, right? Forget about the conspiracy theories. I'm not into the conspiracy theories, but it took them a year. It took them a year. Because it's mRNA. All you need is the coding sequence, right? So with EVs, the bottleneck is the actual engineering. That's the problem.
Okay, thank you. One more question for Dr. Nur and Dr. Adi also. We know that RNA virus like influenza and coronavirus mutated rapidly. So my question, how the response immune for Dr. Nur and for Dr. Adi, if the genetic virus sequence change, how the EV can we also adjust EV to maintain effectiveness? For Nur, the response immune, and then for Dr. Adi, the IV.
I'm so sorry if you need to repeat it. Is it regarding different, the cross-reactivity?
Yeah, I mean that we know that RNA virus, it's a rapidly mutation.
Oh, okay,
Yeah,
all right,
yes.
all right.
And how about the response immune? It's the same?
It definitely not. Yeah, that's why if it's talking about influenza, I think you probably know better than me for influenza. That's why if we talk about the vaccine, it needs to be taken, seasonally, right? So they change. So with that, the immune response will change as well. Yeah.
I mean that if we first immunization with influenza and strain 1, and then it's also it's effective if we infect with strain another strain.
It will not. It will not. Yeah. I mean that's why they always need to develop a new type of vaccines against the new virus, especially in influenza. Yeah. So it will not. Yep.
Yeah, so for antiviral RNA therapeutics, we target what we call the highly conserved regions. Now, in influenza, there are highly conserved regions, and for a lot of RNA viruses, we all know it's in the polymerase region, right? It's either 3' UTR, or 5' UTR, or the polymerase, because it doesn't have immune pressure, right, for it to mutate. Now, but siRNA, just like other traditional drugs, remdesivir, ribavirin, can induce mutations over time, right? How do we navigate that, circumnavigate that? We can use a cocktail of RNA drugs that targets different regions so that the mutational pressure, the pressure to mutate is less for the virus, right? And as I said, it's not just influenza, could be RSV, human metapneumovirus, right? They all mutate at the end of the day. So it's about how smart you can design your drug, right? If I have a influenza drug that I want to last, and my drug also, I didn't mention, spans avian flu, also swine flu genes. So it's highly conserved across that. All my COVID drug, that's against MERS also. SARS-CoV-1, MERS, all the way to beta coronaviruses. So there's no point patenting a drug and designing one if it's only against SARS-CoV-2 Omicron, right? It has to be highly conserved region, and that hunting around takes time, right? And you got to test it. Yeah, so same thing with EVs, right? So EVs, whatever RNA put in, it's got to be highly conserved, right? It's got to be a good. So the API, the active pharmaceutical ingredient, has to be something that's against multiple strains. Will it ever be resistant to mutations? No drug will be. Impossible, right? This is the pressure that viruses have against us, over the evolutionary period, evolutionary timeline that we've been around on planet Earth. So yeah, they're going to keep mutating, and we're going to keep pumping out new drugs every end.
That's very interesting. May I add another question to Prof. Lucia? Yeah. Do you think that this highly conserved part of the mRNAs, the part, the exon or intron, or whatever part that is carrying the virogenic part, do you think?
You mean on the viral RNA?
Yeah.
Yeah.
This highly conserved part that you mentioned.
Yes. Yes.
Because we are talking about highly mutated virus, right?
Yeah.
Hence, we need the seasonal vaccine.
Correct.
Because it keeps mutating every time.
Yeah.
So you mentioned, we can somehow playing with the highly conserved part.
You can. So in influenza, so the vaccine is, H and N, right? Hemagglutinin and neuraminidase. Now, they're highly variable, right? There's a reason why it's variable, because it's exposed. But there are highly conserved regions in the hemagglutinin epitopes. The problem is the immune system cannot see it.
Yeah.
That's a problem.
Yeah.
So that's why we can't develop highly conserved vaccines, right?
Yeah.
It's hidden for a reason, right? But as a drug, you can.
Well, can you use somehow a step in the immunohistochemistry called the epitope antigen revealing steps that these specific epitopes can show off, so you can be more specific.
Yeah, it's possible. Yeah, I mean there are. Well, I've found residues in hemagglutinin. Hemagglutinin is a poor target, in my opinion, as a drug. It's a very poor target. But I found some residues that are highly conserved across various, including swine flu. Right? I'm like, what if I denature that protein and just show it to the immune, But I'm not in vaccine space, so maybe you can do that. No,
Maybe if
I have a job in your lab. I will do it. Yeah, I will move to
yeah, we can
the gene, whatever.
Yeah, we can apply for a grant together.
Yeah, I think I have a question, if I may. Is any question from the participants first? Yeah,
Maybe from the online. Any question from the Zoom? There's not yet. Okay, maybe a question from this room. There are. Yeah.
it's very interesting, no? Students, yeah. Open invitation from Dr. Adi's lab, yeah. Anyone can visit him. 2 months probably that we can do some joint work, yeah. Dr. Adi mentioned that he's not go in depth yet into the immune response, because we know that the EV has so many types, right? The exosomes, the apoptosis body. Sometimes I thought that you are using the more natural EV like apoptosis body, but you don't actually.
Yeah.
What kind of base that you developed? Not PEG. I know the bamboo thing, the charcoal something. Is it the only one?
No, we just, there are so many receptors you can target. We just decided to target ApoE and transferrin receptor for now, to allow it to enter. But you can engineer anything on EVs. All your favorite protein, by the way. Anything. You want a protein that can engage natural killer cells? Yeah, go for it. Anything you want, Yeah.
Because as a medical doctor, we used to think holistically. So this is our biggest challenge. We overthink
Exactly.
everything.
Yeah.
Not like you you can keep focused, like you want to target this, and then you keep on. But
All my projects are high failure. They're high failure rates.
Yeah.
I like talking about it, but the success rate's 10%. But when it's successful,
if you
I'll
are
spin out a company
successful,
straight away.
it will be Nobel, I think, Nobel
Yeah,
Prize,
Nobel.
I think, inshallah,
Yeah, inshallah. Nobel doesn't mean successful either. It could be rubbish. A lot of my projects were rubbish, but the successful ones
I won't say
you
rubbish,
spin
but,
out.
I think it is interesting to have more researchers like you. Like you keep thinking outside the box.
Yeah, you have to.
mentioned about the cholesterol binding. If I'm a medical doctor, I will start to think which cholesterol. So many cholesterol,
Yeah,
LDL.
of course.
This is
Yeah.
LDL, I think.
It's LDL.
The LDL.
Yeah.
What about the HDL?
Yeah.
For the students, what about apoprotein E? Yes. What about the other apoprotein? I think they think I am still thinking that kind of like my students, I think. But this is interesting, I think. Very interesting. For Dr. Hafiza, I have a question, A simple question, because I'm not the flavivirus expert, Prof. Chris also, You mentioned that flavi is yellow, right? So many flavivirus means yellow, but not all flavivirus infection cause icterus, jaundice, yellow, not all, we know that they have basically similar structure, mRNA, something like that. So what causes the difference? The yellow fever can cause icterus, not for the dengue fever, not common, the icterus. You know the icterus, the yellow thing? Do you know? Okay, so we here in the clinic, if we got the yellow fever, we can see in the sclera, the patient will go yellow, hence the name yellow virus. Yellow virus is a part of the flavivirus. Also the dengue virus, JE virus, West Nile virus. But how come the yellow virus causes the icterus? We know that it has the predilection to the hepatocytes, hence altering the bilirubin metabolism. We know that so far. But not in other viruses like dengue. It has another predilection, which is the myeloid cells lineage. Is it? Yes? And the Zika virus, Dr. Hafiza, the predilection is more into the nervous system, hence the microcephaly, I think. But what about the biomolecular underlying mechanism?
Maybe you can teach us more. Thank you.
Okay. So the genus contains, 70 viruses into 52 viral species. And symptomatically, you can group viruses together. So there's the hemorrhagic viruses, like dengue and a few others. There's the encephalitic ones, such as Japanese encephalitis, Zika, arguably. So that's the main two classes, the encephalitic and the hemorrhagic. So yellow fever is because the liver is badly damaged. So jaundice, yeah. So that's why the skin goes yellow. And the sclera, you're right, goes yellow. And I know there's probably a picture on one of my slides of it. But that's because of the real liver damage that was going on. And none of the other ones damaged the liver to that extent. So that's why it's the only one.
Any specific epitopes? We are talking about the epitopes, right? Maybe why a virus preserve that specific epitopes in the liver cells, and why the other one specifically, like, for example, lymphocyte cells.
Well,
Any specific epitopes, you think?
I'm not sure about epitopes, but every virus has a specific tropism that is going to go for different tissues. So dengue replicates very well in the liver. That's why we do a lot of investigation in liver cell lines. What else? Some of the other ones don't replicate very well in the liver. So there's many cell types that are infected or can be infected, but each virus has its own unique tropism. So I'm going to infect these cells, this tissue, etc., and that's all different. The liver is quite a common place for infection of a number of viruses. Dengue replicates very well in there. As we say, yellow fever. I can't think any more liver-tropic ones. No, it's not a flavivirus, my friend. The hepatitis C, yeah, okay, but I don't think that's a flavi. It's a,
Oh, we're talking about flavi.
it's a pestivirus or something like that, I think.
Yeah. I think mainly dengue and also Zika. Zika is pantropic because it can infect more than one type of cells. So it's either it becomes very specific or it can be more than one cells.
Sometimes chickpea virus can cause jaundice also, right?
Yes.
Chikungunya. In Indonesia,
Mainly
it's
they
so
will lead to
yellow.
arthritis, right?
That's interesting, isn't it? Dengue fever proliferate heavily in the liver, but not causing jaundice. Somehow different with the, for example, yellow fever virus.
Yep. Yep.
They are all from the flavivirus. This is very interesting. Dr. Adi, because I'm a neuroscientist, I cannot help to ask you. The blood-brain barrier.
Yes.
You show us two different diagrams.
Yeah.
The first one, I think, it's more into the diffusion osmotic
Yep.
process. The other one is via
active.
the channel.
Yeah.
You are mentioning the I forgot the
Transferrin
name.
receptor, transferrin receptor.
The protein using
Yeah,
the channel,
ApoE,
something like that.
yeah.
What about the other component of the blood-brain barrier?
I
such
agree.
as the
We,
podocyte, of the astrocyte, and everything. Did you do everything in vitro, docking study,
Yes.
or what?
So
Animal
we
rodents?
only screen the ones that we think is correct. Obviously, there are other blood-brain barrier targets that should be pursued, but we chose ApoE and transferrin through an iterative process, and then we did it in vitro. Actually, the way we work in our lab is do it in animals first, then in vitro.
Backwards.
Backwards. Because I tell all my students, go hard, fail hard. If you fail hard, just come up with another project. So we go animal first, and it's worked. Now I tell my student, hey, now do it in
Go
vitro.
look in depth
Then
into the
you understand the
parameter.
mechanism. So when you develop something, don't do the mechanism and then animal. I say go straight to animals. If it doesn't work, then don't waste your PhD time. So the way I do things is very different. It's non-traditional.
Can I ask a bit?
But we burn a lot of money.
Yes, the money,
We
and
do.
then your iCook, they allow you to do without any in vitro work?
Of course.
We don't.
Yeah, yeah.
Unlimited funding, probably.
Not unlimited. I mean,
That's one significant difference. And then they're so lenient in terms of using animals.
yeah.
Too backward,
No,
so many risks.
when we apply for ethics, we apply it generally.
Yeah.
You know, like we do animal work every day. You know, every day, right? So we know the rules. Yeah.
So what your animal model is? Mice, rodents?
All mice.
Pardon?
Mice.
Mice.
Yeah.
Okay.
Yeah. Rodents are expensive.
So many questions for you, but because of the time, I think, thank you very much.
Thank you
Bu
very
Vivi.
much. Yeah, thank you, Prof. Unfortunately, we have a limited time and time's up. It's very interesting. Maybe we can continue at the guest lecture session, Prof. Iska, right? Yeah. Okay, once again, give applause for our remarkable speaker. Thank you very much. Thank you. And now this is the end of the this session. And now I'm back for MC. Thank you.
Okay, thank you so much, Dr. Vivin. Yes, excuse me, sir. Thank you so much. Once again, thank you so much, Dr. Nur Hafiz and Mr. Adi, for that incredible, interesting session that I've also been listening to. We would now like to present a token of appreciation to you both, as well as to our panelists today, to Professor Lucia. We would also like to invite you to come forward and receive a token of appreciation, which will be given by the Master's Program Study Coordinator of Immunology of the Postgraduate School of Universitas Airlangga. Once again, please give a round of applause for our keynote speakers and panelists today. Okay, ladies and gentlemen, it is now time for my favorite part of the day and also your favorite part of the day, which is the lunch break, which will commence until 1:15 p.m. So you may now take your break, take your prayers if needed. The prayer room is on the first floor, and please be back in the conference room by 1:15 p.m. Well, thank you so much and have a lovely lunch break. untuk para peserta, kami himbau untuk makanan siangnya sudah disiapkan di luar. Dipersilakan untuk bisa langsung ambil box makan siangnya yang telah disediakan oleh panitia. To our participants, the lunch boxes have already been provided outside of the room. So we invite you to come and take the boxes that have already been provided by the organizing committee.
Kalau terlalu tinggi jadi autoimun. Kita sadari tahu nggak sih, sistem imun itu harus berada dalam keadaan homeostasis. Kalau terlalu turun, jadi imunodeficiency. Kalau terlalu tinggi, jadi autoimun. Tanpa kita sadari, justru kegiatan sehari kita lah yang menjaga kondisi homeostatis itu.
Seperti tidur yang cukup. Saat tidur, tubuh memperbaiki dan mempersiapkan sistem imun untuk menghadapi hari berikutnya. Makan makanan sehat dan bergizi. Makanan bergizi memberi nutrisi yang dibutuhkan sel imun agar dapat bekerja dengan optimal. Olahraga jangan di-skip ya. Olahraga rutin membantu menjaga sistem imun tetap aktif dan tubuh tetap bugar. Stay hydrated. Jangan lupa banyak minum air putih. Hidrasi yang cukup membantu menjaga sirkulasi darah dan cairan limfa sehingga patroli sel imun ke seluruh tubuh lebih efektif. Biar anti stres, healing jangan lupa. Lakuin hobi yang kalian suka. Misalnya main game, baca buku, dan hangout. Saat stres, kortisol yang disekresikan lewat HPA axis meningkat, sitokin proinflamasi juga meningkat. Makanya harus tetap happy ya guys ya.
Tuh, mudah banget kan?
Yuk usahain hal-hal tersebut untuk menjaga hemostasis sistem imun kita.
Healthy habits, healthy immunity.
Bagaimana sistem imun bekerja? Ketika mikroorganisme patogen masuk ke dalam tubuh, tubuh tidak langsung mengandalkan satu jenis pertahanan saja. Sistem imun bekerja secara bertahap dan terkoordinasi. Mari kita ikuti bagaimana tubuh menghadapi infeksi tersebut. Patogen mulai masuk ke dalam tubuh. Bayangkan terdapat suatu mikroorganisme patogen yang mencoba masuk ke dalam tubuh. Begitu patogen mencapai permukaan tubuh, pertahanan pertama yang dihadapinya adalah barier fisik dan kimia. Ketika patogen berhasil melewati barier fisik dan kimia tubuh, tubuh akan mengaktifkan sistem imun inert. Imunitas inert merupakan pertahanan pertama yang bekerja dengan cepat dan tidak spesifik terhadap jenis patogen tertentu. Jika patogen masih bertahan, sistem imun inert akan membantu mengaktifkan imunitas adaptif melalui presentasi antigen oleh sel dendritik. Imunitas adaptif bekerja lebih spesifik terhadap antigen tertentu. Setelah infeksi teratasi, sebagian sel T dan B akan menjadi sel memori. Sel inilah yang memungkinkan tubuh memberikan respons yang lebih cepat dan kuat ketika menghadapi patogen yang sama di kemudian hari. Sekarang, kalian sudah tahu kan bagaimana sistem imun bekerja. Yuk jaga kesehatan dengan rajin olahraga dan makan makanan bergizi.
sepak di Indonesia meningkat pesat tahun 2026. Yuk, kita lihat animasi edukasi untuk tahu lebih lanjut. Anak kecil gampang tertular campak karena sistem imun masih berkembang. Bayangkan hari pertama, sekolah dengan teman-teman baru yang banyak, membawa virus ke sekolah, lingkungan baru, paparan baru. Disinilah sistem imun bekerja. Tubuh mempunyai database imun. Banyak patogen pada tubuh anak yang baru dan belum dikenali termasuk virus campak. Hal ini menyebabkan sel B dan sel T memori bekerja. Vaksin AMR atau MMR membantu sistem imun mengenali campak dan membangun kekebalan. Jadi ingat, anak bukan makin kuat karena sering sakit. Imunnya sedang berkembang dan vaksin campak MR membantu melindunginya.
Hi, my name is Lydia. I'm an immunology student and also an XLE survivor. Maintaining balanced immune system isn't easy, but when you have a history of autoimmune disease, it will be a bit tricky. Why? Because you have to deal with hair loss, bruises or races, and also never-ending fatigueness even when you are in a remission state already. But hey, no worries. I've been living with SLE for almost 10 years and I will show you how I got my immune system. First thing first, to exercise. Excessive sun exposure may need to be avoided, but a little sunlight in the morning is still necessary to help activate vitamin D, which is good for our immune system. Second one, rest. You need to know when to rest. In my cases, when my body starts overheating because of the systemic inflammation, I rest for a couple days until I'm feeling better. Therefore, the third one, find a flexible job so you can manage your immune system with the work-rest balance and also a job which makes you happy. Fourth, don't forget to maintain your spiritual health so you can lay every life problems to your God and just live in peace. Fifth, maintain your stress, go healing. And last optional tips, silliness can make you more happy. Hope this help.
Judulnya sempat heboh, hantavirus, calon corona kedua. Eits, jangan pari dulu.
Pada April 2026, 8 penumpang kapal pesiar MV Hondius terkonfirmasi terinfeksi adenovirus. 3 di antaranya meninggal dan pada 2 Juli 2026, WHO menyatakan bahwa wabah ini berakhir.
Kenapa penyakit ini tidak menjadi pandemi? Jawabannya terletak pada imunologi. Dikarenakan tikus liar yang sering kita ketahui membawa adanya hantavirus tanpa rasa sakit, sehingga sistem imunnya dapat mengendalikan virus dan tidak menimbulkan peradangan secara berlebih.
Pada manusia justru terjadi sebaliknya. Kerusakan umumnya terjadi akibat respon imun yang berlebihan. Badai sitokin memicu pembuluh darah bocor hingga paru-paru terisi oleh cairan.
Meskipun begitu, Andes virus sulit menjadi pandemi. Penularannya membutuhkan kontak yang sangat erat dan baru terjadi apabila timbul gejala.
Jadi, ancaman sebenarnya bukan hanya sekedar virusnya, tetapi bagaimana kita dapat mencegah kontak antara hewan liar dan juga manusia.
Guard the animals and we guard everyone with control.
Kenapa? Rasanya pusing juga. Pucat banget loh. Bentar ya, punya sesuatu buat kamu. Nah, kamu butuh ini supaya kamu bisa melindungi. Makasih ya, Bian. Udah, sudah dulu. Iya. Loh, kamu lagi sakit. Kalau makan kayak gini sih nggak bagus aja untuk kesehatan kamu dan imun kamu. Emang imun apaan, Bian? Nah, supaya kita tahu tentang imun, yuk kita tanya Mas Imin. Mas Imin. Yuk, yuk.
Halo semuanya, kenalin aku, Mas Imin. Imun aku. Bentar. Nah, ini udah pas. Sekarang aku mau jawab pertanyaan terkait apa sih sistem imun itu. Jadi teman-teman, sistem imun itu adalah sistem pertahanan tubuh kita. Ibaratnya tubuh kita itu punya tentara-tentara yang siap untuk melawan berbagai macam penyakit yang masuk di tubuh kita. Nah, untuk tahu lebih dalam, kita harus memahami konsep triad epidemiologi. Apa aja sih itu?
Pertama adalah agen, yakni faktor yang secara langsung dapat menyebabkan penyakit. Kemudian ada host, yakni individu yang dapat terkena penyakit. Lalu disusul dengan environment, yakni kondisi di sekitar host yang mempengaruhi peluang agen bertemu dengan host atau berkembang biak.
Kalau dari triad epidemiologi tadi nggak imbang, itu bisa bikin suatu penyakit. Contohnya seperti flu bilang tadi itu. Maka dari itu kita bisa menerapkan PHBS atau perilaku hidup bersih dan sehat seperti mencuci tangan, menjaga kebersihan diri dan juga lingkungan, serta tunjang dengan makan makanan yang kaya akan nutrisi agar sistem imun kita semakin kuat untuk melawan berbagai penyakit yang ada. Maka dari itu, mari semuanya kita perkuat imun tubuh kita agar tubuh kita terjauh dari berbagai penyakit.
sini aku ingin kilas balik tentang sistem imunku dalam mempertahankan sistem kekebalan tubuh. Aku bekerja selama 12 jam tanpa memperhatikan asupan nutrisi dan kesehatan lingkungan dan yang terjadi pada sistem kekebalan tubuhku. Di umur 20 tahun ke atas aku terkena campak lagi dan harus diopname. Pertama, tidur yang cukup, makan makanan bergizi dan seimbang, olahraga, jaga kebersihan, dan yang terakhir, healing. Your immune system deserve your daily care. Bye!
Sering merasa lelah padahal sudah tidur. Bisa jadi sistem imunmu sedang kelelahan. Tanpa sadar, kebiasaanmu justru merusaknya.
Ada beberapa kebiasaan yang merusak sistem imun kita.
Pertama adalah begadang. Terlalu sering mengonsumsi junk food, stres yang berlebihan dan tidak dikelola, serta kurang gerak karena duduk seharian. Semua ini perlahan merusak sistem imunmu.
Beberapa tips untuk menjaga sistem imun.
Tidur 7-8 jam. Sama dengan recharge imun. %70% imun ada di usus. Isi piringmu warna-warni. Gerak 30 menit setiap hari. Tarik napas dalam 1 menit saja sudah membantu. Cuci tangan dan vaksin sama dengan benteng ekstra. Jaga guardianmu, jaga bersama Imunair 2026. Imunologi bangkit! Sistem imun adalah pertahanan tubuh kita. Ia bekerja setiap hari, melawan kuman, virus, dan bakteri yang mencoba masuk ke dalam tubuh. Pahlawan utamanya adalah sel darah putih yang mendeteksi dan menghancurkan benda asing, didukung oleh kelenjar getah bening dan organ limpa. Sel ini memproduksi antibodi, yaitu protein khusus yang bertugas mengikat, menonaktifkan, dan merekam memori virus untuk perlindungan jangka panjang. Ada 2 jenis pertahanan, imun bawaan yang merespons ancaman secara cepat, dan imun adaptif yang menyerang patogen secara spesifik. Hati-hati, kurang tidur, tingkat stres yang tinggi, pola makan tidak sehat, hingga asap rokok bisa melemahkan sistem pertahananmu. Jika sistem ini rusak, tubuh akan mudah infeksi, luka sulit sembuh, bahkan bisa memicu penyakit autoimun, di mana sel sehat diserang. Perkuat imunmu, makan bergizi, tidur 7-9 jam, rutin olahraga, dan penuhi asupan vitamin. Pola hidup sehat, imun kuat.
Kenapa sih kita harus jaga pola hidup kita? Karena sistem imun atau penjaga tubuh kita butuh nutrisi dan istirahat yang cukup biar nggak mogok saat lawan virus.
Jaga penjagamu, saya lawan.
Ingat sekali lagi, akan saya lawan.
kalau baterai kamu sisa 20%, kamu pasti charger kan? Tapi kalau kamu yang lagi low baterai, pasti tubuh kamu akan dipaksa jalan terus. Padahal sistem imun kita juga butuh kondisi yang baik untuk bekerja secara optimal. Terus gimana kamu charger Guardian? Nah, yang pertama itu kita harus tidur yang cukup. Yang kedua, makan bergizi yang seimbang. Yang ketiga, jangan lupa bergerak. Yang keempat, kelola stres. Dan yang terakhir itu yang kelima, jangan lupa vaksinasi. Tapi ingat, tujuannya itu bukan bikin sistem imun kita sekuat kuatnya, tapi tujuannya agar sistem imun kita tetap seimbang dan dapat merespon dengan tepat.
Kalau HP aja kamu charge, masa Guardian tubuh enggak?
Bayangkan ada seseorang yang menjaga tubuhmu 24 jam per hari.
Dia nggak pernah tidur loh.
Nggak libur juga.
Dan setiap hari dia melawan jutaan ancaman.
Namanya sistem imun.
Sistem imun merupakan sistem pertahanan alami tubuh yang melindungi kita dari berbagai ancaman seperti bakteri, virus, mikroorganisme, dan berbagai macam penyakit lainnya.
Tapi ada satu hal yang sering kita lupa.
Kalau kita ingin terus dilindungi, kita juga harus menjaga sang penjaga. Nah, ini masalahnya.
Lalu, bagaimana cara kita untuk menjaga sistem imun?
5 cara menjaga sistem imun. Yang pertama, makan makanan yang bergizi. Yang kedua, minum air yang cukup. Yang ketiga, selalu menjaga kebersihan. Yang keempat, tidur yang cukup. Dan yang kelima, selalu berolahraga.
Jadi, siapa yang menjaga kita?
Sistem imun.
Dan siapa yang harus menjaga sistem imun?
Kita.
Sakit tenggorokan biasa bisa berujung merusak jantung, bahkan meninggal dunia. Ini difteri, dan kasusnya lagi naik di Indonesia. Difteri disebabkan bakteri yang melepaskan racun ke tubuh, bisa merusak tenggorokan, jantung, ginjal, dan saraf. Tahun ini kasusnya dilaporkan di banyak daerah, dari Papua Barat Daya, NTB, Kalimantan Barat, sampai Jakarta dan Jawa Barat dengan puluhan nyawa melayang. Padahal vaksinnya udah ada di program imunisasi dasar sejak bayi. Tapi kalau cakupan imunisasi turun, bakteri ini dapat celah buat menyebar lagi. Ini yang disebut re-emerging disease. Yuk luruskan bareng-bareng. 1. Difteri kan penyakit zaman dulu, faktanya ini re-emerging disease yang aktif jadi KLB tahun ini. 2. Anak jarang sakit, nggak perlu vaksin DPT, padahal anak yang belum diimunisasi lengkap justru paling berisiko kena komplikasi berat. 3. Nanti aja vaksinnya, penularan difteri cepat lewat batuk dan bersin, menunda berarti membuka celah lebih luas. Difteri, campak, polio, semua bisa dicegah kalau imunisasi kita lengkap dan tepat waktu. Ini bukan cuma soal diri sendiri, tapi soal menjaga orang-orang di sekitar kita juga. Cek status imunisasimu dan keluarga sekarang, terutama vaksin DPT. Jangan tunggu jadi kasus berikutnya. Guard your guardian, mulai dari vaksin.
Dokter, sistem imun itu sebenarnya apa?
Sistem imun adalah sistem pertahanan yang melindungi tubuh kita dari berbagai ancaman.
Berarti sistem imun bertugas melawan semua virus, jamur, patogen, dan bakteri yang masuk ke tubuh.
Benar, sel imun akan mengenali benda asing, lalu memutuskan apakah dilawan atau tidak.
Lalu, bagaimana dengan autoimun? Banyak perempuan di atas 20 tahun yang
Untuk
terkena.
kejadian autoimun, sistem gagal mengenali target, sistem imun yang seharusnya menyerang patogen, malah menyerang tubuh sendiri.
Jadi, sel tubuh malah menyerang dirinya sendiri.
Respons imun yang keliru dapat menyebabkan peradangan pada jaringan tubuh. Coba bayangkan ada seorang satpam. Namun satpam tersebut justru mengira karyawan gedung sebagai pencuri.
Kemudian, apakah autoimun selalu menyerang bagian tubuh yang sama?
Tidak, pada beberapa penyakit autoimun, pada diabetes tipe 1, menyerang sel beta pankreas. Ada juga yang menyerang banyak organ sekaligus.
Lalu contoh penyakit autoimun apa saja, Dok?
Ada lupus menyerang organ seperti kulit, sendi, ginjal, dan bahkan jantung. Diabetes tipe 1 menyerang pankreas, ada kelenjar tiroid, dan masih banyak lagi.
Apa yang harus kita lakukan?
Kita dapat menerapkan gaya hidup sehat, jaga pola makan sehat dan seimbang, perbanyak konsumsi buah dan sayur, cukup aktivitas fisik, dan hindari stres.
Sekarang saya jadi lebih paham tentang autoimun. Terima kasih banyak, Dokter.
Bagus. Yang penting, jangan mendiagnosis sendiri dan konsultasikan dengan tenaga medis.
Pemberian vaksin. Jarum suntik memasukkan cairan vaksin ke dalam jaringan kulit, menyebarkan partikel antigen di dalamnya. Respons imun awal, sel dendritik mendekati dan menangkap antigen tersebut, lalu membawanya menuju kelenjar getah bening. Aktivasi sel imun di kelenjar getah bening. Sel dendritik mengaktifkan sel T helper, yang kemudian membantu mengaktifasi sel B. Produksi antibodi dan memori. Sel plasma yang terbentuk menghasilkan antibodi untuk menetralkan virus. Sementara sel memori terbentuk untuk mengingat ancaman tersebut. Infeksi kembali dan pertahanan. Saat virus kembali masuk di kemudian hari, sel memori langsung aktif memproduksi antibodi masif untuk menyerang dan menghancurkan virus. Perlindungan jangka panjang, sel memori tetap bersiaga di dalam tubuh, memberikan perisai perlindungan yang bertahan selama bertahun-tahun, dan dapat diperkuat kembali sewaktu-waktu menggunakan suntikan booster.
Halo guys, hari ini temenin keseharian aku yuk.
Kamu siapa? Novi, kamu lagi nge-vlog ya?
Iya. By the way,
Kamu kenal dia gak sih?
siapa? Aku kira semua kebiasaan ini cuman berdampak pada diriku. Ternyata ada yang ikut kewalahan.
Sebenarnya kamu siapa?
Aku sistem imun kamu. Ternyata dia selama ini sistem imun yang bekerja tanpa aku sadari.
Jadi selama ini kamu ikutin aku?
Iya,
Tapi kenapa kamu kelihatan capek?
karena setiap pilihan kamu itu beban buat aku juga. Kalau dia selalu menjaga aku, sekarang giliranku menjaga dia.
Barusan kita sudah melihat bahwa setiap pilihan yang kita lakukan dapat mempengaruhi bodyguard di dalam tubuh kita, yaitu sistem imun. Sistem imun adalah pertahanan alami yang bekerja tanpa henti untuk melindungi tubuh dari berbagai kuman dan penyakit. Karena itu, kebiasaan yang sederhana seperti tidur yang cukup, makanan yang bergizi, olahraga yang rutin, minum air yang cukup, dan mengelola stres juga sangat penting agar sistem imun dapat bekerja dengan optimal. Jadi, jangan tunggu sakit untuk mulai peduli. Yuk mulai jaga sistem imun kita dari hari ini.
Di tengah hiruk-pikuk kota, polusi udara, asap rokok, dan makanan instan, tanpa kita sadari, mengintai kesehatan keluarga setiap hari. Secara imunologi, polusi dan gaya hidup tidak sehat memicu lonjakan reactive oxygen species atau ROS yang merusak sel-sel imun vital seperti makrofag dan sel T.
Tapi, keluarga kami memilih antitesis, dimulai dari piring makan, mengonsumsi makanan sehat, kaya nutrisi, dan antioksidan bersama-sama. Diimbangi dengan rutinitas olahraga dan jalan kaki bersama di pagi hari untuk memperlancar sirkulasi darah dan menjaga vitalitas tubuh. Namun di usia 50-an, olahraga dan gaya hidup sehat saja terkadang belum cukup menetralkan sisa RS karena kemampuan alami tubuh memproduksi antioksidan mulai menurun. Disinilah pentingnya antioksidan harian, mulai dari konsumsi vitamin C, buah kaya penangkal radikal bebas, hingga melengkapinya dengan terapi inhalasi gas hidrogen secukupnya. Sinergi nutrisi rebusan, olahraga teratur, dan asupan aktif membalikan efek buruk polusi dan makanan tidak sehat, memperlancar sirkulasi, serta memulihkan stamina keluarga. Penggabungan seluruh antitesis ini, nutrisi, sirkulasi darah dari olahraga, serta kerja sinergis vitamin C dan molesilnya, sel T dan makrofag terlindungi secara optimal. Menciptakan lingkungan keluarga yang sehat dan bebas penyakit kini jadi nyata. Guarding the Guardian, persembahan dari Yeni and Farms.
Deadline besok, tugas belum selesai, akhirnya begadang lagi. Tapi tubuhmu merasakan dampaknya. Saat tidur, sel imun diproduksi. Kurang tidur buat kita rentan sakit. Saat tidur, tubuh memproduksi berbagai molekul dan sel imun yang membantu melawan infeksi. Ketika waktu tidur berkurang, respons imun juga dapat menurun. Setiap hari, jutaan bakteri dan virus menyerang. Namun, sistem imun kita siap melindungi. Dari benteng pertahanan hingga pasukan khusus, semuanya bekerja sama menjaga tubuh tetap sehat. Agar imun bekerja optimal, tidur 7-9 jam, makan makanan bergizi, rutin berolahraga, kelola stres dengan baik, cukupi kebutuhan cairan. Tidak ada makanan atau suplemen ajaib yang bisa langsung membuat imun kuat. Kuncinya adalah kebiasaan sehat yang dilakukan secara konsisten. Jaga gaya hidupmu dan biarkan sistem imun melakukan tugasnya. Kebiasaan sehat akan meningkatkan imunmu.
Hai Sobat Imun. Tahu nggak sih, sistem imun itu harus berada dalam keadaan homeostasis. Kalau terlalu turun, jadi imunodefisiensi.
Kalau terlalu tinggi, jadi autoimun.
Tanpa kita sadari, justru kegiatan sehari kita lah yang menjaga kondisi homeostatis itu.
Seperti tidur yang cukup. Saat tidur, tubuh memperbaiki dan mempersiapkan sistem imun untuk menghadapi hari berikutnya. Makan makanan sehat dan bergizi. Makanan bergizi memberi nutrisi yang dibutuhkan sel imun agar dapat bekerja dengan optimal. Olahraga jangan di-skip ya. Olahraga rutin membantu menjaga sistem imun tetap aktif dan tubuh tetap bugar. Stay hydrated. Jangan lupa banyak minum air putih. Hidrasi yang cukup membantu menjaga sirkulasi darah dan cairan limfa sehingga patroli sel imun ke seluruh tubuh lebih efektif. Biar anti-stres, healing jangan lupa. Lakuin hobi yang kalian suka. Misalnya main game, baca buku, dan hangout. Saat stres, kortisol yang disekresikan lewat HPA axis meningkat, sitokin proinflamasi juga meningkat. Makanya harus tetap happy ya guys ya.
Tuh, mudah banget kan?
Yuk usahain hal-hal tersebut untuk jaga dan mempertahankan sistem imun kita.
Healthy habits, healthy immunity.
Bagaimana sistem imun bekerja? Ketika mikroorganisme patogen masuk ke dalam tubuh, tubuh tidak langsung mengandalkan satu jenis pertahanan saja. Sistem imun bekerja secara bertahap dan terkoordinasi. Mari kita ikuti bagaimana tubuh menghadapi infeksi tersebut. Patogen mulai masuk ke dalam tubuh. Bayangkan terdapat suatu mikroorganisme patogen yang mencoba masuk ke dalam tubuh. Begitu patogen mencapai permukaan tubuh, pertahanan pertama yang dihadapinya adalah barier fisik dan kimia. Ketika patogen berhasil melewati barier fisik dan kimia tubuh, tubuh akan mengaktifkan sistem imun inert. Imunitas inert merupakan pertahanan pertama yang bekerja dengan cepat dan tidak spesifik terhadap jenis patogen tertentu. Jika patogen masih bertahan, sistem imun inert akan membantu mengaktifkan imunitas adaptif melalui presentasi antigen oleh sel dendritik. Imunitas adaptif bekerja lebih spesifik terhadap antigen tertentu. Setelah infeksi teratasi, sebagian sel T dan B akan menjadi sel memori. Sel inilah yang memungkinkan tubuh memberikan respons yang lebih cepat dan kuat ketika menghadapi patogen yang sama di kemudian hari. Sekarang, kalian sudah tahu kan bagaimana sistem imun bekerja. Yuk jaga kesehatan dengan rajin olahraga dan makan makanan bergizi.
Kasus campak di Indonesia meningkat. Pesat tahun 2026 yuk, kita lihat animasi edukasi untuk tahu lebih lanjut. Anak kecil gampang tertular campak karena sistem imun masih berkembang. Bayangkan hari pertama, sekolah dengan teman-teman baru yang banyak, membawa virus ke sekolah, lingkungan baru, paparan baru. Disinilah sistem imun bekerja. Tubuh mempunyai database imun. Banyak patogen pada tubuh anak yang baru dan belum dikenali termasuk virus campak. Hal ini menyebabkan sel B dan sel T memori bekerja. Vaksin AMR atau MMR membantu sistem imun mengenali campak dan membangun kekebalan. Jadi ingat, anak bukan makin kuat karena sering sakit. Imunnya sedang berkembang dan vaksin campak MR membantu melindunginya.
Hi, my name is Lydia. I'm an immunology student and also an XLE survivor. Maintaining balanced immune system isn't easy, but when you have a history of autoimmune disease, it will be a bit tricky. Why? Because you have to deal with hair loss, bruises or races, and also never-ending fatigueness even when you are in a remission state already. But hey, no worries. I've been living with SLE for almost 10 years and I will show you how I got my immune system. First thing first, to exercise. Excessive sun exposure may need to be avoided, but a little sunlight in the morning is still necessary to help activate vitamin D, which is good for our immune system. Second one, rest. You need to know when to rest. In my cases, when my body starts overheating because of the systemic inflammation, I rest for a couple days until I'm feeling better. Therefore, the third one, find a flexible job so you can manage your immune system with the work-rest balance and also a job which makes you happy. Fourth, don't forget to maintain your spiritual health so you can lay every life problems to your God and just live in peace. Fifth, maintain your stress, go healing. And last optional tips, silliness can make you more happy. Hope this help.
Judulnya sempat heboh. Hantavirus. Calon corona kedua. Eits, jangan pari dulu.
Pada April 2026, 8 penumpang kapal pesiar MV Hondius terkonfirmasi terinfeksi adenovirus. 3 diantaranya meninggal, dan pada 2 Juli 2026, WHO menyatakan bahwa wabah ini berakhir.
Kenapa penyakit ini tidak menjadi pandemi? Jawabannya terletak pada imunologi. Dikarenakan tikus liar yang sering kita ketahui membawa adanya hantavirus tanpa rasa sakit, sehingga sistem imunnya dapat mengendalikan virus dan tidak menimbulkan peradangan secara berlebih.
Pada manusia justru terjadi sebaliknya. Kerusakan umumnya terjadi akibat respon imun yang berlebihan. Badai sitokin memicu pembuluh darah bocor hingga paru-paru terisi oleh cairan.
Meskipun begitu, Andes virus sulit menjadi pandemi. Penularannya membutuhkan kontak yang sangat erat dan baru terjadi apabila timbul gejala.
Jadi, ancaman sebenarnya bukan hanya sekedar virusnya, tetapi bagaimana kita dapat mencegah kontak antara hewan liar dan juga manusia.
Guard the animals and we guard everyone with control.
Kenapa? Pusing juga. Pucat banget loh. Bentar ya, aku ada sesuatu buat kamu. Nah, kamu butuh ini supaya kamu bisa melindungi. Makasih ya, Bian. Udah dulu. Iya. Loh, kamu lagi sakit. Kalau makan kayak gini sih nggak bagus lah untuk kesehatan kamu dan imun kamu. Emang imun apaan sih, Nah, supaya kita tahu tentang imun, yuk kita tanya Mas Imin. Mas Imin. Bekerja. Yuk.
Halo semuanya, kenalin aku, Mas Imin. Imun aku. Bentar. Nah, ini udah pas. Sekarang aku mau jawab pertanyaan terkait apa sih sistem imun itu. Jadi teman-teman, sistem imun itu adalah sistem pertahanan tubuh kita. Ibaratnya tubuh kita itu punya tentara-tentara yang siap untuk melawan berbagai macam penyakit yang masuk di tubuh kita. Nah, untuk tahu lebih dalam, kita harus memahami konsep triad epidemiologi. Apa aja sih itu?
Pertama adalah agen, yakni faktor yang secara langsung dapat menyebabkan penyakit. Kemudian ada host, yakni individu yang dapat terkena penyakit. Lalu disusul dengan environment, yakni kondisi di sekitar host yang mempengaruhi peluang agen bertemu dengan host atau berkembang biak.
Kalau dari triad epidemiologi tadi nggak imbang, itu bisa bikin suatu penyakit. Contohnya seperti flu bilang tadi itu. Maka dari itu, kita bisa menerapkan PHBS atau perilaku hidup bersih dan sehat seperti mencuci tangan, menjaga kebersihan diri dan juga lingkungan, serta tunjang dengan makan makanan yang kaya akan nutrisi agar sistem imun kita semakin kuat untuk melawan berbagai penyakit yang ada. Maka dari itu, mari semuanya kita perkuat imun tubuh kita agar tubuh kita terjauh dari berbagai penyakit.
Hai, di sini aku ingin kilas balik tentang sistem imunku dalam mempertahankan sistem kekebalan tubuh. Aku bekerja bekerja selama 12 jam tanpa memperhatikan asupan nutrisi dan kesehatan lingkungan dan yang terjadi pada sistem kekebalan tubuhku. Di umur 20 tahun ke atas aku terkena campak lagi dan harus diopname. Pertama, tidur yang cukup, makan makanan bergizi dan seimbang, olahraga, jaga kebersihan, dan yang terakhir, healing your immune system deserve your daily care. Bye!
Sering merasa lelah padahal sudah tidur. Bisa jadi sistem imunmu sedang kelelahan. Tanpa sadar, kebiasaanmu justru merusaknya.
Ada beberapa kebiasaan yang merusak sistem imun kita.
Pertama adalah begadang. Terlalu sering mengonsumsi junk food, stres yang berlebihan dan tidak dikelola, serta kurang gerak karena duduk seharian. Semua ini perlahan merusak sistem imunmu. Beberapa tips untuk menjaga sistem imun. Tidur 7-8 jam. Sama dengan recharge imun. %70% imun ada di usus. Isi piringmu warna-warni. Gerak 30 menit setiap hari. Tarik napas dalam 1 menit saja sudah membantu. Cuci tangan dan vaksin sama dengan benteng ekstra. Jaga guardianmu, jaga bersama Immunaire 2026. Imunologi bangkit! Sistem imun adalah pertahanan tubuh kita. Ia bekerja setiap hari, melawan kuman, virus, dan bakteri yang mencoba masuk ke dalam tubuh. Pahlawan utamanya adalah sel darah putih yang mendeteksi dan menghancurkan benda asing, didukung oleh kelenjar getah bening dan organ limpa. Sel ini memproduksi antibodi, yaitu protein khusus yang bertugas mengikat, menonaktifkan, dan merekam memori virus untuk perlindungan jangka panjang. Ada 2 jenis pertahanan, imun bawaan yang merespons ancaman secara cepat, dan imun adaptif yang menyerang patogen secara spesifik. Hati-hati, kurang tidur, tingkat stres yang tinggi, pola makan tidak sehat, hingga asap rokok bisa melemahkan sistem pertahananmu. Jika sistem ini rusak, tubuh akan mudah infeksi, luka sulit sembuh, bahkan bisa memicu penyakit autoimun, di mana sel sehat diserang. Perkuat imunmu, makan bergizi, tidur 7-9 jam, rutin olahraga, dan penuhi asupan vitamin. Pola hidup sehat, imun kuat.
Kenapa sih kita harus jaga pola hidup kita? Karena sistem imun atau penjaga tubuh kita butuh nutrisi dan istirahat yang cukup biar nggak mogok saat lawan virus.
Jaga penjagamu, saya lawan.
Ingat sekali lagi, akan saya lawan.
baterai kamu sisa 20%, kamu pasti cari charger kan? Tapi kalau kamu yang lagi low baterai, pasti tubuh kamu akan dipaksa jalan terus. Padahal sistem imun kita juga butuh kondisi yang baik untuk bekerja secara optimal. Terus gimana kamu charger Guardian? Nah, yang pertama itu kita harus tidur yang cukup. Yang kedua, makan bergizi yang seimbang. Yang ketiga, jangan lupa bergerak. Yang keempat, kelola stres. Dan yang terakhir itu yang kelima, jangan lupa vaksinasi. Tapi ingat, tujuannya itu bukan bikin sistem imun kita super kuat ya, tapi tujuannya agar sistem imun kita tetap seimbang dan dapat merespon dengan tepat. Kalau HP aja kamu charge, masa Guardian tubuh enggak?
Bayangkan ada seseorang yang menjaga tubuhmu 24 jam per hari.
Dia nggak pernah tidur loh.
Nggak libur juga.
Dan setiap hari, dia melawan jutaan ancaman.
Namanya sistem imun.
Sistem imun merupakan sistem pertahanan alami tubuh yang melindungi kita dari berbagai ancaman seperti bakteri, virus, mikroorganisme, dan berbagai macam penyakit lainnya.
Tapi ada satu hal yang sering kita lupa.
Kalau kita ingin terus dilindungi, kita juga harus menjaga sang penjaga. Nah, ini masalahnya. Lalu, bagaimana cara kita untuk menjaga sistem imun? 5 cara menjaga sistem imun. Yang pertama, makan makanan yang bergizi. Yang kedua, minum air yang cukup. Yang ketiga, selalu menjaga kebersihan. Yang keempat, tidur yang cukup. Dan yang kelima, selalu berolahraga.
Jadi, siapa yang menjaga kita?
Sistem imun.
Dan siapa yang harus menjaga sistem imun?
Kita.
Sakit tenggorokan biasa bisa berujung merusak jantung, bahkan meninggal dunia. Ini difteri, dan kasusnya lagi naik di Indonesia. Difteri disebabkan bakteri yang melepaskan racun ke tubuh, bisa merusak tenggorokan, jantung, ginjal, dan saraf. Tahun ini kasusnya dilaporkan di banyak daerah, dari Papua Barat Daya, NTB, Kalimantan Barat, sampai Jakarta dan Jawa Barat dengan puluhan nyawa melayang. Padahal vaksinnya udah ada di program imunisasi dasar sejak bayi. Tapi kalau cakupan imunisasi turun, bakteri ini dapat celah buat menyebar lagi. Ini yang disebut re-emerging disease. Yuk luruskan bareng-bareng. 1. Difteri kan penyakit zaman dulu, faktanya ini re-emerging disease yang aktif jadi KLB tahun ini. 2. Anak jarang sakit, nggak perlu vaksin DPT, padahal anak yang belum diimunisasi lengkap justru paling berisiko kena komplikasi berat. 3. Nanti aja vaksinnya, penularan difteri cepat lewat batuk dan bersin, menunda berarti membuka celah lebih luas. Difteri, campak, polio, semua bisa dicegah kalau imunisasi kita lengkap dan tepat waktu. Ini bukan cuma soal diri sendiri, tapi soal menjaga orang-orang di sekitar kita juga. Cek status imunisasimu dan keluarga sekarang, terutama vaksin DPT. Jangan tunggu jadi kasus berikutnya. Guard your guardian, mulai dari vaksin.
Dokter, sistem imun itu sebenarnya apa?
Sistem imun adalah sistem pertahanan yang melindungi tubuh kita dari berbagai ancaman.
Berarti sistem imun bertugas melawan semua virus, jamur, patogen, dan bakteri yang masuk ke tubuh.
Benar, sel imun akan mengenali benda asing, lalu memutuskan apakah dilawan atau tidak.
Lalu, bagaimana dengan autoimun? Banyak perempuan di atas 20 tahun yang
Untuk
terkena.
kejadian autoimun, sistem gagal mengenali target, sistem imun yang seharusnya menyerang patogen, malah menyerang tubuh sendiri.
Jadi, sel tubuh malah menyerang dirinya sendiri.
Respons imun yang keliru dapat menyebabkan peradangan pada jaringan tubuh. Coba bayangkan ada seorang satpam, namun satpam tersebut justru mengira karyawan gedung sebagai pencuri.
Kemudian, apakah autoimun selalu menyerang bagian tubuh yang sama?
Tidak, pada beberapa penyakit autoimun, pada diabetes tipe 1, menyerang sel beta pankreas. Ada juga yang menyerang banyak organ sekaligus.
Lalu contoh penyakit autoimun apa saja dok?
Ada lupus menyerang organ seperti kulit, sendi, ginjal, dan bahkan jantung. Diabetes tipe 1 menyerang pankreas, ada kelenjar tiroid, dan masih banyak lagi.
Apa yang harus kita lakukan?
Kita dapat menerapkan gaya hidup sehat, jaga pola makan sehat dan seimbang, perbanyak konsumsi buah dan sayur, cukup aktivitas fisik, dan hindari stres.
Sekarang saya jadi lebih paham tentang autoimun. Terima kasih banyak dok.
Bagus. Yang penting, jangan mendiagnosis sendiri dan konsultasikan dengan tenaga medis.
Pemberian vaksin. Jarum suntik memasukkan cairan vaksin ke dalam jaringan kulit, menyebarkan partikel antigen di dalamnya. Respons imun awal, sel dendritik mendekati dan menangkap antigen tersebut. Lalu membawanya menuju kelenjar getah bening. Aktivasi sel imun di kelenjar getah bening. Sel dendritik mengaktifkan sel T helper, yang kemudian membantu mengaktifasi sel B. Produksi antibodi dan memori. Sel plasma yang terbentuk menghasilkan antibodi untuk menetralkan virus. Sementara sel memori terbentuk untuk mengingat ancaman tersebut. Infeksi kembali dan pertahanan. Saat virus kembali masuk di kemudian hari, sel memori langsung aktif memproduksi antibodi masif untuk menyerang dan menghancurkan virus. Perlindungan jangka panjang, sel memori tetap bersiaga di dalam tubuh, memberikan perisai perlindungan yang bertahan selama bertahun-tahun, dan dapat diperkuat kembali sewaktu-waktu menggunakan suntikan booster.
Halo guys, hari ini temenin keseharian aku yuk. Kamu siapa? Novi, kamu lagi nge-vlog ya? Iya. By the way, kamu kenal dia gak sih? Siapa?
Aku kira semua kebiasaan ini cuman berdampak pada diriku. Ternyata ada yang ikut kewalahan.
Sebenarnya kamu siapa?
Aku sistem imun kamu.
Hah? Ternyata dia selama ini sistem imun yang bekerja tanpa aku sadari.
Jadi, selama ini kamu ikutin aku?
Iya,
Tapi, kenapa kamu kelihatan capek?
karena setiap pilihan kamu itu beban buat aku juga.
Kalau dia selalu menjaga aku, sekarang giliranku menjaga dia.
Barusan, kita sudah melihat bahwa setiap pilihan yang kita lakukan dapat mempengaruhi bodyguard di dalam tubuh kita, yaitu sistem imun. Sistem imun adalah pertahanan alami yang bekerja tanpa henti untuk melindungi tubuh dari berbagai kuman dan penyakit. Karena itu, kebiasaan yang sederhana seperti tidur yang cukup, makanan yang bergizi, olahraga yang rutin, minum air yang cukup, dan mengelola stres juga sangat penting agar sistem imun dapat bekerja dengan optimal. Jadi, jangan tunggu sakit untuk mulai peduli. Yuk mulai jaga sistem imun kita dari
Di tengah hiruk pikuk kota, polusi udara, asap rokok, dan makanan instan, tanpa kita sadari, mengintai kesehatan keluarga setiap hari. Secara imunologi, polusi dan gaya hidup tidak sehat memicu lonjakan reactive oxygen species atau ROS yang merusak sel-sel imun vital seperti makrofag dan sel T.
Tapi, keluarga kami memilih antitesis, dimulai dari piring makan, mengonsumsi makanan sehat, kaya nutrisi, dan antioksidan bersama-sama. Diimbangi dengan rutinitas olahraga dan jalan kaki bersama di pagi hari untuk memperlancar sirkulasi darah dan menjaga vitalitas tubuh. Namun di usia 50-an, olahraga dan gaya hidup sehat saja terkadang belum cukup menetralkan sisa RS karena kemampuan alami tubuh memproduksi antioksidan mulai menurun. Disinilah pentingnya antioksidan harian, mulai dari konsumsi vitamin C, buah kaya penangkal radikal bebas hingga melengkapinya dengan terapi inhalasi gas hidrogen secukupnya. Sinergi nutrisi rebusan, olahraga teratur, dan asupan aktif membalikan efek buruk polusi dan makanan tidak sehat, memperlancar sirkulasi, serta memulihkan stamina keluarga.
Penggabungan seluruh antitesis ini, nutrisi, sirkulasi darah dari olahraga, serta kerja sinergis vitamin C dan molasilnya, sel T dan makrofag terlindungi secara optimal.
Menciptakan lingkungan keluarga yang sehat dan bebas penyakit kini jadi nyata. Guarding the Guardian, persembahan dari Yeni and Farms.
Deadline besok, tugas belum selesai, akhirnya begadang lagi. Tapi tubuhmu merasakan dampaknya. Saat tidur, sel imun diproduksi. Kurang tidur buat kita rentan sakit. Saat tidur, tubuh memproduksi berbagai molekul dan sel imun yang membantu melawan infeksi. Ketika waktu tidur berkurang, respons imun juga dapat menurun. Setiap hari, jutaan bakteri dan virus menyerang. Namun, sistem imun kita siap melindungi. Dari benteng pertahanan hingga pasukan khusus, semuanya bekerja sama menjaga tubuh tetap sehat. Agar imun bekerja optimal, tidur 7-9 jam, makan makanan bergizi, rutin berolahraga, kelola stres dengan baik, cukupi kebutuhan cairan. Tidak ada makanan atau suplemen ajaib yang bisa langsung membuat imun kuat. Kuncinya adalah kebiasaan sehat yang dilakukan secara konsisten. Jaga gaya hidupmu dan biarkan sistem imun melakukan tugasnya. Kebiasaan sehat akan meningkatkan imunmu.
Hai Sobat Imun. Tahu nggak sih, sistem imun itu harus berada dalam keadaan homeostasis. Kalau terlalu turun, jadi imunodefisiensi.
Kalau terlalu tinggi, jadi autoimun.
Tanpa kita sadari, justru kegiatan sehari kita lah yang menjaga kondisi homeostatis itu.
Seperti tidur yang cukup. Saat tidur, tubuh memperbaiki dan mempersiapkan sistem imun untuk menghadapi hari berikutnya. Makan makanan sehat dan bergizi. Makanan bergizi memberi nutrisi yang dibutuhkan sel imun agar dapat bekerja dengan optimal. Olahraga jangan di-skip ya. Olahraga rutin membantu menjaga sistem imun tetap aktif dan tubuh tetap bugar. Stay hydrated. Jangan lupa banyak minum air putih. Hidrasi yang cukup membantu menjaga sirkulasi darah dan cairan limfa sehingga patroli sel imun ke seluruh tubuh lebih efektif. Biar anti stres, healing jangan lupa. Lakuin hobi yang kalian suka. Misalnya main game, baca buku, dan hangout. Saat stres, kortisol yang disekresikan lewat HPA axis meningkat, sitokin proinflamasi juga meningkat. Makanya harus tetap happy ya guys ya.
Tuh, mudah banget kan?
Yuk usahain hal-hal tersebut untuk jaga dan mempertahankan sistem imun kita.
Healthy habits, healthy immunity.
Bagaimana sistem imun bekerja? Ketika mikroorganisme patogen masuk ke dalam tubuh, tubuh tidak langsung mengandalkan satu jenis pertahanan saja. Sistem imun bekerja secara bertahap dan terkoordinasi. Mari kita ikuti bagaimana tubuh menghadapi infeksi tersebut. Patogen mulai masuk ke dalam tubuh. Bayangkan terdapat suatu mikroorganisme patogen yang mencoba masuk ke dalam tubuh. Begitu patogen mencapai permukaan tubuh, pertahanan pertama yang dihadapinya adalah barier fisik dan kimia. Ketika patogen berhasil melewati barier fisik dan kimia tubuh, tubuh akan mengaktifkan sistem imun inert. Imunitas inert merupakan pertahanan pertama yang bekerja dengan cepat dan tidak spesifik terhadap jenis patogen tertentu. Jika patogen masih bertahan, sistem imun inert akan membantu mengaktifkan imunitas adaptif melalui presentasi antigen oleh sel dendritik. Imunitas adaptif bekerja lebih spesifik terhadap antigen tertentu. Setelah infeksi teratasi, sebagian sel T dan B akan menjadi sel memori. Sel inilah yang memungkinkan tubuh memberikan respons yang lebih cepat dan kuat ketika menghadapi patogen yang sama di kemudian hari. Sekarang, kalian sudah tahu kan bagaimana sistem imun bekerja. Yuk jaga kesehatan dengan rajin olahraga dan makan makanan bergizi.
Kasus campak di Indonesia meningkat. Pesat tahun 2026 yuk, kita lihat animasi edukasi untuk tahu lebih lanjut. Anak kecil gampang tertular campak karena sistem imun masih berkembang. Bayangkan hari pertama, sekolah dengan teman-teman baru yang banyak, membawa virus ke sekolah, lingkungan baru, paparan baru. Disinilah sistem imun bekerja. Tubuh mempunyai database imun.
terlalu tinggi jadi autoimun.
Kita sadari, justru kegiatan sehari kita lah yang menjaga kondisi homeostatis itu.
Seperti tidur yang cukup. Saat tidur, tubuh memperbaiki dan mempersiapkan sistem imun untuk menghadapi hari berikutnya. Makan makanan sehat dan bergizi. Makanan bergizi memberi nutrisi yang dibutuhkan sel imun agar dapat bekerja dengan optimal. Olahraga jangan di-skip ya. Olahraga rutin membantu menjaga sistem imun tetap aktif dan tubuh tetap bugar. Stay hydrated. Jangan lupa banyak minum air putih. Hidrasi yang cukup membantu menjaga sirkulasi darah dan cairan limfa sehingga patroli sel imun ke seluruh tubuh lebih efektif. Biar anti stres, healing jangan lupa. Lakuin hobi yang kalian suka. Misalnya main game, baca buku, dan hangout. Saat stres, kortisol yang disekresikan lewat HPA axis meningkat, sitokin proinflamasi juga meningkat. Makanya harus tetap happy ya guys ya.
Tuh, mudah banget kan?
Yuk usahain hal-hal tersebut untuk jaga dan mempertahankan sistem imun kita.
Healthy habits, healthy immunity.
Bagaimana sistem imun bekerja? Ketika mikroorganisme patogen masuk ke dalam tubuh, tubuh tidak langsung mengandalkan satu jenis.
Manggil badai, pasti tubuh kamu
Halo, saya Anas Fadliwijaya.
Nama saya Rian Anggiaris.
Halo Sobat Imun, perkenalkan nama saya Ina Sasna Aziza.
Hal imun yang pertama ini membahas tentang imunohistokimia, di mana ini sangat relevan dengan bidang yang saya geluti sekarang sebagai staf pengajar atau dosen yang mengampu mata kuliah.
Check. Okay, good afternoon everyone and welcome back from the lunch break. I hope everyone has already enjoyed your lunch boxes. I certainly have already also enjoyed my lunch box. It was quite tasty, the meat and the rice. Now, without further ado, it is now time for our oral presentation sessions. We already have our panel of judges ready and also our moderator today. So without further ado, without wasting any more time, I will hand off the session for the oral presentations to of our moderator, Dr. Ayu. Please, the stage is now yours.
Okay, thank you very much, Mas Nabil, as our MC. So we are going to moving on to the oral presentation session. In this session and in this room, we will have 8 presenters, oral presenters, and each presenters will present around 7 minutes, and then followed by discussion around 3 minutes. And we have a panel of judges. We have Professor Fiskasari Kalanjati, our coordinator for Master Program in Immunology, and we have Professor Teresia Indah Budi, Head of Department in Faculty of Dentistry, as our judges today. So without any further ado, we will start from our first oral presenter, Mr. Ali Zainal Abidin, who will be presenting online in Zoom platform. Are you ready, Mr. Ali Zainal Abidin? For online presenters, you can share the screen by yourselves. Can you hear us, Mr. Ali Zainal Abidin? Okay. Mr. Ali Zainal Abidin, apakah bisa di-end dulu share screen-nya dan di-restart kembali, dimulai kembali untuk share screen-nya? Baik, sambil menunggu Mr. Ali Zainal Abidin, kami akan pindah ke presenter selanjutnya, Bapak Muhammad Akilah Herdiansyah. Apakah hadir luring atau di Zoom? Oke, Bapak Ali Zainal Abidin, slide-nya sudah terlihat, waktunya 7 menit maksimal untuk presentasi Anda. Apabila melebihi kami mohon izin untuk langsung di-cut seperti itu dan lanjut ke sesi tanya jawab dengan judges. Silahkan dimulai, Bapak. Oke, another technical difficulties. Akan kami tunggu 1 menit seperti itu, nggih. Sekiranya Bapak Ali Zainal Abidin masih belum bisa connect dalam 1 menit, kita akan pindah ke presenter selanjutnya. Baik, Bapak Ali Zainal Abidin, suaranya belum terdengar dengan jelas.
Belum terdengar ya?
Oke. Apakah sudah siap? Boleh kami diberi tanda aba-aba, Bapak?
Suara saya sudah terdengar belum ini? Suara saya sudah terdengar belum?
Baik.
Halo?
Baik. Kami sudah bisa mendengar suara Bapak, bisa dimulai.
Oke.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, all the judges, all of the committee and colleges. My name is Ali Zainal Abidin, and I'm here to send my team to present our research. First of all, as we know, breast cancer remains a major public health challenge in Indonesia with a substantial burden of incidence and mortality, particularly as many patients continue to present with advanced disease. HR positive HER2 breast cancer expresses estrogen and or progesterone receptor without HER2 overexpression and is primarily treated with endocrine therapy. Clinically, two major challenges remain. First is bone is a preferential metastatic site for HR positive disease and is commonly involved in advanced breast cancer. And the second one is endocrine resistance may develop through alteration in estrogen receptor signaling and activation of alternative survival pathways, ultimately limiting to the long-term effectiveness of endocrine therapy. Our previous findings from microRNA profiling using nanostring identified multiple microRNAs associated with bone metastasis in HR positive HER2 breast cancer, along with several target genes and significantly enriched signaling pathways. Among this pathway, FOXO and TGF-beta were particularly important because of their established roles in endocrine resistance. This finding suggests a potential molecular intersection between bone metastasis progression and endocrine resistance. Based on this biological rationale, we focus on plasma microRNAs, which are stable and minimally invasive biomarker candidates that may reflect this molecular alteration. Therefore, this study had two objectives. Ultimately, this study provides preliminary evidence toward minimally invasive molecular profiling and future personalized monitoring. And this study was an analytical observational study using a cross-sectional design, and the study received ethical approval from the Ethics Commission of FKKMK Universitas Gadjah Mada. For the research workflow, we collected 5 milliliters of peripheral blood from 12 participants for patients with bone metastasis, for without bone metastasis, and for healthy controls. And plasma was isolated, followed by total RNA extraction and microRNA profiling. Finally, in silico analysis was performed to identify differently expressed microRNAs, validated target genes, and signaling pathway, particularly FOXO and TGF-beta. Now moving to our results, approximately 798 endogenous microRNAs were detected across the three study groups, showing distinct plasma circulating microRNA expression patterns. When HR positive HER2 breast cancer patients with bone metastasis were compared with those without bone metastasis, 40 microRNAs were significantly dysregulated, consisting of 25 upregulated and 15 downregulated microRNAs. And this table highlights the top 10 upregulated and top 10 downregulated microRNAs identified in the metastatic group. The upregulated microRNAs shown were then further analyzed to identify their experimentally validated target genes and to explore the molecular pathway potentially involved in bone metastasis and endocrine resistance. Next, this heat map illustrates the expression pattern of the selected dysregulated microRNAs in plasma from patients with bone metastasis HR positive HER2 breast cancer compared with those without bone metastasis. And next, we analyze experimentally validated target genes using the multi-minor package in R, and the analysis focused on the top 10 upregulated microRNA identified in patients with bone metastasis in HR positive HER2 breast cancer group to prioritize the most relevant candidate biomarker. From this analysis, we found 14 validated target genes were identified, and several microRNA converge on the same target genes, suggesting coordinated regulation of interconnected molecular networks. These validated target genes were then further analyzed to identify enriched signaling pathway potentially linking bone metastatic progression with endocrine resistance. Next, pathway enrichment analysis was performed to identify signaling pathway potentially linking bone metastasis with endocrine resistant, and the results show that FOXO and TGF-beta emerge a prominent enriched pathway with relatively high gene representation and approximately 5% and more than 2.5% of input genes mapped on to this pathway respectively. This finding support FOXO and TGF-beta signaling as a potential pathway connecting bone metastatic progression with endocrine resistance. Importantly, FOXO and TGF-beta signaling emerge among the pathway associated with bone metastatic related microRNAs signature. FOXO signaling is closely linked to endocrine responsiveness, as reduced FOXO3A activity has been associated with tamoxifen resistance and immune cell regulation. Meanwhile, TGF-beta signaling associated with promotes tumor bone interaction and immune oscillating progression, while disruption of TGF-beta SMAD axis has also been linked to endocrine resistance. However, as immune function was not directly assessed, this remains a biologically plausible hypothesis generating model rather than evidence causality. 9 circulating microRNAs emerge as candidate biomarker associated with FOXO and TGF-beta related signaling networks. Among them, MIR15B5P has been linked to tamoxifen resistance through the FOXO1 axis, while MIR142 3P may influence endocrine responsiveness through ESR1 regulation. In addition, MIR19A and 19B 3P showed close association with above pathways, supporting the potential molecular link between endocrine resistance and bone metastatic progression. In conclusion, a 9 circulating microRNA emerge as a candidate biomarker linking bone metastatic HR positive or negative breast cancer with endocrine resistant related FOXO and TGF-beta signaling. These preliminary findings support their potential for minimally invasive molecular profiling, but however, a larger perspective cohort, longitudinal endocrine response assessment, and functional validation are required before clinical implementation. The following slide presents the key reference. Thank you for your attention. I look forward to your question and an insightful discussion. Thank you.
Okay, thank you very much, Mr. Ali Zainal Abidin. Can we give a round of applause? So we are going to move on to our judges. Prof. Fiska, Prof. Tere, if there is any question.
Okay, thank you, Dr. Ayu. Dr. Zainal, ya? Zainal, ya? Okay. I have just one question. What is your reason for stating that TGF-beta as a signaling molecule bone metastasis in breast cancer? As we know that the TGF-beta is anti-inflammatory cytokine. So what's the relation about the anti-inflammatory cytokine and breast cancer? Okay.
Thank you, Prof, for the question. How could the TGF-beta connect bone metastasis? Because TGF-beta provides a biologically plausible intersection between the process and the bone microenvironment. TGF-beta signaling can contribute to osteolytic tumor bone interaction, while alteration in the TGF-bSMAD axis has also been a link to endocrine resistance in HR positive or HER2 negative breast cancer. Therefore, it may represent a shared pathway, although our study does not establish causality. Thank you, Professor.
Okay, thank you.
Thank you very much. Mr. Ali, Ali Zainal Abidin, you are the first author, right?
Yes.
I cannot see him actually. Can I see him? The Zoom? So my question is, in your abstract, you do not mention about the N sample from the plasma, but then in the result, you mention a total of 46 circulating microRNAs. that showed significantly altered expression. Yeah, can you explain to us the details on your materials and methods more? And this is a very in-depth study. Have you and or your other team published this particular study, same variables or similar variables, to other platform like a journal before in this conference? Yeah, thank you.
Professor, for the question. Before we never published this research result before, because we still analyze more the data, and the reason why we use the small size in this research, because the sample size we use 12 was determined based on capacity of the nanostring encounter panel, consisting for patient with bone metastasis, for without bone metastasis, and for healthy controls. Despite the relatively small sample size, the platform enables simultaneous profiling of more than 800 microRNAs, with approximately in this result, in the research results, we got around 798 endogenous microRNA detected in our study.
Okay, thank you very much, Mr. Ali Zainal Abidin. So we are going to move on to our second presenters who will be presenting through Zoom platform. Miss Amalia Octaviana Hartini already present. Sudah ada di Zoom nggih? Atas nama Amalia Octaviana Hartini.
Iya.
Oke, baik. Kami persilahkan untuk share screen. Waktunya 7 menit ya, nanti kalau sudah lewat 7 menit akan langsung kami cut seperti itu, mohon izin. Kami persilahkan. Apakah sudah siap? Are you ready? Bisa dicoba bicara? Kami belum bisa mendengar suaranya.
Sudah siap.
Okay, we can hear you now. Please, you can start your presentation.
Jangan continue to share.
Assalamualaikum warahmatullahi wabarakatuh. My name is Amalia Octaviana Hartini. I am student of Muhammadiyah University. Today I'm going to present the abstract our manuscript about effect of murotal exposure frequency on eosinophil response in Rattus norvegicus exposed to mosquito coil smoke. Allergic reaction are hypersensitivity response that may involve increased eosinophil counts following allergen exposure. Mosquito coil smoke can act as an environmental stressor that disrupts immune function and induces allergic response. Quranic recitation, known as murotal, may promote relaxation, reduce stress, regulate nervous system activity, and modulate immune response. This study aims to investigate the effect of different murotal exposure frequency on eosinophil percentage as an indicator of cellular immune response. Research design through experimental with post-test only control group design. Treatment methods. 24 Rattus norvegicus rats were divided into 6 groups. 1 control group, and 5 treatment groups. All groups were exposed to smoke from mosquito coil as an environmental stressor. The treatment groups receiving murotal exposure 1 to 5 times daily for 40 days, with each exposure lasting 10 minutes. Eosinophil percentage in whole blood samples were measured using a hematology analyzer. The Mann-Whitney U test was used to assess difference between the control and treatment groups after assessment of normality using the Shapiro-Wilk test and homogeneity using Levin test with a significance level of 0.05. The result of the research is eosinophil percent range from 0.0% to 0.2% in the control group and from 0.0% to 2.0% in the treatment group. The highest percentage, 2.0%, was observed in the group receiving murotal twice daily. The Mann-Whitney U test saw no statistically significant difference between the control and treatment groups. The highest eosinophil percentage observed in the group receiving murotal twice daily may indicate a serological response to exposure to mosquito coil smoke. This finding is supported by Ivani and Istiqomah, who state the allergen exposure can stimulate IgA production and activate eosinophil, thereby leading to an increase in blood eosinophil counts. Variation among treatment groups may reflect individual biological response. However, murotal exposure frequency didn't significantly alter eosinophil percentage. This finding suggests the murotal exposure alone may be insufficient to modulate eosinophil response following mosquito coil smoke exposure under the experimental condition applied in the study. Further studies using large sample, longer intervention periods, and additional immune biomarker are required to clarify its potential immunomodulatory effects and relevance to health. That's all from me. I hope my presentation was clear. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Give a round of applause to Miss Amalia Octaviana Hartini. We are moving on to our judges. Prof. Tere, kami persilahkan.
Thank you, Dr. Ayu. Amalia. What's your name? Amalia. Amalia. Okay.
Yes.
Amalia, what's the method is used to measure eosinophil?
Yes.
What's the method for measuring the eosinophil?
Whole blood with hematology analyzer.
Oh, blood analyzer. Okay. And is there the connection, the eosinophil with allergy? Because you said about allergy or IgA.
Yes.
Please explain about this. Explain how the eosinophil have the connection with the allergy or related with allergic.
You may answer, or would you like Prof. Tere to reply? Boleh dalam bahasa Indonesia, Prof. Tere, monggo.
Ya, mungkin nanti dijawab juga dalam bahasa Indonesia boleh. Tadi kamu mengatakan tentang alergi ya, atau IgE ya, yang saya hanya melihat abstrak soalnya ya. Coba jelaskan hubungan antara eosinofil itu dengan alergi atau IgE.
Izin Prof. Tere, saya izin menjawab. Jadi eosinofil ini dipilih karena eosinofil berperan pada reaksi alergi dan hipersensitivitas, di mana paparan alergen atau bahan iritan ini seperti salah satunya obat anti nyamuk bakar dapat mempengaruhi respon eosinofil.
Oke, thank you.
Monggo Prof. Fiska. Oke, terima kasih Amalia ya. Pertanyaan saya, please explain to us the relation between the obat nyamuk bakar with the allergic reaction and Murrotal effect in no more than 10 sentences, I think. Thank you.
Because this is basic research, I just want to see effect of carbon dioxide on immune system, especially eosinophil in blood. Therefore, we are use mosquito coil smoke.
Is there any other comment, Prof. Fiska? That's it. That's all. Itu aja jawabannya, dirasa sudah cukup.
Iya, Ibu.
Belum diterangkan tentang korelasinya dengan Murrotal. To be specific, the kind of Murrotal, the surah, the length, the murojaah, you know, that the way the reader of the Al-Quran that you play in your study, they have this specific style, right? You haven't mentioned the detail. Anda harus sebutkan detailnya. Bisa diterangkan?
Izin menjawab, Ibu. Jadi Murrotal itu kan diketahui mampu memberikan efek relaksasi. Nah, jadi dari relaksasi tersebut digunakan sebagai terapi untuk menurunkan stres yang akibat dari paparan CO2 tersebut untuk perubahan kadar eosinofil dalam darah.
Okay, thank you very much, Miss Amalia. I think that's all for your session. We are going to move on to the next presenter. So our third presenter will be Mr. Muhammad Akila Herdiansyah, and he will be presenting offline.
Sorry, Miss.
Oh.
I'm changing from offline to online.
Oh, okay.
Sorry.
Okay, it's all right. We can hear you, so you can share your screen and start your presentation. 7 minutes for presenting, and we will cut you or stop you if your time is up. The time is yours, Mr.
Okay.
Muhammad Akila.
Okay, please. Let me share screen first. Have you seen my slide?
Okay, we can see it clearly.
Okay. It's very clear, right? Okay. I will present my research. Thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon to the honorable professor and also the committee and also all of the presenter and also the participant of the World Immunology Day International Conference today. In here, I'm Muhammad Akila Herdiansyah from Faculty of Science and Technology from S3. I will present about my research about the host-directed immunomodulatory potential of Boston-Berger rotunda against pan-serotype dengue virus by the integrating in vitro evidence with the redox-associated computational analysis. This is the introduction of my research. There, as we know, from viral replication to host redox homeostasis, the Boston-Berger rotunda as a host-directed immunomodulatory candidate against pan-serotype dengue virus. First of all, like we know, the dengue infection, it has the 4 serotypes from dengue 1 until dengue 4. The dengue 1 until dengue 4 infection with different serotype can increase the risk of the severe dengue or ADE. Currently, the dengue treatment is mainly supportive, is supportive for the supportive purpose. The second is the host response regulation by the dengue. It will increase the loss of, or what we can call reactive oxygen species by the mitochondrial dysfunction, the pro-inflammatory signaling increasing by the nuclear factor kappa B, tumor necrosis factor alpha, and also the interleukin 6, and also the immune dysregulation and the endothelial dysfunction or the vascular permeability. It will consequence by the organ dysfunction, vascular leak, and also the severe disease. Therefore, the therapeutic gap of this research from until now, the conventional antiviral strategy is only target the virus, the dengue virus also. However, how we also target the host response? That's the question. Therefore, this research will have the host-directed therapy to restore the redox homeostasis without compromising the antiviral activity. Indonesia is the largest country with a lot of the herbal plant that can be used as the herbal potential. One of the herbal is the Boesenbergia rotunda, or Indonesia what we can call tembu kunci. The tembu kunci or fingerwood, it has a lot of flavonoids, phenolic compound, and other bioactive compounds that can be used as the drug. This compound from the previous research can be used as the antioxidant potential and also the antiviral activity. But therefore, the dual action strategy by the viral control by reducing the dengue infection from the force serotype of the dengue and also the host protection by increasing the redox homeostasis is needed. The research gap of this research by the immunomodulatory potential of Boesenbergia rotunda against pan serotype dengue remains largely unexplored. The question is, can Boesenbergia rotunda simultaneously suppress the dengue infection and modulate the host redox homeostasis? This study explored this throughout integrated in vitro evidence by the cell and redox-associated computational analysis. Our vision is to target the virus, restore the host, assess the better outcomes in dengue disease. This is the methods of this research. By the integrated experimental and also computational approach, we use the experimental using the ferroetic cells. For the plant materials, first of all, we use the extraction with the maceration ethanol with the ethanol 96% to obtain the Boesenbergia rotunda ethanolic extract. And also, we conduct the metabolic profiling, the LC-RMS with the USPLC, X-exative or the trap mass, XRMS to obtain the metabolite identification or the diversity of the metabolite with the MS-CoV and Chem-Spider. And after that, from the compound, we use the biological activity prediction by the pass-on-length or way-to drug to predict the key biological activities, for example, like the antioxidant and also the antiviral and immunomodulatory. And after that, from the biological activity prediction, we use three of the compound as the potential of the antioxidant and antiviral to be in the molecular docking with the envelope proteins and also superoxide dismutase or SOD with the Pyrex to obtain the binding affinity with the unit of kilocalorie per mole and interaction analysis. The molecular dynamics to know about the complex stability validation is needed for the molecular docking validation with the RMSF or root mean square fluctuation to know about the fluctuation of the ester by the biophysical and biochemical, and also the Ramachandran plot. And after that, for the in vitro, we use the in vitro anti-dengue assay with the ferroetic cells as cells and infection with dengue1 until 4. And for the dengue replication, we use the viral tox glue assay to obtain the EC5T. And for the cytotoxicity, we use the CC5T with the MTT. And after that, we combining this between the CC5T and also the EC5T to obtain the selectivity index of this extract. Last is the ATME toxicity analysis. We use the in silico prediction with the PKCSM. ATME is absorption, distribution, metabolism, expression, and also the toxicity. And also the toxicity, we use the PROTOX to obtain the LD5T and toxicity class. This study integration is used to identify the Boesenbergia rotunda as a potential natural immunomodulator and anti-dengue agent for the modulation of redox homeostasis in the cell. Okay, next. This is the result of this research. Boesenbergia revealed the diverse bioactive metabolite profile. There are 38 metabolites and also the major chemical classes is flavonoid, coumarin, phenolic compounds, and major metabolites. We know that phenosembrin as a biomarker, imperatorin, soporanon, for the antiviral and antioxidant activity with the highest activity. And after that, this is the schematic of this research. The superannon was prioritized as the lead candidate for mechanistic analysis with the envelope protein across multiserotype dengue virus and superoxidant dismutase protein immunomodulation in the cell. And this is the molecular docking experiment result. The binding affinity with the highest is the DNV3 with the minus 7.7 kilocalorie per mole, and also the SOD minus 7.2 kilocalorie per mole. And all of the complex mostly dominated by van der Waals force, hydrophobic interaction, and hydrogen bond. One candidate to biological direction. It is the molecular dynamics to know about the validation. The lower residue fluctuation in all complex is below the 2 Armstrong and stable binding pocket environment with the persistent ligand protein interaction. This is the in vitro results. As we know from the in vitro research, there are DNV3 and DNV4 with the selectivity index is the highest than the other and highest than the other of the antiviral commercially. And this is the discussion, the broad spectrum of the antiviral potential. Beta wasn't very rotunda in bit alpha than serotype with the DNV4
Mr. Muhammad
is the highest.
Akila, we are very sorry that your time is up. So we are going to move on to the discussion session with the judges. Professor Teresia, monggo, Prof.
Thank you. Very interesting your presentation. You have many methods for measuring the activity of the molecular, and should all of them be done, or should I choose based on my needs? What's your opinion?
Okay. Thank you, Professor Teresia, for the interesting question. This is the method. It's the one of the combination of them of all of the. This is the integration, Prof. By, for example, like the LC-RMS to know about the metabolite profile. At first, we identify our goal is to obtain the potential metabolites, Prof. For the anti-dengue, and after that. We also combining with the molecular docking at first to prediction first what kinds of compound that will binding into the superoxidant dismutase and also the envelope proteins of the DNP, and after that we confirming this prediction by the in vitro Prof. So this integration will provide one single data, big data that will provide that our extract is as the new anti-dengue candidate like that, Prof. So this is the
thank you.
Combination data.
Prof. Fiska, please.
Thank you, Prof.
Thank you. Siapa tadi ya namanya ya?
Muhammad Akila.
Oh iya, Ding. Muhammad Akila ya. So sorry. Iya. Sudah pernah ditampilkan atau diterbitkan di platform lain hasil studi Anda ini?
Oh, sorry, Prof. This is our preliminary study, so we didn't publish it yet.
You haven't.
Yeah. This is the first one that the immunology is the first that I published this paper.
Good. Would you mind explaining in a simple way the difference in the molecular level of DNV3 and DNV4 in relation to each of these do you call it the variable with your phytopharmaca? Thank you.
Thank you, Prof. Fiska. It's very interesting question. That we're also finding something new in this research by as we know that it can good at binding into the DNV3 and also DNV4, Prof. And for the molecular level, I will answer with the computational with the molecular docking and also molecular dynamics, Prof. From we know at the molecular levels, the DNV3 and DNV4 will binding into the cell first, and after that it will fast for fusion, And in this research, we found that the exposure of the Boeschen-Berger rotunda or temu kunci in Indonesia, it will inhibit the binding between the DENV3 and also DENV4. That's why in the molecular docking, we found that the binding is very high on the DENV3 and also DENV4, Prof. And also we found something interesting from this research that will I publish in this conference. We found that this Boeschen-Berger rotunda can have the highest of the potential as the anti-dengue, the new anti-dengue, Prof. Because it has the selectivity index by the CC50, the cytotoxicity is very low, Prof. It's very good. It's not toxic, and also the CC50 can be inhibit the replication of the dengue, like that, Prof. So first of all, by the inhibit the DENV fusion to the cell, it will inhibit the NS2B NS3 protein, Prof. And NS2B NS3 is the protein of the dengue that can be used for the dengue to replicate itself. Yeah, that's the molecular insight, Prof.
Okay. Thank you very much, Mr. Muhammad Akila Herdiansyah. So that would conclude your session, and we are going to move on to the next presenter, Mr. Yeni Purnama, who will be also presenting online. Pak Yeni, apakah sudah siap? Mr. Yeni Purnama. Okay, we can see your screen. So you have 7 minutes for presentation. If the time is up, we are going to stop your presentation. Monggo, Dr. Yeni. Dr. Yeni.
Assalamualaikum warahmatullahi wabarakatuh. Mungkin ini agak susah ya. Assalamualaikum warahmatullahi wabarakatuh. Apakah suara terdengar?
Terdengar jelas.
Oke. Ini sepertinya presentasi saya kok kurang bagus ya.
Apakah bisa diulang share screen-nya dulu?
Oke, siap. Excuse me. Apakah sudah terlihat, Ibu? Mohon maaf.
Sudah terlihat tapi belum full screen.
Kami sudah di full screen tapi belum ya, sudah gimana?
Apakah perlu dibantu? Bisa dibantu share dari sini?
Boleh Bapak ini. Boleh. Mohon maaf ini mungkin ada gangguannya. Laptop saya ada masalah mungkin.
Ya, di end share dulu. Silakan Bapak untuk memulai.
Okay. Thank you. This is Professor, reviewer, moderator, and ladies and gentlemen. Allow me this morning to present case report of very premature infant with pseudomonas ergosepsis, distress syndrome, and bronchoalveolar dysplasia, who received standard therapy and additional secretum therapy. The patient had been extubed 3 times due to failure to be weaned from the ventilator. This patient was born in RSPAD Gatot Subroto. In infant was premature and very low birth weight, and due to several HFO and the parent had requested to do not resuscitation. They believed was no hope. In important the hierarchy experience spot or most intensive care. HFO, conventional ventilation, and free flow oxygen. The patient is currently on HFN with 21% oxygen, the same as room oxygen. Next, baby is born 3 July with RDS and 20 and 29 weeks exertion and very low blood weight, and therapy in HFO therapy, parenteral nutrisi, and first antibiotic is surfactant and caffeine citrate. On 3 July, this is sebelumnya, and did 10 July 2000 growth the Pseudomonas aeruginosa, additional is neonatal sepsis to Pseudomonas aeruginosa. The patient is ready to transition from to conventional ventilator, and 3 attempt extubation have been made due to clinical improve, but they more unsuccessful. The antibiotic switch to Lini 1 to ceftazidime, Lini 2. At the time I was treating on the 21 in July with body weight 1,445 and additional diagnosis, blah blah blah, and other therapy include parenteral nutrition and caffeine citrate and continue substitution treatment positive that regimen and adjuvant therapy secretum, and interval 12, 24, and 28. Until June, the 1,000 centimeter, the patient is successful extubate for conventional ventilator to transfer to NFVP. The baby was successful. We need from NFVP to CPAP and is currently IFN with flow at 2 liter minutes and room air 21 oxygen. Oral milk intake target caffeine citrate and body weight reaches to 1,875 gram. Allow me show a brief video selanjutnya, tapi mohon maaf the video tidak bisa. Increase ventilator, AFN and CPAP and AFN. Okay, next. Why is this important? Babies is very premature, is not yet infected. There are interstate host responsive rapidly between excessive inflammation and immune dysfunction. Immature barrier, immature immune needs to exposure and drug. Next. Clinical case summary, primary diagnosis, neonatal sepsis and positive blood culture with Pseudomonas aeruginosa, respiration complication, HMD, grade 3 and rapid disease syndrome, upload mortality, ventilator dependent BPD and atelectasis. Standard terapi, surfactant, ventilator antibiotik, caffeine citrate, and adjuvant terapi secretum. Next. The premature open the door on patient. Review in the not yet mature pathogenes in the gut, kemudian bloodstream invasion and immature organ. Next. Reaction to Pseudomonas activated the innate axis plus Pseudomonas aeruginosa, the SPS fluency signal, the induce the TLR4 and FKB, and induce cytokine and leak endothelium. Next. Reaction 3, the lung of primary target, RDS induce ventilator and induce lung inflammation. Next. Biological treatment from inflammation to immune dysfunction in babies is lymphocyte down, HLA, DR, amyloid down, and dendritic down. CRP not to interrupt because excessive immunoglobulin and relative analysis. Okay, lanjut. Jadi pneumonia BPD, one axis the premature lung injury. It is a premature radiation MD infection, ventilator oxygen BPD. Randomly control in preterm infant with signaled the safety visible but have not definitively proven induced except for BPD. Next. Standard terapi remain foundation, antibiotik, kemudian scafen citrate, diet regimen, and etc. Next. Secretum was the mean non-viral exosome, potensial this protein, paracrine, signaling, kemudian cytokine, chemokine, kemudian ini ya, lihat ini. Next. Menuliskan eksternal 3, secretum effect syndromitis, secretum soluble factor enter the tissue, cargo protein and miRNA, macrophage change response, dan interleukin 27, dan interleukin 6, 10 alpha, and animal is very good. Okay. Balance evidence, BPD is good.
Mr. Yani, we are very sorry that your time is up.
Okay.
We are going to move on to the discussion session.
Dengan bahasa Indonesia, Dok, karena ini sedang di gedung jadi agak berisik.
Baik. Profesor Teresia, monggo dalam bahasa Indonesia requestnya.
Terima kasih Dr. Ayu. Dr. Yani, ini kasus yang sulit dan menarik ya. Apakah di kasus ini sudah dilakukan ya pemberian skretom itu?
Sudah diberikan, Prof. Jadi mohon maaf, pasien ini sebetulnya sudah 3 kali dia dicoba ekstubasi dari ventilator konvensional ke ventilator yang konvensional itu tidak berhasil. Jadi pada waktu itu kami putuskan untuk memberikan skretom sebanyak 4 kali dengan jarak 12, 24, dan 48 jam. Pada 2 hari setelah pemberian sekretom, pasien ini bisa terekstubasi. Jadi bisa menggunakan alat ventilator yang konvensional. Bukan konvensional, jadi menggunakan alat ventilator yang tidak melalui ETT. Dan sampai sekarang pasien ini sudah survive, full fit, artinya sudah minum full dan tanpa infus. Dia tinggal punya PR, masih ada apne. Apne tidak bernafas, dikarenakan mungkin karena proses waktu awal kehamilannya tidak bagus. Dan pasien ini memang sudah orang tua sudah mengatakan donor situation, jadi sudah tidak sudah nyerah, artinya sudah tidak pengharapan. Tetapi pasien ini saat ini sudah full fit, sudah menggunakan namanya HFN, jadi menggunakan tekanan oksigen, tapi oksigennya oksigen ruangan, jadi sudah cukup bagus, Prof.
Ya, jadi skretom sangat bermanfaat untuk hal ini ya. Apakah ada karakteristik skretomnya, Dokter Yani?
Sebetulnya sebelum saya memberikan itu saya berkomunikasi dengan produk CCR yang di Semarang. Kami ada dua produk, satunya adalah antiinflamasi, satunya lagi adalah yang biasa. Kami disarankan memberikan yang biasa, kan di situ lebih banyak ada growth hormone, ada efek paracrine, ada efek chemokine yang mungkin barangkali semua unsur bisa bisa memenuhi maksud daripada pasien ini. Karena pasien ini sangat berat, Prof. Jadi pada waktu itu memang sebetulnya kalau kita menghadapi BPD harusnya dilakukan menggunakan ETT, tapi kami tidak berani karena ini masih baru pertama kali kami menggunakan subkutan dengan harapan supaya pengeluaran obatnya dikit-dikit, Prof. Dan ternyata ada fenomena menarik di pada saat ini, terjadi peningkatan leukosit yang sangat tinggi, tetapi kami tidak memberikan antibiotik dan itu bisa turun dengan sendirinya. Dan kulturnya negatif, Prof.
Iya. Terima kasih, Dokter Yani.
Prof. Fiska, silakan.
Baik, menurut Anda apa strength and limitation dari pemberian sekretum pada kasus ini? Terima kasih.
Mohon maaf, Prof, belum limitation maksudnya ya, limitasi.
Strength apa, limitationnya apa? Strength limitationnya apa menurut Anda sebagai mahasiswa SD imun ya? Baik.
kekuatan dari kasus ini adalah kami sudah mencoba dengan berbagai cara yang sudah maksimal karena dari awal HFO ventilator konvensional dan sudah dicoba ekstubasi 3 kali tidak berhasil, Prof. Jadi kami berpikir kami berharap dengan pemberian sekretum ini bisa mempengaruhi kondisi bayi yang dimana sekretum ini berfungsi sebagai imunomodulator sehingga diharapkan kondisi sepsis, kondisi inflamasi yang terjadi pada ini bisa tertangani. Limitnya memang menjadi masalah karena pemberian sekretum pada bayi itu memang belum pernah dilakukan, Prof. Jadi sebagian besar memang dari penelitian sebagian besar kepada hewan dan in vivo atau pada in vitro secara laboratorium. Jadi memang ini kami ketika akan memberikan juga kami mengedukasi ke keluarga, kami menyampaikan jurnal-jurnal yang jurnalnya sederhana, tentunya sederhana sehingga keluarga pasien memang berkenan diberikan sekretum ini. Jadi memang kami agak ngeri sedap, Prof. Mohon maaf, memang ini baru pertama kali dan menurut saya ini peluang bagi kami klinisi dengan harapan pemberian terapi standar ditambah dengan terapi asupan terapi ini ternyata mungkin selain sinergi. Saat ini selain sinergi, Prof. Apakah nanti itu bisa sinergi, kita nggak bisa memastikan, Prof. Terima kasih, Prof.
Okay. Thank you very much, Mr. Yeni Purnama. Can we give a round of applause? Okay. So that conclude your session. And we will be moving on to the next presenter, Mr. Farel Arif Muhammad, who will also be presenting online. Mr. Farel Arif, apakah sudah siap?
Ya, sudah.
You can share your screen and start your presentation for 7 minutes. Yeah, Mr. Farel Arif, thank you very much.
Okay, thank you. Okay, good afternoon, distinguished chairperson, respected members of the scientific committee and fellow participants. My name is Farel Arif Muhammad from the Faculty of Veterinary Medicine, Universitas Airlangga, Indonesia. It is my pleasure to present our study entitled Protective Effect of Moringa oleifera leaf extract against ethylene glycol induced spermatogenic cell necrosis. And without any further ado, let us begin with the background of this study. Next. So for the ethylene glycol, or EG, is a toxic compound that can cause systemic tissue injury following exposure. And if you remember, in 2022, the tragedy happened in Indonesia where 1,000 until million children in Indonesia were deaf because of this ethylene glycol. And that they bought from the medical store, the fever medication, and until now that would be the greatest tragedy in Indonesia that happened in Indonesia. And on the other hand, one important mechanism underlying this toxicity is the development of oxidative stress. Oxidative stress disrupts cellular redox balance and can subsequently trigger inflammatory signaling. When this process persists, it may lead to cellular injury, including apoptosis and necrosis, and ultimately impair tissue function. And importantly, oxidative stress and inflammation are closely interconnected. Therefore, EG and induced tissue injury is not only a matter of direct chemical toxicity, but may also involve inflammatory and immune-related mechanisms. And in this context, moringa is of interest because its leaves contain various bioactive compounds with antioxidant and potential immunomodulatory properties. And based on that rationale, we formulated the following objective. Next. The objective of this study was to evaluate the protective effect of moringa MLE against EG-induced spermatogenic cell necrosis in male with Ratus norvegicus. On the other hand, we wanted to determine whether administration of moringa could help preserve spermatogenic cell integrity following EG exposure. To address this objective, we conducted the following experimental study. Let's move to the next slide. A total of the 25 male Ratus norvegicus were used in study and randomly allocated into 5 experimental groups. The experimental groups consist of a control group and EG-exposed groups receiving MLE at doses of 200, 316, and 500 milligrams per kg BW. EG was administered at 265 milligrams per kg BW, and the treatment period lasted for 21 days. And the MLE was prepared using the maturation method with 96% ethanol as the solvent. At the end of the experimental period, the animals were euthanized and the testicular tissues were collected. And the tissues were then examined histopathologically using hematoxylin and eosin, staining with particular attention to spermatogenic cell necrosis. And now I will move to next main findings of our study. So, for our results showed that administration of MLE reduced spermatogenic cell necrosis in EG-exposed rats. Among the tested doses, the 500 milligrams per kg PW dose demonstrated the greatest protective effect. This finding suggests that MLE administration was associated with improved preservation of spermatogenic cell integrity following EG exposure. Therefore, the highest dose evaluated in the study showed the most prominent protective response. And to better visualize this protective effect, we also evaluate the testicular tissue histopathologically. Let's move to the next slide. So this slide presents the histopathological findings of the testicular tissues. In the EG-exposed condition, we observe more pronounced cellular damage, particularly involving the spermatogenic cells. Following administration of MLE, the tissue appearance showed an improvement compared with the EG-exposed condition, and this protective pattern was not apparent at the 500 milligrams per kgW dose, which was consistent with our quantitative findings. And overall, these histopathological observations support the potential effect of MLE in maintaining testicular cellular integrity following EG exposure. Let's move to the next slide. So for the discussion, from an immunological perspective, the protective effect observed in the study may be related to the interaction between oxidative stress and inflammatory signaling. EG-induced oxidative stress may contribute to activation of inflammatory pathways, which can subsequently promote cellular injury. One relevant pathway is tumor necrosis factor receptor 1, TNFR1, which is involved in inflammatory and cell death signaling. However, I would like to emphasize that TNFR1 was not directly measured in this present study, and therefore we do not conclude that moringa directly inhibits TNFR1. Instead, TNFR1 associated signaling is proposed as a potential mechanistic framework that may help explain the relationship between oxidative stress, inflammation, and cellular injury. Next. So our findings can also be viewed within a broader series of the studies investigating the protective effect of moringa against EG-associated multi-organ injury. In the lung, related research demonstrated reduced interalveolar septal thickening and alveolar congestion with supporting evidence involving TNFR1 and caspase-3 related pathway. At the center, in the kidney, related findings showed reduced malondialdehyde or MDA, increased superoxide dismutase activity, reduced calcium oxalate crystal formation, and improved renal histopathology. And now, in the testes, which is the focus of the present study, we observed the reduced spermatogenic cell necrosis and preserved the testicular cellular integrity. Taken together, these findings suggest that moringa may provide multi-organ protection against EG-associated oxidative and inflammation-related injury. And importantly, these integrated findings also provide a basis for further investigation of the immunological mechanism underlying the protective effect. Next. So in conclusion, the MLE attenuated EG-induced spermatogenic cell necrosis in male rats. Among the tested doses, 500 milligrams per kgW demonstrated the greatest protective effect. And this finding support further investigation of moringa as a natural antioxidant and potential immunomodulatory agent against oxidative stress associated tissue injury, particularly in the male reproductive system. And next. That concludes my presentation. I would like to express my sincere gratitude to the conference committee and to all of you for your kind attention. Thank you.
Okay, thank you very much. So we are going to move on to the discussion session. Prof. Fiska, would you like to first give the answer?
Thank you. So Farrell, good study, yeah. My question is, how did you and your team decide the 3 different dosage, yeah, and the reason, yeah, of these 3 different dosage, and then tell us in a short way, yeah, the pattern of immune response in your study, the inflammation, yeah, response in each treated group. What kind of pattern that you can see after your study has been done? Thank you.
Okay, thank you, Prof, for the question. So basically, the three Moringa doses, which is the 200, 600, 16, and 500 mg per kg, we received that from the previous study. And for the immune response, we did not directly measure inflammatory or immune markers in the present study, but therefore we cannot describe a specific inflammatory pattern for each treatment group.
Thank you very much, Prof. Fiska. Prof. Tere, the time is yours, Prof.
Okay, thank you, Dr. Ayu. I have two questions. As we know that Moringa oleifera has many component active. So which one the specific component active that can protect the spermatogenic necrosis?
Okay, thank you, Prof, for the question. One of the major or the specific bioactive compounds in Moringa is quercetin, as far as I know, and also flavonoids with strong antioxidant and potential anti-inflammatory properties. And the other important compounds, such as the camprefol and phenolic compounds, that may relate to these findings of our study.
Okay, and the last question. You mentioned that the Moringa oleifera will protect the necrosis in the spermatogenic. How can?
Okay, thank you, Prof. So Moringa may protect the spermatogenic cells through its antioxidant activity. Its bioactive may help reduce oxidative stress and protective cellular structure from its oxidative damage. And if I'm not mistaken, the antioxidant can menyatukan dan menghubungkan bagian yang nekrosis itu, jadi nekrosisnya menjadi hilang, Prof.
Okay, thank you.
Okay, so that would end the session for Mr. Farel Arif Muhammad. Can we give him a round of applause? So we are going to move on to the next session with Miss Larasita Prameswari. She will be presenting in this room. Miss Larasita, okay. So can you move forward to the front seat? Humas bisa dibantu untuk PPT-nya? Okay. The time is 7 minutes for your presentation and we will stop you if you're not finished. Ms. Larasita, you may start.
Thank you, Mrs. Ayu. Good afternoon, everyone. My name is Larasita Prameswari from Immunology Program Postgraduate School Airlangga University, Surabaya. Today, I would like to present my systematic review about effects of Moringa oleifera on gut microbiota modulation in experimental ulcerative colitis, a systematic review of preclinical studies. Next. Ulcerative colitis, or UC, is one type of inflammatory bowel disease that characteristic by chronic, relapsing, and persistent inflammation of the colonic mucus. and several pathology features involving dysregulated intestinal immune response, gut microbiota dysbiosis, impaired intestinal epithelial barrier, and excessive pro-inflammatory signaling. For the current therapies can control the inflammation and the progression of the disease, but treatment response varies on each individual, and long-term therapy may be associated with adverse effects of corticosteroid usage. Next. These figures illustrated the interaction between gut microbiota, intestinal barrier, and the compare of healthy intestine and intestine in UC. The gut microbiota is play central roles for maintain the intestinal barrier and homeostasis of the gut. But in the UC, gut dysbiosis and loss of tight junction of intestinal barrier can penetrate microbial and antigen stroke the intestinal barrier and leading the activation of cytokine and leading the activation of inflammation for the long time. Next. Moringa oleifera is a medicinal plant rich in diverse bioactive compounds and including polyphenols, flavonoid, polysaccharides, peptide, and isothiocyanate related compounds. Preclinical studies suggest that Moringa oleifera may exert first anti-inflammatory effects, antioxidant effects, and it can be modulating the gut microbiota by restoration of microbial diversity, enrichment of gut microbiome, and reduction of potential pathogenetic taxa. And then Moringa oleifera can be a protect barrier like increase zonal occludin 1, Claudin 1, and preservation of intestinal mucosal integrity. Next. Also, several preclinical studies have investigated the effects of Moringa oleifera in experimental colitis, but the evidence remain dispersed across different formulation and proposed mechanism. So this systemic review aims to summarize current preclinical evidence immunomodulatory effects of Moringa oleifera through gut microbiota modulation in experimental UC. Next. For the systematic literature review, I was conduct in PubMed, Scopus, and Web of Science to following 2020 PRISMA guidelines. Preclinical studies investigating the effects of Moringa oleifera on gut microbiota and immune response in experimental UC were included. The data on gut microbiota composition, inflammatory response, intestinal barrier integrity, and underlying mechanism were synthesized narratively. Next. This is the PICO of my systematic review. This is for population is experimental models of IBD or ulcerative colitis, including animals and intestinal organoid models. For intervention, Moringa oleifera or bioactive components or extract. For comparison, control groups yang receiving no Moringa intervention, placebo or disease-induced treatment alone. For outcome, I want to see the improvement of inflammation, intestinal barrier integrity, gut microbiota, oxidative stress, disease activity, and histopathology change and underlying the molecular mechanism. Next. This is for the system for the database. Next. For the result, I found from PubMed 25, Scopus 15, and Web of Science 40. And then after remove duplicate and ineligible records, I've screened 37 papers, and then I screening based on title and abstract. So I got 8 paper, and then I screening again, and I found 2 papers not retrieved. So I got 6 studies that include the systematic review. Next. This is for the result of the study. Most of the study use the different models of the ulcerative colitis and different intervention or exposure. This is for the assessment or method. So mostly they assessment the inflammatory gains and histopathology, cytokine, and metabolomic of the microbiome. And the main finding, mostly Moringa oleifera can reduce the inflammatory and oxidative stress, and then alleviate inflammation and intestinal barrier disruption by inhibit GAK state signaling and modulate the gut microbiota and lipid or amino acid metabolism. Next. For the component of the Moringa oleifera in the research I got is astragalin. Astragalin is the one component of polyphenol that can be found in Moringa oleifera that improve the disease activity and colonic injury. Then they found that can be improve intestinal barrier barrier function and modulate gut
Miss
dysbiosis.
Larasita, we are very sorry, your time is up. So we are going to move on to the discussion session. Prof. Fiska, would you like to ask first?
Yes, this is a very good systematic review, I think, yeah. So sorry that your timing is off, yeah, so we cannot see the discussion. This is the minor point from your presentation. My question is, all in all, yeah, if you already, because you haven't presented the discussion, yeah. My question is, what is the conclusion, yeah, from your systematic review, yeah?
My conclusion is Moringa oleifera has promising from the future to be the one of microbiota targeted therapy, but there is preclinical studies only. So I think it can be continue to be the well-designed human studies to be see the efficacy effectivity on the gut humans.
Be very careful if we are talking about any drugs, yeah, or chemical compound. You have to selectively use the term, yeah. Efficacy and effectivity are different, yeah. In the pharmacokinetics, different in the pharmacodynamics, yeah. Thank you.
Prof. Teresia, the time is yours, Prof.
Tes. Oh, tes bisa ya? Okay, thank you, Dr. Ayu. Larasita, according to your presentation, is it the Moringa oleifera as a microbiota? You mentioned that as a microbiota, or as a anti-inflammation in the ulcerative colitic? Which do you choose it?
In my systematic review, Moringa oleifera acts to balancing to reduce the dysbiosis. So inflammation is one of the consequences of the ulcerative colitis, but gut microbiota is the one of factor that maybe influence the inflammation of the ulcerative colitis.
Yes, okay, thank you.
Okay, thank you very much, Miss Larasita. So that will conclude your session. Give a round of applause to Miss Larasita Prameswari. We are going to move on to our next speaker, Miss Alvia Andriani, that will be presenting on site. Miss Alvia, not yet present. So maybe while waiting for Miss Alfia's confirmation, we are going to move on to our next presenter, Miss Maria Evata Krismawati. Would you mind to sit on the front? You may start your presentation and you have 7 minutes. Miss Maria, the time is yours.
Okay, thank you, Bu Ayu, for the time, and good afternoon, the panelists, judges, and everyone. Maybe Humas can help share the screen. Okay. I'm Maria Evata, and now I will present our study with the title, The Pathogenesis Analysis of Salivary Gland Tumors Through the Expressions of Programmed Ligand 1 or PDL1. Next. Okay, so as we know that pleomorphic adenoma or PA is the most common salivary gland tumor. It approximately 45 until 75% of salivary gland tumor, and this PA generally considered benign, but it may recur, metastasis, or undergo malignant transformation into carcinoma ex pleomorphic adenoma or CAXPA. And also the other tumors, the second highest is Warthin tumor, is another common salivary gland tumor and characterized by prominent lymphoid stroma, because it contains prominent lymphoid stroma and may involve complex epithelial immune interaction. And we know that immune checkpoint inhibitors like PD-1 and PD-L1 is can help tumor cells evade immune surveillance, and it's a relevant marker for studying this tumor because we can assess the diagnostic, prognostic, and therapeutic markers for tumors, but has not been widely studied in salivary gland tumor. So next. So in this study, we try to evaluate the distribution and cellular localization of PDL1 in PA, CAXPA, and Warthin tumor, and to explore its possible relationship with immune escape and tumor progression. So in this study, we use the method with the design observational study, and the sample PA is 22, CAXPA 5, and Warthin tumor 5, and one positive control of head and neck squamous cell carcinoma. And in this study, we use marker anti-PD-L1 rabbit monoclonal antibody by EpCAM at a dilution of 1/100. And for the assessment, we assess the membranous and cytoplasmic tumor cell staining, and also lymphoid stroma and surgical margin staining. For the review or analysis, PD-L1 expression was evaluated using Olympus microscope and photograph using Optilab Mykonos by 3 observers. Next. Okay, so in this study, we only assess qualitative assessment of PD-L1 expression by immunohistochemistry with 3 assessments. For the first is PD-L1 negative, it absence of brown staining on the tumor cell membrane with or without cytoplasmic staining in anti-PD-L1 immunohistochemistry. And the second is PD-L1 expressed. The meaning is presence of diffuse brown staining on the tumor cell membrane with or without cytoplasmic staining. And also the third is PD-L1 overexpressed, strong brown staining on the tumor cell membrane with or without cytoplasmic staining in tumor cells. And histopathological interpretation may not detect PD-L1 expression, positive PD-L1 expression in tumor cell, or high intensity PD-L1 expression. And this PD-L1 staining was assessed qualitatively according to the present distribution and intensity of brown immunoreactivity in tumor cells. And we got it from the protocols book of the antibody that we use in this study. Next. So the result is from 7 of PA from 22 sample show membranous expression. We can look at the histopathology picture in the right side. And 13 of 22 were negative. And also for 5 of CISPA show membranous expression, and 1 of 5 was negative. And also from the Warthin tumor, 2 of 5 of Warthin tumor show lymphoid stromal expression, and 5 of 5 is 100% expressed. But in Warthin tumor, no membranous expression. Next. Okay. Okay, this is the histopathological appearance from the positive control SSC and negative control normal salivary gland, and then the pleomorphic adenoma, CA-XPA, and Warthin tumor. Next. Okay, so the discussion. The most clinically relevant pattern was membranous staining in PA and CA-XPA. Although the CA-XPA group was small, membranous expression was more frequent in CA-XPA than in PA. This finding support the possibility that PD-L1 mediated immune escape is involved in malignant transformations, but the result should be interpreted as hypothesis generating association rather than the proof of causality. Next. Okay. In contrast, in Warthin tumor, demonstrate PD-L1 expression predominantly in the cytoplasm rather than in the cell membrane, and it shows cytoplasmic PD-L1 staining in all 5 cases and no membranous PD-L1. So it can be present that cytoplasmic PD-L1 may have a role in tumor cell growth and development. Next. Okay. So for the future study, it should maybe we conduct the ingest checkpoint in some ways as membrane localized PD-L1, so its therapeutic significance remains
your time is up.
Oh, yeah.
We are very sorry. So we are going to move on to the discussion session. Prof. Teresia.
Okay, thank you, Dr. Ayu. Maria, very interesting your case. I want to ask about your presentation. Yeah, is it you research about the salivary gland tumors?
Yes, Prof.
Okay.
Before, yeah.
Yes, and in your research, you got the squamous cell carcinoma also.
Yeah.
Yes, and then the pleomorphic adenoma.
Yeah.
Yes, and the adenoma.
CA carcinoma X pleomorphic adenoma. So it diagnosed
carcinoma
X pleomorphic
X,
adenoma.
oh.
Yeah, the recurrent of pleomorphic adenoma is carcinoma.
Okay,
Yeah.
okay. Yes, and in your research, which the tumor become to malignancy according to your research?
Which, sorry, Prof. Which tumor?
Which the tumor? Because squamous cell carcinoma, it is malignancy.
malignant,
Okay, as a control. Okay, and which the tumor about the pleomorphic adenoma or
the pleomorphic adenoma.
Which the tumor become to malignancy according to your research?
Who become the malignancy is the diagnostic before of the medical record is pleomorphic adenoma. So
Why?
Oh, why?
Yes, why?
Okay. Because in our studies, we find that maybe the PD1 or PDL1 in expressed in pleomorphic adenoma. So
bigger express, yeah.
Yeah.
Okay.
In some tumors, it can the expression can be overexpressed. So maybe the immune checkpoint inhibitors also express more, so the tumor can escape from the immune cells and then they can be malignant, right?
Okay, thank you.
Thank you, Prof.
Prof. Fiska, the time is yours, Prof.
Oh yeah.
We are going to take a small break while waiting for Adan.
Baik, ya. Bagus studinya ini bagus ya. Saya nggak menyangka ini dikerjakan ya sama adik-adik kita ya. Alhamdulillah. Pertanyaan saya, menurut Maria, karena Maria yang presentasi meskipun bukan first author ya, bias apa yang terjadi selama melaksanakan studi dan bagaimana cara tim Anda meminimalisir ya efek bias tersebut. Silakan.
Permisi Prof, boleh menjawab dalam bahasa Indonesia? Ya, baik, karena ini termasuk penelitian menggunakan imunohistokimia, jadi cukup challenging juga karena mungkin di Indonesia juga masih belum berapa banyak yang mencoba pada tumor-tumor di oral cancer gitu. Jadi mungkin untuk yang tricky itu sebenarnya untuk mengetahui konsentrasi berapa dalam pengenceran antibodinya itu yang nantinya bisa memunculkan imunohistokimianya gitu. Jadi sampai ketemu 1:100 itu memang butuh trial yang berkali-kali. Jadi kita motong jaringannya berkali-kali, membuat sediaannya itu juga berkali-kali, dan mungkin yang menjadi bias itu pewarnaannya ya, karena proses imunohistokimianya itu sendiri. Jadi memang kami berkali-kali mencoba dan yang kami lakukan trial terus-menerus itu Prof. Jadi supaya tidak bias warnanya ini benar-benar memang terekspresi atau memang ada defect, atau sebenarnya itu coklat-coklatnya itu bukan imunohistokimia ya, bukan hasilnya gitu. Jadi
istilahnya Mbak,
unspecific binding
bisa background,
ya, background
bisa
ya.
artefak,
Jadi
atau
mungkin
false positif ya?
Ya, false positifnya. Jadi mungkin pas antigen retrieval sama blockingnya itu kita lebih hati-hati sih Prof, karena dari situ kan epitop kebuka atau nanti di blockingnya itu benar atau enggaknya ya dari situ, bagian itu. Mungkin dari situ sih untuk mengatasinya.
Ya,
Prof.
Saya kira cukup. Terima kasih.
Okay, so that would end the session for Miss Maria Evata Krismawati. Can we give her a round of applause? Okay. Since our last presenter have not give any confirmation, so we would like to conclude the oral presentation session. But before this session is closed, Professor Fiska Sari will be giving some words to us.
Yeah. Thank you very much for each participant and the team to participate in our World Immunology Day International Conference the 5th 2026. Adik-adik semua sudah lihat ya. Apakah sulit adik-adik yang baru masuk? Enggak ya. Apakah menakutkan? Tidak. Apakah bahasa Inggrisnya harus perfect? Absolutely not. Ya, nanti adik-adik kalau misalnya presentasi ke luar negeri, nanti akan bertemu dengan berbagai peserta dari luar negeri yang bukan native speaker English. Sama pating pecototnya. Ya, dan kita juga enggak paham tuh ngomong apa peserta dari China terutama ya dan India, tapi kok ya percaya diri. Itu yang ingin ditularkan oleh Pak Yeni, Maria ya, dan teman-teman dari institusi yang lain tadi ya. Karena Anda sekolah WAJIB loh, sifatnya untuk mengikuti dan mempresentasikan karya ilmiah Anda. It is a mandatory actually to present your scientific research in a scientific forum ya, during your school period. Please ya, be confident ya. Learn a lot, have fun, and then I just want to remind you a minor detail. You have to put your co-authors. Please do not just put a single author. Yeah, for example, Mbak Laras, yeah, and Pak Yeni. It might be good for you to put your team, yeah, as your second or third and everything, yeah, the co-authors. So it's not normal just to put a single author name, ya, hanya mengingatkan untuk yang lain. Kesan saya yang lainnya sudah bagus banget, ya. Belajar terus tuh ya, nggak harus nunggu Anda bisa secara perfect. Itu saja ya, karena saya lagi jadi kepala sekolah, nuwun sewu ya Prof. Tere ya. Untuk yang lain, insya Allah 2027 ikut semua ya. Oke. Ini event diselenggarakan oleh Prodi untuk mahasiswa dan alumni supaya kalian tuh bersenang-senang, ya, unjuk gigi, ya, unjuk kemampuan. Nggih, matur nuwun. Saya kembalikan Bu Ayu, monggo.
Terima kasih banyak Prof. Fiska atas arahannya. Boleh kita tepuk tangan sekali lagi untuk semua presenter siang ini, Bapak Ibu. Untuk para juri saged bergeser untuk diskusi di ruang transit VIP Prof Tere dan Prof Fiska. So I would like to give back the time to Mas Nabil. Thank you very much, Mas Nabil.
Okay, thank you so much, Dr. Ayu, for moderating the oral presentation session, and also to Prof. Fiska and Prof. Tere for being wonderful judges in the oral presentation. So we invite you once again to go to the discussion room and discuss who will be our winners. Dan untuk para peserta, saya memberikan kesempatan nih untuk bisa menang hadiah mini award lah. Mini prize karena silakan Prof, terima kasih. Kami membuka kesempatan untuk 2 peserta bisa maju dan memberikan kesan-pesan selama mereka mengikuti WICID tahun ini. Mungkin siapa yang berani maju? 2 orang aja. You will get a prize. Sudah disiapkan sama panitia barusan. Atau saya tunjuk aja, oh masnya yang di belakang boleh mas, silakan. Ini langsung 2 aja atau satu-satu dulu? Satu-satu dulu, oke mas. So FYI, because this is an international conference, let's do it in English. All right, come up here. Mungkin ini sih di tengah dong Mas. Angle-nya sudah baik, sudah bagus. Ini saya nggak kelihatan gendut kan Mas, Mbak. Oke, mungkin bisa dimulai dengan perkenalan diri.
Hello everyone, my name is Hanif. I'm from Faculty of Medicine, Airlangga University. The student from Prof. Fiska Sari. He is the lecturer there, and in the first semester, he teach us about anatomy. The lecturer said the anatomy is the most difficult study. Okay.
And what do you think of this conference?
Okay, this is a great moment for us because in the first, Airlangga University is the go international university institution. So if we didn't meet this event, I think, wow, it's just only a title, not the real project. And I think because we have a lecturer from Mahidol and from University Malaya, she and he can know more about Indonesia. Wow, Indonesia is a great nation. And
This conference was to be held again next year.
yeah.
Come again?
Yes, of course, because my home in Surabaya, and I think Airlangga University is near my home. BTW, my home is in Karangmenjangan.
In Karangmenjangan, oh, it's very good. What semester are you in?
Fourth semester.
Fourth semester.
Yes, and people say Akam sih, anak kampung sini. Okay. Okay. Thank you for.
Also fun fact,
Yes.
I'm a dentist recently.
Wow.
So we are neighbors.
Okay, great.
And I've been taught anatomy by Prof. Fiska and it's quite difficult.
Okay, yes.
So as promised, we have a special prize.
Okay, thank you.
I don't know what is inside, but thank you for joining our conference today. Let's take a quick picture together.
Okay, I think it's very heavy, real good, I think. Because the price is rising right now.
Yes, unfortunately.
Yeah. Okay. Thank you for everyone. Oh yeah, my name is Hanif. Okay.
Thank you, Hanif. Segampang itu loh, guys. Ayo, siapa? Satu lagi mau? One more. Mungkin cewek, monggo. Ini Mbak yang hitam-hitam. Kayaknya mukanya familiar juga deh. Mbak Asti. Silakan Mbak Asti, silakan. Ayo, come here. It's a very simple task. Yes. Bagaimana tim humas? International conference. Underline international. Huh? It's okay, Blibad. It's okay. Gencana. Okay, so can you tell us your name? What do you study? Where are you from? Your nomor rekening and also your nick? No, okay, I'm kidding. And also, why did you decide to join the conference this year?
Okay guys, my name is Astika Juniansari. Now I'm teaching in Polytechnic Indonesia Surakarta. I'm baru ya, a new teacher in Polytechnic Indonesia Surakarta. I teach D4 analis kesehatan, teknologi laboratorium medis, gitu.
Okay, and why did you decide to join this year?
Because I'm with my student make a video for the the Reels competition in immunology World Immunology Day.
What do you think of the conference today? Was it exciting? Did you learn anything new?
Yeah, of course. In uh, setiap in every years we make this event, and so very apa ya, maksudnya kayak excited pastinya setiap in this years maybe. Oh, yeah.
If there was World Immunology Day 2027, would you join?
Oh yeah, of course. Maybe I will bawa my student. Oh yeah, bring my student in Airlangga University. Okay, thank you so much.
Okay, and as easy as that, we've got another prize for you. Wow, a keychain. It's very special. You should attach it to your bag right now. Okay, thank you so much for your time. I have no more prizes, but perhaps I would like to invite one more person to come up front to give their testimony. Ah, yes, the woman wearing the black blazer, please come. I have no prize, by the way, so this is all ikhlas. Lo, 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 lo. Ingat, Tuhan mencintai seseorang yang ikhlas, gitu. Okay, so please tell us your name. Where are you from? What do you do? What do you study if you don't work? And why did you decide to join the conference today? Why did you decide to join the conference? Okay, and out of the four speakers here, so Professor Erwin, Professor Duncan, Dr. Nuhafiza, and Dr. Adi, which one was your favorite that brought the best topic, in your opinion? Hmm? Okay, and because we're talking a lot, so I'm gonna ask you more questions. If you were to join, if this conference was there again next year, 2027, the sixth WICID, would you choose to join again? Inshallah. Oh, why? Oh, okay, okay. Next year. Okay, so you'll definitely come next year, right? I mean, okay, then I'll see you then next year. It's your last. Yeah, even if it's your last semester, you can still join, right? I will remember this face. Inshallah. Okay. All right. Thank you so much. What was your name again? Sally. Okay, thank you so much, Mbak Sally, for your time. Oh, see? When you ikhlas, pahala comes soon with rezeki. All right, look at the camera and smile. I don't know which smile though. Okay, yeah, you can give this to me. Oh yeah, I'll give this to you. Okay. You know, while we are still waiting from the organizing committee, previously we heard from Prof. Viska about the oral presentation, right? But now I would like to ask Prof. Tere, menurut Prof, bagaimana conference hari ini and what do you think of all the oral presenters today?
Very, very interesting. Dan apa ya? Wow, gitu ya. Ternyata risetnya bagus-bagus ya. Jadi supaya memotivasi yang adik-adiknya, gitu ya, supaya punya riset-riset juga yang ya recently ya. Jadi banyak yang baru. Jadi saya senang sekali. Ya, itu aja. Ya. Jadi kalau ada seminar seperti ini internasional ya, terutama internasional ya, tapi nasional juga sekalian enggak apa-apa. Ikutilah ya, karena itu merupakan pengalaman untuk kalian. Jadi nanti kalau jadi master ya, sudah jadi master itu bukan cuma ilmuwan saja yang berada di balik buku atau di balik laptop, tapi juga harus bisa mendiseminasikan hasil-hasil dari risetnya. Minimal orang lain juga bisa merasakan kepakaran kalian nanti kalau sudah lulus. Itu harapan saya ya.
Banyak Prof. Tere dan mungkin Prof. Viska ada tambahan kata-kata nggih untuk teman-teman peserta oral presentation? Sudah ya tadi nggih. Nggih bener. Bener juara ini apakah sudah siap dari teman-teman panitia? Okay, ladies and gentlemen, it is now time to announce the winners of the Reels competition, not the oral presentation, the Reels. How many of you made video Reels today? How many of you made Reels for this competition? Who joined the competition? Siapa yang jadi peserta Reels? Ada nggak yang di sini? Loh ini nggak sih? Banyak nggak sih? Loh. Oke, mungkin sekarang langsung saja kita umumkan. Nanti akan ada 3 juara harapan serta 3 juara. Of course, I understand making the videos must be very hard. I myself as an editor know that editing videos is extremely difficult. Trying to come up with a concept, coming up with a script, and it's time for you to see if your hard work has already paid off. Okay, now these are the finalist reels that we will see together before I announce the winners.
20%, kamu pasti cari charger kan? Tapi kalau kamu yang lagi lo baterai, pasti tubuh kamu akan dipaksa jalan terus.
Okay.
Padahal sistem imun kita juga butuh kondisi yang baik untuk bekerja secara optimal. Terus gimana kamu charger?
Look, the Immunology Reels competition winners. 6th place goes to Drum roll, drum roll. Sixth place goes to the Imun Imut. Are you here, Imun Imut? Wildan Abdul Farel, Clavia Evatina Dimulian, and Ramadeva Octaviana. Any of you here? Not here? Okay. We will now move on to the second honorable mention. The second honorable mention of the Reels competition goes to Yeni and Fams by Yeni Purnama, an immunology student here at the postgraduate school. Are you here? No, she's not here also? Online? Okay. The first honorable mention goes to Team Imin. Revado, Mila, and Ayu, are you here? No, you're not? Okay. Ladies and gentlemen, it is now time to announce the top 3 winners of the Reels competition. Are you ready to hear the winners? Yes, I can't hear you. Are you ready? Yeah, okay. Third place of the Reels competition goes to Andi Farhadipa Fashar. Are you here? You're here? Okay, please come up. Next, I would like to invite the Immunology Masters Program Coordinator, Prof. Visma. Oh, next. Okay. Stay there, alright? Okay. Second place of the Reels competition goes to TLM Imun by Astika, Mailana, and Renda. Are you here? Maybe a representative? Yes, please come up. Congratulations, Astika. Okay. The first place winner of the Reels competition. This goes to a person who has edited it for hours and hours and has gained the most likes perhaps. Without further ado, first place winner of the Reels competition goes to Fox P3 by Iqbal Felia and Nia. Are you here? You're here, right? Yes, please come up to the stage.
Timnya juga semua maju toh timnya ya. Timnya yang hadir maju semua. Ayo, nggak apa-apa ya.
The teams of the winners, can you come up? Okay. Now I would like to invite the Immunology Masters Program Coordinator of the Postgraduate School of Universitas Airlangga, Professor Viska, to please come and present the prizes to our winners. First award goes to the third place winner of the Reels competition. Now I would also like to invite Professor Tere to come up and also present the second place prize to our winner. Professor Tere as the head of the Oral Pathology Division of the Faculty of Dental Medicine, Universitas Airlangga. And next, the first place prize will be presented by Professor Teresia. Okay. And also by Professor Viska. Ladies and gentlemen, these are the winners of the Reels competition. The teams who have worked hard to edit hours and hours to create meaningful and educational videos in the field of immunology. Okay. All right, everyone, please look at the camera and smile. 1, 2, 3. Smile. 1, 2, 3. Can we have Uner hebat? Yes. 1, 2, 3, and smile. Look at the camera in the middle by Mas Dani. One more? Okay. Okay, thank you so much to Professor Terry and Professor Visca, and also to our winners of the team of the Reels competition. You may now return to your seats. Okay, the Reels competition have already been announced. What is missing? Should we just go home right now? Or how about we look at the videos of our finalists? Please enjoy.
Nah, kalau baterai kamu sisa 20%, kamu pasti cari charger kan? Tapi kalau kamu yang lagi low baterai, pasti tubuh kamu akan dipaksa jalan terus. Padahal sistem imun kita juga butuh kondisi yang baik untuk bekerja secara optimal. Terus gimana kamu charger guardia? Nah, yang pertama itu kita harus tidur yang cukup. Yang kedua, makan bergizi yang seimbang. Yang ketiga, jangan lupa bergerak. Yang keempat, kelola stres. Dan yang terakhir itu yang kelima, jangan lupa vaksinasi. Tapi ingat, tujuannya itu bukan bikin sistem imun kita semakin kuat ya, tapi tujuannya agar sistem imun kita tetap seimbang dan dapat merespon dengan tepat. Kalau HP aja kamu charge, masa guardian tubuhmu enggak?
Bayangkan ada seseorang yang menjaga tubuhmu 24 jam per hari. Dia nggak pernah tidur loh. Nggak libur juga. Dan setiap hari dia melawan jutaan ancaman. Namanya sistem imun. Sistem imun merupakan sistem pertahanan alami tubuh yang melindungi kita dari berbagai ancaman seperti bakteri, virus, mikroorganisme, dan berbagai macam penyakit lainnya. Tapi ada satu hal yang sering kita lupa. Kalau kita ingin terus dilindungi, kita juga harus menjaga sang penjaga. Nah, ini masalahnya. Lalu, bagaimana cara kita untuk menjaga sistem imun? 5 cara menjaga sistem imun. Yang pertama, makan makanan yang bergizi. Yang kedua, minum air yang cukup. Yang ketiga, selalu menjaga kebersihan. Yang keempat, tidur yang cukup. Dan yang kelima, selalu berolahraga. Jadi, siapa yang menjaga kita? Sistem imun. Dan siapa yang harus menjaga sistem imun? Kita!
Hai Sobat Imun.
Tahu nggak sih, sistem imun itu harus berada dalam keadaan homeostasis.
Kalau terlalu turun, jadi imunodefisiensi.
Kalau terlalu tinggi, jadi autoimun.
Tanpa kita sadari, justru kegiatan sehari-hari kita lah yang menjaga kondisi homeostatis itu.
Seperti tidur yang cukup. Saat tidur, tubuh memperbaiki dan mempersiapkan sistem imun untuk menghadapi hari berikutnya. Makan makanan sehat dan bergizi. Makanan bergizi memberi nutrisi yang dibutuhkan sel imun agar dapat bekerja dengan optimal. Olahraga jangan di-skip ya. Olahraga rutin membantu menjaga sistem imun tetap aktif dan tubuh tetap bugar. Stay hydrated. Jangan lupa banyak minum air putih. Hidrasi yang cukup membantu menjaga sirkulasi darah dan cairan limfa sehingga patroli sel imun ke seluruh tubuh lebih efektif. Biar anti stres, healing jangan lupa. Lakuin hobi yang kalian suka. Misalnya main game, baca buku, dan hangout. Saat stres, kortisol yang disekresikan lewat HPA axis meningkat, sitokin proinflamasi juga meningkat. Makanya harus tetap happy ya guys ya.
Tuh, mudah banget kan?
Yuk usahain hal-hal tersebut untuk jaga hemostasis sistem imun kita.
Healthy habits, healthy immunity.
Healthy habits, healthy immunity. I like that tagline. Gimana perasaan untuk teman-teman pemenang setelah lihat videonya ditampilkan? Kok mukanya pada merah-merah ya itu? Good job, good job. Nah, while waiting for the results of the oral presentation, I would like to ask a few questions to some of our oral presentation participants. Di sini yang jadi peserta oral presentation siapa ya tadi? Ayo ngaku. Di sana. Oh, Mbak, ayo ke depan yuk. Main-main sama Nabil yuk. kok aneh ya? Just a few questions I would like to ask about your feelings while presenting your research. Yes, come up. Yeah, come up. As I understand, you made a systematic review, right? Ini nggak ada mic lagi ya ini. Ini nggak ada mic lagi ya. Ini di sini, saya angle kanan saya, mohon maaf. Okay, can you tell us your name and what do you do? Where are you from? Your bank account details and your family name.
My name is Laresta Prameswari. I'm a student from Magister of Immunology program. I'm from Mataram, Lombok. My background is medical doctor. Yeah.
Okay, so how were your feelings when you presented your research?
I don't have feeling because my systematic review is I got the idea from my paper assessment from the first semester, and I interested in gut microbiota and oral gut access. Yeah, it's yeah.
When you get the papers and then you synthesize all the data, it's easy.
No.
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 Okay. But what do you think? How do you think that you did overall?
Gimana ya? Sebenarnya juga itu systematic review mendadak ya saya buat juga itu. Karena sebelumnya saya habis seminar juga sebagai presenter poster presentation di Jakarta, dan minggu ini langsung jadi presenter lagi. Kebetulan juga membahas soal gut microbiota, tapi untuk yang sebelumnya saya membahas tentang metabolic dysfunction associated with steatohepatitis yang masih relate sama testis saya. Gitu. Jadi saya mengolah kembali lah. Ya, mengolah kembali. Tapi masih tetap fokus ke gut microbiota sama penyakit. Iya. Agak gimana ya?
Tapi do you often present your research in conferences also?
Yeah, before. And tahun lalu. Ya. Iya, ini yang kedua.
Okay. Kalau semisal tahun depan ada lagi BJID 2027 scientific paper oral presentation, bakal ikut lagi nggak?
Tergantung temanya apa. Karena kan saya memang ada rencana mau daftar PPDS gizi klinik, jadi saya fokusnya ke imunologi nutrition. Jadi kalau misalkan memang ada membahas soal nutrition kayak yang di tahun ini tentang autoimun ya. Jadi saya coba bahas ke gut microbiota kan masih ke arah nih, masih ada hubungannya sama ini. Gizi klinik.
Gizi klinik.
Iya.
Di UNER juga?
Antara di UI sama UNER.
Selamat ya dok, semangat ya dok. Semoga lancar.
Amin, amin, amin. Amin, amin.
Terima kasih banyak ya dok. Thank you. You may now return to your seat. Okay, thank you. Okay, are we still ready? We are ready. Okay, ladies and gentlemen, it is time to go home. Enggak. No. It is now time to announce the winners of the oral presentation of the 5th World Immunology Day Conference 2026. I understand that from the feedback of one of the participants, making and presenting the paper was definitely not an easy task to do. It was difficult, but I am proud to see that you have all progressed together and have come to this moment. So, without further ado, let us now announce the winners of the scientific oral presentation. Oral presentation, the third honorable mention goes to Larasita Prameswari. Are you here? Always please come out. See? Your hard work paid off. Okay, the second honorable mention of the oral presentation goes to Farel Arif Muhammad. Are you here? Online? Okay, that's fine. Now, the first honorable mention of the oral presentation goes to Yeni Purnama, who is also joining online. Okay, I hope you guys are listening to this. Okay, ladies and gentlemen, the top 3 winners of the oral presentation. Makin deg-degan ya. Okay, third place goes to Ali Zainal Abidin. Are you here with us or are you joining online? Online as well, okay. Second place goes to Maria Evata Krismawati Surya. Where are you? You are here with us. Congratulations, please come up. Okay. First place of the oral presentation of this year's World Immunology Day Conference goes to 3, 2, 1, Muhammad Akila Herdiansyah. Are you here with us? Joining us online? Okay, that's okay. Okay, I would now like to invite Professor Viska and also Professor Tere to come up front and present the awards of the oral presentation. The first award will be presented by Professor Tere as the head of the Department of Oral Pathology and Maxillofacial of the Faculty of Dental Medicine, Universitas Airlangga. Next, the next award presented by Professor Viska as the head of the Immunology Master's Degree Program of the School of Postgraduate of Universitas Airlangga. All right. Ladies and gentlemen, please. Okay, please look at the camera, everyone, and smile. 1, 2, 3. Once again, 1, 2, 3. Can we have Uner hebat? 1, 2, 3, and smile. Now for the Imun Gaya. Once again, congratulations to our winners of the oral presentation. Okay, ladies and gentlemen. Okay, ladies and gentlemen, the announcement of Imu Uner post and oral presentation winners today marks the end of the conference this year. And on behalf of the entire organizing committee, we would like to thank you all for your participation this year. And on behalf of the organizing committee, we apologize for any mistakes and any problems that have been made during the conference. And I, Nabil Wafiuddin, as your MC, bid farewell to all of you and also to all of the online participants. But before we end today's conference, let's take a group picture outside. Okay? So don't go home yet. Once I close today's session, let's take a group photo in front of the postgraduate school building. That's it from me. Thank you so much for your attendance today and see you next year. Wassalamualaikum warahmatullahi wabarakatuh. Baik, kami persilakan kepada seluruh peserta bisa langsung ngambil barang-barangnya dan mobilisasi ke depan gedung Pasca Sarjana untuk melakukan sesi dokumentasi. Cek. Baik, kami persilakan kepada Mas, Mbak, dan Bapak Ibu bisa langsung mobilisasi ke depan gedung Pasca Sarjana. Mari kita foto bersama. Dan untuk kenang-kenangan kita pada sore hari ini, tidak seperti kenangan dia yang sudah saya lupakan. Gak apa-apa, kenangannya sudah ada yang baru kok, Mbak, aman. Silakan Bapak Ibu bisa langsung merap ke depan gedung Pasca Sarjana, nggih.